So hello everyone, good evening. So just confirm me once if I'm audible. अगर मेरा आवाज आ रही है तो बताना एक बार. Okay, the video and audio both are clear. दोनों clear है? Okay, ठीक है. Thank you so much. Yes, good to go. So this will be your last and the final class for the plan bidding. जो plan bidding के लिए जो मैराथन हमने स्टार्ट किया था, the marathon that we have started for the plan bidding, this will be the last video and the final video for the plan bidding. लिख के दे देता हूं, एक बार याद भी कर लेना जितना हमने इस 3 क्लास में कवर कराया है, इस मैराथन में जितना भी प्लान बिडिंग के कॉन्सेप्ट्स कवर कराया है, मोर और लेस सब सारे क्वेश्चंस, सब कुछ क्वेश्चंस जितने भी आपके आईसीआर पीजी एग्जाम 2026 में आने वाले हैं लगभग इसी में आएंगे. ओके, सो एवरी पॉसिबल क्वेश्चन दैट माइट कम इन द एग्जाम विल बी इन द विद इन दिस मैराथन ओनली. बहुत सारे स्टूडेंट्स पूछ रहे थे, इतना सारे पर्सनल मैसेज आ रखे थे, सर मैराथन देखेंगे तो काफी है, सर पीडीएफ पढ़ेंगे तो काफी है. तो व्हाटएवर माइट बी योर क्वेश्चन, द आंसर इज वेरी सिंपल. अगर एट द एंड ऑफ द डे लाइक इस पॉइंट पे आके आप कुछ पढ़े हो या नहीं पढ़े हो कोई फर्क नहीं पड़ेगा, लेकिन आप मैराथन को बहुत अच्छे से देख लिया आपने, एंड इफ यू हैव वॉच्ड एंटायर मैराथन वेरी सीरियसली, आपने हर एक पॉइंट को बहुत अच्छे से नोट किया है और उसको पढ़ा है, उसको बाद में रिवाइज किया है, then definitely 80 to 90% of things are very clear. लिख के ले लेना, एग्जाम जब लिखने जाओगे, जब क्वेश्चंस आपके एग्जाम हॉल में आएंगे तो मैराथन सेशन से याद आएंगे आपको. ठीक है, कितना आपने पढ़ा है, कितना आप याद करोगे तो आपके ऊपर डिपेंडेंट है कि इसके बाद जितना ज्यादा रिगरसली आप रिवाइज करोगे, जितना ज्यादा इस पीडीएफ्स को ज्यादातर रिवाइज करते जाओगे, याद करते जाओगे, दैट विल डेफिनेटली हेल्प. ठीक है, मोस्ट एक्सपेक्टेड क्वेश्चंस प्लीज. देखो मोस्ट एक्सपेक्टेड सब कुछ हो गया, अब मोस्ट एक्सपेक्टेड का तो भूल ही जाओ. जितना हमने कराया था मोस्ट एक्सपेक्टेड दैट आर मोर देन सफिशिएंट, जितना क्वेश्चंस आपने सॉल्व कराया है हर एक सब्जेक्ट का, लाइक व्हाटएवर क्वेश्चंस दैट वी हैव सॉल्व टिल नाउ, जेनेटिक्स का हो गया, प्लांट पैथोलॉजी का, प्लांट ब्रीडिंग का, सीड टेक का, सब कुछ खत्म है. सब कुछ साइड पे रखो, कंप्लीटली फोकस ऑन मैराथन. जितना क्वेश्चन हमने ऑलरेडी रिपीटेड पीवाईक्यूज में भी कराया है, मोस्ट एक्सपेक्टेड करके एमसीक्यूज में भी कराया है, और कुछ क्वेश्चंस आप लोग मॉक टेस्ट में भी अटेंड भी कर रहे हो. सो दोज आर सफिशिएंट. उसको एक साइड पे रखो, उसको भी रिवाइज करते रहना. दूसरे साइड पे आप करेंगे मैराथन. मैराथन्स आर समथिंग जो हम कुछ नहीं सोचे थे, लाइक वी डिड नॉट इवन थिंक और वी डिड नॉट इवन इमेजिन कि इतना अच्छे से हम कर पाएंगे. लिटरली बता रहा हूँ, जिस तरीके से हमने इस बार मैराथन कराया है ना, लिटरली पूरा साल का कंटेंट आपको सिर्फ 3 क्लास में मिलना और उसको 5 से 6 घंटे में कवर करना, ऐसा बिल्कुल नहीं है. कि अगर आप मैराथन सीरीज को देखोगे, so it's not something that we have like only discussed few things and continue to discuss each and everything. No, we have literally discussed each and every point that is needed for your exam के लिए जो भी जरूरत था, जो भी नीड था, सब कुछ मैराथन में कवर है. मैराथन का डेफिनेशन हमने पहली बार प्रॉपरली जस्टिफाई किया है, जो कोई और नहीं कर सकेगा. राइट, सी अगर आपने मैराथन बहुत अच्छे से देख लिया ना, तो मतलब भी समझ रहे होंगे कि व्हाट आई मीन टू से, कि अगर आपने मैराथन सही से देख लिया है, सो दिस मैराथन्स आर वेरी डिफरेंट फ्रॉम व्हाट अदर मैराथन्स आर रनिंग. सो इस मैराथन में हमने जैसे मास्टर कोर्स में पढ़ाया था, बिल्कुल सेम टू सेम ऐसा ही आप लोगों को पढ़ाया है. आई गेस लाइक मोस्ट ऑफ यू आर नोइंग इट, एंड बहुत लोगों ने प्यार दिया, बहुत लोगों ने अच्छे अच्छे कमेंट्स किए और अप्रिशिएट किया. So I definitely appreciate your appreciation. इसके लिए बहुत खुश हूँ मैं. उसी एनर्जी के साथ मैं दोबारा आया हूँ. Like this will be the last marathon. प्लान बिडिंग के लिए फाइनल मैराथन होने वाला है, तो आज का सेशन भी आप सब लोग एंजॉय करो. दिस इज द वन टाइम मोमेंट, लाइफ में कभी नहीं आने वाला है. जितना आप प्रेशराइज फील कर रहे हो उतना ही आप एंजॉय करो इस मोमेंट को, ताकि ये वाला माहौल कभी नहीं मिलने वाला है. एंड एग्रीकल्चर एजुकेशन में हमने कुछ नया किया है, क्योंकि एमएससी पीएचडी जहां पे कोई कॉन्सेंट्रेट नहीं करता था, जहां पे कोई किसी के पास कोर्स नहीं होता था कि बच्चों को पढ़ाने के लिए, जहां हमारे टाइम पे यूट्यूब तो दूर की बात है, ऐसा कुछ स्ट्रक्चर गाइडेंस भी नहीं था. बट अभी अगर देखोगे 2026 में यूट्यूब खोलोगे तो आधा चीजें वहीं से कवर हो रहे हैं. आपको सेल्फ स्टडी में ज्यादा टाइम ही नहीं देना पड़ रहा है. स्क्रीन टाइम आपका बढ़ रहा है हालांकि, बट हाँ अभी 2026 में जस्ट लाइक एवरी अदर कॉम्पिटेटिव एग्जाम, जितने भी कॉम्पिटेटिव एग्जाम है, जितने भी बीड वाले एग्जाम है, उसी लेवल के कंटेंट आपको इस स्पेसिफिक एग्जाम में भी मिल रहा है. हालांकि बीड तो है नहीं, पीजी पीएचडी में लिखने वाले कम लोग हैं, उसमें पढ़ने वाले और भी कम लोग हैं, बट फिर भी चाहे वो वॉचिंग वाले 10 हो, 20 हो, 40 हो, 50 हो, लाइक व्हाटएवर माइट बी द लाइव वॉचिंग व्यूअर्स, बट स्टिल आपको बहुत बेस्ट क्वालिटी इसमें कंटेंट मिल रहा है. बट मैंने एक चीज बोला था, आई टोल्ड वन थिंग, आई डिमांडेड वन थिंग. मेरे तरफ से एक डिमांड था, मेरे तरफ से एक रिक्वेस्ट था कि अगर आप लोगों को जेनेटिक्स का मैराथन चाहिए, लाइक इफ यू वांट द जेनेटिक्स मैराथन फ्रॉम माय एंड, इफ यू वांट द सीड्स एंड मैराथन फ्रॉम माय एंड, मैंने एक चीज रिक्वेस्ट की थी, I wanted 100 comments on that course reveal video, but unfortunately आप लोगों ने कितने लोगों ने कमेंट किया मुझे पता है, लेकिन बट फिर भी बहुत सारे स्टूडेंट्स अभी तक कमेंट नहीं किए हैं, उसमें कुछ भी उनका इंप्रेशन नहीं है और कुछ भी नेगेटिव और पॉजिटिव वो कुछ भी कमेंट नहीं है। हालांकि अगर आप 4 से 5 घंटे का क्लासेस लगातार आप पूरा देख रहे हो, कंप्लीट डे एंड तक आप हमारे साथ हो, तो ऑब्वियसली दैट मींस लाइक वी आर गिविंग द बेस्ट क्वालिटी कंटेंट। फिर भी अगर कमेंट्स का बात आ जाए तो कहाँ है कमेंट्स, कहाँ है आपका अप्रिसिएशन? मैंने कल एक वीडियो निकाला था एक स्टेट गवर्नमेंट एग्जाम का, उसका व्यूज चला गया है 3.8 के विद इन अ डे। कल शाम को निकाला था, आज शाम पे ऑलरेडी 3.8 का व्यूज है। बट कोर्स सिविल वाले में कितने व्यूज बने हैं अभी? अभी उसमें 3.2 के बना है। सी आई टोल्ड यू लाइक व्हाटएवर एफर्ट्स दैट वी आर मेकिंग, पूरा साल में इतना एफर्ट्स हम कर रहे हैं, बट फिर भी आपकी तरफ से कितना रिस्पॉन्स आप कर रहे हो? दिल से सोचो ना कि कितना आप कर पा रहे हो, कितना आप नहीं कर पा रहे, लाइक हाउ मच यू आर गिविंग एंड हाउ मच यू आर नॉट गिविंग। ओनली वन थिंग वी आस्क फॉर, वी डिड नॉट मेक इट मेंबरशिप ओनली। देखो अगर बना सकते तो YouTube में जो मैराथन हम बना रहे हैं उसको मेंबरशिप ओनली कर सकते थे, क्योंकि इतना क्वालिटी इतना टाइम एंड एफर्ट्स तो हम दे ही रहे हैं, बट फिर भी हमने इसको कंप्लीटली फ्री रखा, क्योंकि लास्ट मोमेंट में जितने भी स्टूडेंट्स हैं सारे हमारे स्टूडेंट्स ही है। जितना MSc PhD करने वाले हैं, वी आर ऑल इन अ सिंगल सेम कम्युनिटी विद द सेम माइंडसेट, जो एग्रीकल्चर के लिए कुछ सोचते हैं, एग्रीकल्चर के लिए कुछ पैशन है, उसी के अकॉर्डिंग हमने इसमें कुछ भी नहीं रखा था, कंप्लीटली फॉर फ्री रखा था। बट फिर भी अनफॉर्चुनेटली इसमें कमेंट्स बहुत ही कम है। तो मैं सिर्फ लास्ट एक और चांस दे रहा हूं सब लोगों को, सभी लोगों को एक लास्ट आखिरी चांस दे रहा हूं। देखो मेरा लास्ट एंड फाइनल रिक्वेस्ट है आप सभी लोगों को, कि अगर आपको दिल से सब कुछ अच्छा कंटेंट लग रहा है कि A to Z सब कुछ अच्छा है इसमें, डेफिनेटली खुल के कमेंट करो, खुल के शेयर करो और खुल के सारे स्टूडेंट्स को पहुंचाओ, ताकि इसका व्यूज बनना चाहिए। क्योंकि अगर जहां पे अच्छा क्वालिटी कंटेंट है वहां पे व्यूज होना भी चाहिए ना, दैट इज समथिंग विच इज एक्सेप्टेबल। मैंने अभी लिंक शेयर किया है, आई हैव शेयर्ड द लिंक ऑफ द कोर्स रिवील। मुझे अभी के अभी ही कमेंट्स चाहिए। जितने लोगों ने कमेंट्स नहीं किया, जितने लोगों ने कमेंट्स किया, दोबारा एक बार करो, दोबारा एक बार शेयर करो। पता नहीं कितने लोगों ने इसमें व्हाट्सएप में शेयर किया होगा, कितने लोगों ने पर्सनली अपने स्टेटस में लगाया होगा, कितने लोगों ने पर्सनली अपने जूनियर्स को शेयर किया होगा। जूनियर्स होंगे, सुपर जूनियर्स होंगे, या फर्स्ट ईयर वाले हो, सेकंड ईयर वाले हो, सबके पास ये चीज पहुंचना चाहिए, इसका व्यूज बढ़ना चाहिए और इसमें कमेंट्स भी आना चाहिए। चाहे आप पॉजिटिव कमेंट करो या नेगेटिव कमेंट करो, आप पूरा आपकी मर्जी है, इट इज कंप्लीटली योर फ्रीडम। आपके पास पूरा फ्रीडम है, आप कुछ भी कर सकते हो, क्योंकि हमने कमेंट्स ऑफ नहीं करके रखा है। बट वी वांट समथिंग, वी वांट रिएक्शन फ्रॉम ऑल ऑफ यू। कोई भी बंदा अगर यहाँ पे आके 5-6 घंटे के लिए कंटेंट बना रहा है, 2 दिन 3 दिन रात बैठ के, दिन रात बैठ के, एट द एंड ऑफ द डे कुछ नहीं मिल रहा है, तो क्या मतलब हुआ? क्योंकि यू नो इट राइट, लाइक वी हैव टू कंटिन्यू दिस। एक साल का नहीं है, क्लाइमैक्स हो गया, पिक्चर खत्म है, नेक्स्ट ईयर हम कुछ और करने जा रहे हैं। वी आर डूइंग द सेम एंड सेम प्रोसेस एवरी सिंगल ईयर। बट आपके तरफ से अगर कोई रिएक्शन नहीं आ रहा है, कोई अप्रिसिएशन आ रहा है, दैट्स नॉट एक्सेप्टेबल। ठीक है, इसके बाद आपको देखो, मेरा प्लान बिल्डिंग कॉन्सेप्चुअल था, 3 क्लासेस में खत्म हुआ। बट इफ यू नो, जेनेटिक्स एक मेजर पार्ट है पूरा प्लांट साइंस में, जहां पे मेजॉरिटी ऑफ द क्वेश्चंस आएंगे, और कितना ट्विस्ट होके आएंगे, कितना डीप में आएंगे और क्या आएंगे, ये सबको ऑलरेडी पता है। यू ऑलरेडी नो इट, लाइक हाउ द क्वेश्चंस विल बी, हाउ मच efforts you have to make in that subject and how much efforts a teacher has to make in that subject। तो आपको ये जो पूरा जेनेटिक्स का जो भंडार है, अगर आपको चाहिए प्रॉपरली पूरा फाइन ट्यून, ऐसा नहीं ऊपर ऊपर से बताते जा रहा हूं और बाकी सारी चीजों को स्किप कर रहा हूं, चाहे आप मास्टर कोर्स का स्टूडेंट हो या ना हो, सबके लिए अगर आप चाहते हो कि हेल्पफुल हो, लाइक इफ यू थिंक दैट इट शुड बी हेल्पफुल फॉर ऑल ऑफ यू। एंड मेन चीज, हम इसको सिर्फ इंग्लिश में करना चाहते थे, क्योंकि हमारा कोर एक्सपर्टीज इंग्लिश में था। बट सी, नाउ एवरीथिंग हैज चेंज्ड बिकॉज ऑफ यू पीपल। हमने सब कुछ चेंज करा दिया आप लोगों के लिए। मैंने हिंदी, जहां तक मैं बोलता भी नहीं था, अभी देखो मैक्सिमम चीजें हिंदी में हो रहे हैं, राइट? सो दैट इज ऑल बिकॉज ऑफ द काइंड ऑफ रेस्पॉन्स वी आर गेटिंग। लाइक जितना रेस्पॉन्स हमें ज्यादा मिला, इससे ही हमने फाइन ट्यून करके इंग्लिश वाले ऑडियंस को भी रखा, हिंदी वाले ऑडियंस को भी कंसिडर किया, और कोर्सेज को दोनों में बनाया, और एनर्जी इतना डबल किया कि हिंदी में भी बता रहा हूँ, इंग्लिश में भी बता रहा हूँ। वी आर डूइंग इट इन बोथ हिंदी एंड इंग्लिश। यू नो एवरीथिंग राइट एट दिस मोमेंट, बट नो रेस्पॉन्स फ्रॉम ऑल ऑफ यू। आई नीड द मैक्सिमम रेस्पॉन्स, आपके तरफ से रेस्पॉन्स होना ही होना है। ठीक है, बुक्स था, पूरे साल में क्या किया हमने, पूरा साल में सब कुछ छोड़ छाड़ के, सब कुछ छोड़ छाड़ के फिर बुक्स में पूरा टाइम दिया, पूरा एनर्जी दिया, कितने लोगों को इसमें इन्वॉल्व किया, कितना और चीजों को सैक्रिफाइस किया, बट व्हाट वी आर गेटिंग एट द एंड ऑफ द डे। सो आई नीड इट फ्रॉम ऑल ऑफ यू। लाइक मैं एक ही चीज आपसे मांग रहा हूं, दैट इज रिएक्शन, दैट इज योर फीडबैक, दैट इज योर सपोर्ट। सो जितने लोग भी डन, ऑलरेडी डन बोल रहे हैं, मुझे पर्सनली पता है कि आपका नाम मैं पर्सनली जानता हूं, जितने लोगों ने कमेंट किया, क्योंकि हर एक कमेंट को मैंने फील करके पढ़ा है। बट बाकी लोग जितने भी स्टूडेंट्स अभी तक कुछ भी कमेंट नहीं किए हो, ना लाइक किए हो, ना शेयर किए हो, सो दिल से रिक्वेस्ट है, प्लीज डू इट, सो दैट वुड बी वेरी वेरी अप्रिशिएबल। वी विल टेक दिस platform to the next level, to the another level. When you come into your PhD, when you go into any other agricultural exam, जो definitely आप लिखोगे, we will ensure कि you will get no doubt the best quality content. यही हमारा सपना था, यही मेरा विजन था कि मैंने तो स्टार्ट ICAR PG से किया था, लेकिन I don't want to stop it here. We see anyhow we have reached that highest point जहाँ पे हम PhD level तक भी कोचिंग कराते हैं. So understand the kind of knowledge that our mentor should hold if someone has to teach for the PhD. If we have reached that level of PhD, which is the top-notch level, then we can literally work on every other segment of agriculture and provide you the best quality content. So this is possible because of the potential we hold, right? So for that to happen, we all need your support. We all need your encouragement. We all need the reach that we have to reach every single student who is preparing for this ICAR PG, PhD and the ASRP. Do it, do it, do it. So if you do it, like if I get the comments, like if the comments crosses like 100 comments right now or right from this moment, I'll come in the very next day continuing the genetic series. तभी आऊंगा जब आप लोग करोगे, आप दूसरे से कराओगे, चाहे आप करो दूसरे से कराओ, सब चीजें करो जो आपसे पॉसिबल है, एंड इट शुड रीच ऑल द अपकमिंग स्टूडेंट्स. आगे से कोई नहीं बताएगा कि सर हमें पता नहीं था प्लेटफॉर्म के बारे में, हमें बहुत लेट से पता चला. ऐसा बिल्कुल कोई बताना नहीं चाहिए. लाइक नो वन शुड से दिस, लिटरली दैट हर्ट्स अ लॉट फ्रॉम इनसाइड. सो अभिषेक, आर यू रेडी फॉर द मैराथन? तो वेट ही कर रहा है. सो टुडे विल बी द फाइनल, द क्लाइमैक्स मैराथन फॉर द प्लान बिडिंग. So cheer up for that. So finally हमारा प्लान बिडिंग खत्म होने आ रहा है, but still marathons are going on. उसके बाद भी marathons आएगा, and I am hopeful and I am very positive that आप लोग कमेंट करोगे and I will be coming up with the next video. Genetics का वीडियो से आऊंगा, ठीक है? Let's not waste the time. Let's start this session. And thank you so much for all the students. जितने भी स्टूडेंट्स अभी तक प्यार दिए हैं, जितने भी स्टूडेंट्स अभी तक कमेंट किए हैं, जितने भी स्टूडेंट्स अभी तक शेयर किए हैं, सबके लिए बहुत बहुत धन्यवाद. From bottom of my heart, I thank you people, but still it still need more encouragement. It still need more reach. That is only possible from your end. हमारे तरफ से कुछ भी पॉसिबल नहीं है. हमारे तरफ से एक क्वालिटी कंटेंट पॉसिबल है जो हम दिल खोल के दे रहे हैं. बाकी सब कुछ आपके हाथ में है. You are the power. Like you are the only one who can do this. Please support and please do it. Please reach the target so that I can come up with the genetics content. Thank you so much.
हेलो, गुड इवनिंग.
जो 100 कमेंट्स क्रॉस बोल रहे हैं ना, ये 100 कमेंट्स पहले हो गया था, उसके बाद बोला था मैंने 100 कमेंट्स क्रॉस करने के लिए. आज के बाद, इस टाइम के बाद आई नीड 100 मोर. दैट्स व्हाट आई एम सेइंग. ठीक है.
गुड इवनिंग, गुड इवनिंग. अरे इतने बोलने के बाद भी कमेंट नहीं आ रहा है, मतलब मैं पार्ट 3 करना है नहीं करना है क्या? मेरा वीडियो में कमेंट आ रहा है, मैं वो उसके बात करना, तो मुझे आई एम रियली हैप्पी. ओके, मैं क्या, हमारा वीडियो, मेरा वीडियो आ रहा है, मैराथन का वीडियो आ रहा है, मैं सब पे कमेंट आ रहा है, मैं तो पूरा खुश है. ठीक है. हम एक और वीडियो अपलोड किया था, वो लॉन्च वीडियो, उसका फीडबैक दे दो बस. ठीक है. As he told, we are not worried about positive or negative, just comment चाहिए था. ठीक है. इसलिए सारे देखता है ना स्टूडेंट्स, नेक्स्ट बैच स्टूडेंट्स देखता है, मतलब क्या चल रहा है इस चैनल में. ठीक है. उनको अभी थोड़ा पॉजिटिव वही आना चाहिए, इसलिए. ठीक है. ओके, देन if you want to talk about our session, this is final session part 3. ठीक है. धुरंधर 3 होने वाला है आज. सारे मतलब ब्रीडिंग मेथड्स तक कवर किया था. आज हम थोड़ा स्ट्रेस ब्रीडिंग देख के, उसके बाद पॉलीप्लॉइडी, डिस्टेंट हाइब्रिडाइजेशन, म्यूटेशन ब्रीडिंग, ठीक है, वो सारे देख के कवर करेंगे पूरा. थोड़ा जेनेटिक इंजीनियरिंग और थोड़ा मार्कर्स के बारे में थोड़ा इंट्रोडक्शन. ठीक है. वॉट एवर मे बी द पॉसिबल क्वेश्चंस आर द टारगेट एरिया ऑफ दिस पर्टिकुलर एग्जाम. ओके, according to that we will see each and everything. शुरू करेंगे. बताओ कुछ बताओ, कुछ बताना हो तो अभी शुरू में बताओ. सर आई एम अफ्रेड ऑफ यू. अरे भाई क्यों? How many hours it will take to complete this? I don't know. as I always tells. Okay, I don't know. Yeah, Pooja, I saw that my name is also written in the comment, but we want, okay, in my name, just take your time and go to that video, the course launch video. Okay, it is in the description box also, and you can search the new video which is we have launched. Okay, the journey of Agri Addict. उसमें डाल दो, मेरा नाम ले लो, उसमें भी आप कमेंट डाल दो बस. It starts, sir. I have given it, sir. The semester exams are juniors are going to, so I stopped themselves. Okay, with more gifts. Yeah, you will get gifts. No worries. Let's start. Yeah. 20 minutes gone. Arey Sinchan. Looking nice, sir. Thank you. Thank you so much. Anyway, now you already know about me very well. Okay, I don't want to repeat it again. Okay, I just want to tell you guys that this is my introduction. अभी तक अभी मैं इतना मानता हूँ सबको आप मेरा बारे में पता है, ठीक है? Before starting today's session, just I want to tell you one thing. Okay, one more thing. मैं ऐसे क्लास देख रहा था, कुछ यूट्यूब में आ रहा था, कुछ मैराथन चल रहा है, दूसरा उधर इधर भी चल रहा है. सारे लोग इधर कौन अटेंड करता है ना, उधर ही सारे लोग उधर भी अटेंड कर रहा है. मैं देखा था एक वीडियो ऐसे, ठीक है? थोड़ा एक में ज्यादा फोकस करो, एक चैनल को हमेशा फॉलो करो, ठीक है? वो तो थोड़ा बॉन्ड भी बन जाएगा, उसके बाद थोड़ा रेगुलर और वो भी हो जाएगा, ठीक है? मैं देखा था कुछ रतन और हमारा सिंचन, वो सब जगह में है, ठीक है? दोनों संजू चौधरी, अभिषेक सर द ब्रीडिंग मास्टर. थैंक यू यार. रोम ग्रीस, छोड़ दो यार, मैं जाके आ गया ऑलरेडी, ठीक है? सर हाउ विल द एंटायर सिलेबस बी कवर्ड? सिलेबस कवर कैसे होगा? अरे कैसे, हम कवर किया है ना. हु विल गेट इट डन? कवर किया ना हम, और क्या चाहिए आपको? ब्रीडिंग किंग अभी सर, ठीक है यार, ठीक है. First, we'll what we in the stress breeding we will classify into biotic. Okay, in biotic there are two major parts. That's one is your disease resistance and insect resistance. Then your abiotic, in that part too. Okay, the drought and heat are major thing. If they are targeting anything, okay, maybe they can ask the responses. Okay, physiological responses of the plants to the stress. Okay, maybe for the drought and heat. Okay, these are the four important stress in case of our crop plants. So we will start with first one. Okay, in biotic stress, okay, it is an stress is nothing and is any situation, okay, where the plants will get abnormal functions, abnormality. Okay, if I want to talk about any stress, that is a biological pressure placed on an organism, okay, by the other living organisms. If it is biotic stress, okay, if any biological pressure, okay, from by the other abiotic agents like your heat and drought, then it is abiotic stress. It refers to any external factor that prevent an organism from the reaching its maximum growth, reproduction and yield potential. Yield is nothing but yield, we can say in our term. Okay, any factor, okay, which prevents the particular growth, reproduction and yield, okay, then we will call it as a stress factor. The condition is nothing but stress. If we classify our biotic stress, then your airborne fungi, these are the major contributor for the biotic stress. Then your soil-borne fungi, okay, then your bacteria, virus, and at last nematode and insects. So we will, these factors, the fungi, bacteria and viruses will comes in the disease. Okay, we will call it as a disease. The infection, okay, we will call it as a disease. In case of these cases, nematode and insect, okay, the infestation from these two factors, okay, we will call it as a insect attack. Okay, it is yield loss due to disease. It is up to 20 to 30%. Okay, first we will target disease. We will see what exactly the disease is. It is defined as any physiological abnormality Okay, whether in case of you are also in case of you also, if fever comes, what we will tell? It is a physiological abnormality or disruption in a plant's normal, again, growth, development and reproduction. If anything which is interfering the growth, development and reproduction, okay, the factor we will call it as the stress factors, and the condition, the situation is, in this case, particular case, it is disease. Okay, what is disease resistance? It is the capacity of a plant to prevent or limit that particular growth of the pathogen. In case of disease, okay, it is a capacity of a plant to prevent that particular spores, okay, to get germinated or to get enter into the plant system. Okay, the inhibition phase, we will call it as a disease resistance. आवाज आ रहा है क्या सही सही से, और ब्लर हो रहा है क्या कुछ? बताओ. All clear. Okay. Before starting anything, as I told, we have discussed this particular thing in our live classes also. Okay, in our master course. This is very important. Before starting any stress breeding, okay, anything related to stress, first we should know what exactly the meaning of susceptible, tolerance, and your resistance and immunity. Okay, this is more important. It is based on the rate of reaction of your particular spore. Okay, rate of reproduction of your spores. Okay, how they are reproducing. Based on that, we will classify the range of reaction. Based on the range of reaction or the rate of reproduction of particular spores, we will classify the reaction into 4 steps. First one is your susceptible reaction, where your range of reaction is 1. That means the complete reaction will occur. For example, the host plant is unable to defend itself effectively against the pathogen. What happens here? The pathogen, that means you can take spores, any spores. In case of rust, your iridospores. What will happen? They will successfully infect the plant. Okay, then grows and reproduce that pathogen and cause the visible disease symptoms, often leading to the significant damage or death of the host. Okay, the normal procedure, the normal infection of the particular any disease. Okay, it will infect the plant, then it will grow, it will reproduce and cause the symptoms. Okay, often leading to the significant yield loss or finally crop death. This is your susceptible reaction, where your range of reaction is completely 1. Okay, then tolerance reaction. Okay, here also the range of reaction is 1. How we can tell that? The host plant become infected by the pathogen. Okay, and that pathogen does grow and reproduce within it, but the host plant is able to endure the pathogen presence and activity with a minimal or reduced disease symptoms and yield loss. In case of tolerance, again, your pathogen will infect and it grows and it will reproduce in the plant system, but the plant will having some the fight back nature, so that there will be minimum or reduced yield loss. Okay, and also the reduced disease symptoms. There will be a presence of pathogen and there will be a reproduction of the pathogen, but the plant will try to cope up with that particular disease symptoms, so there will be a minimum yield loss. This is your tolerance reaction. Again, your R is equal to 1. That means your reproduction of the particular pathogen is 1. Next, resistance. Your R is, okay, more than 0 and less than 1. Your R is less than 1 and more than 0. Okay. What will happen here? The host plant possess inherent genetic mechanism. there is some genetic mechanism. There are some genes within the plant to give some resistance reaction, actively prevent or significantly impede the establishment, growth and reproduction of the pathogen. The where the fight back will comes in place of the growth and reproduction of pathogen itself. Okay, there will be some barrier for the growth, for the establishment itself of the pathogen. So the fight back nature will be more in case of resistance, so that the reaction, the range of reaction or the reproduction of that particular pathogen will be less than 1. Okay, and it will be more than 0. There will be some reproduction, okay, but not completely like your the both above cases. Okay, so there will be, it is completely genetic mechanism. Okay, the genetic mechanism. Then your R is 0, that is immune reaction. The where, okay, the plant will completely avoid the, okay, it will completely avoid the first phase itself, that is establishment stage. The host plant is completely unaffected by the pathogen. It is due to hypersensitive reaction. Okay, where there are presence of your vertical resistance genes, okay, there will be a hypersensitive reaction. When your pathogen spore or any spores will lands on your leaf surface or anywhere in the plants where the immune reaction is present, okay, that area will be necrophyte, or it will be the death of that particular tissue, so that there is no further growth of that particular pathogen. Okay, it is completely due to hypersensitive reaction. Okay, your immunity, you should be remember. And the genes which are responsible for this particular reaction will be your vertical resistance gene. Okay, now you got this particular concept, right? The susceptible reaction where R is equal to 1. Okay, complete establishment of disease and complete death of the plant, host plant. Then there will be a tolerance reaction. Okay, the pathogen will grow, establish, and there will be a visible symptoms. But with respect to the yield and disease symptoms, okay, the minimum reduced disease symptoms and yield loss, okay, the minimum yield loss, the plant will try to fight back. In case of resistance reaction, your R is more than 0 and less than 1. Okay, and it is completely inherent genetic mechanism, and it prevents the significant establishment of a growth and reproduction of the pathogen. The intensity of a fight back nature will be more compared to tolerance. Then about immunity, it is a completely, okay, unaffected by the pathogen due to hypersensitive reaction. ठीक है, एक्सप्लेन किया ना अभी टोबाटो. ठीक है, this is about 4 reaction. Again, why I am taking too much time about this, they will definitely ask questions regarding this particular concept. If they want to ask from the disease part, then they will try to target from stress part, any stress part. Okay, we will use these 4 terms, okay, all over the disease, all over the stress breeding chapters. Okay, there is nothing to worry about it. If you know the concept now, then it is very easy to understand the breeding things. Okay, and wherever in case of biotic stress, we will always talk about resistance reaction. Okay, since we can completely, you can use genes. Okay, your resistance reaction is due to oligogenic nature, most probably in case of insect resistance, so that we can use genetic mechanism, that means your some major genes, resistance gene, so that we can avoid the pathogen. Okay, in case of biotic stress I am talking. But in case of abiotic stress, where we are not completely, we can escape from the disease, okay, where there is no complete fight back nature in case of your disease, there what we will do, we will use term tolerance. There will be always the stress will be appear, okay, it will be impact on the croplands, but in case of tolerance, okay, there will be minimum yield loss. So in case of your drought, in case of your heat, any abiotic stress, okay, we can go for tolerance mechanism. Okay, not completely resistance there. Since it is quantitative trait, okay, the tolerance, the in case of abiotic stress, okay, we can target tolerance mechanism. but in case of biotic we can target resistance mechanism. ठीक है, ये तो बहुत बेसिक है, ये तो आपको पता होना चाहिए. Now, okay, if we comes to see what exactly the physiological races, pathotypes and differential host. Physiological races are nothing but the strains of a single pathogen differing in their ability to attack the different varieties of a same host species. Okay, there are so many varieties in case of particularly wheat. Okay, the pathogens will target and they are having some ability to attack the particular varieties. Okay, we will name it as different physiological races. Okay, within the pathogens, okay, based on their ability to attack different varieties, we will classify them as a physiological races. Then what about differential host? This is term regarding to host species. The varieties of a host species to identify the physiological races. We are having different the reverse concept. We are having some different varieties. Okay, the each variety is okay attacking by different physiological races. To identify those different physiological races, okay, we will maintain some different varieties. Generally, resistance gene present in different hosts are not known. When we don't know the resistance genes, okay, that time we will call those different varieties as a differential host. Okay, this is regarding varieties, our host varieties. This is regarding the pathogen, different physiological races based on their infection. Okay. Next, pathotypes. These are strains of pathogen classified on the basis of their virulence to known resistance genes of the host. Now we are having some resistance genes in the host plant. Okay. Based on that, there will be some of the strains of a pathogen are virulence on that particular resistance gene. Some are non-virulent. Okay. We are at this particular time where exactly we are seeing, we are taking the resistance gene, whether our strain are attacking or not, that particular resistance gene. Based on that, we will classify the pathogens. That is your pathotype. Here, in case of physiological races, it is completely based on varieties. Okay, we don't know the resistance gene. Okay, whether it is attacking on SD3086. Okay, in case of this, some are attacking SD3086, some are attacking SD3389 of wheat 3386. Some strains are attacking 3385. Okay, we will classify them as physiological races. But here, in case of pathotype, now we are having a resistance gene R1, R2, and R3. Some pathotypes are virulence for this R1. Some are virulent for this resistance gene. Some are virulent for this, but not all the strains are virulent for R1, R2, R3. Based on this particular concept, we are classified them as the pathotypes. Okay, the pathotype classification is more precise than the physiological races. Okay, this is based on the varieties. This is based on the virulence towards the known resistance gene. What about super race? A pathogen that has virulence gene for every vertical resistance gene. Now, as I told, now I have 3 vertical resistance gene. Okay, your pathotype having virulence, particular virulence gene for all these 3 resistance gene. Okay, that in a variety, that race we will call it as a super race. Your UG99, okay, of particular race on wheat stem rust. Okay, whatever the resistance gene which are available for wheat stem rust, okay, there will be a particular virulent genes from that particular pathogen, the race UG99. That is actually Uganda rice. Okay, Uganda 99. So we will call it as a super race. Clear hai? You got difference, right, between the physiological races and pathotypes. This is important. Physiological races and pathotype. Clear? Batao? Yes, good. Then what about the difference between vertical and horizontal resistance? Chaitanya push race or repeat super race. Super race are nothing but, as I told, okay, if in any particular variety or any multi-line, you will use multi-line sometimes. If your multilines contain R1, R2, R3, or your variety contains R1, R2, R3, these are different vertical resistance genes for different races. Okay, we thought that we to avoid the different races, we have using 3 particular genes, but the single race of the pathogen contains virulence gene for all 3 resistance gene. Okay, for that race we will call it as a super race. Your UG99, okay, it is contains the virulence gene for all stem rust resistance gene. Okay, that's why it's very difficult to breed for those particular races. We will call that as a super race. Then what about the difference between vertical and horizontal resistance gene? Your vertical resistance genes are governed by one or few major genes. Okay, R1, R2, R3, like few genes. But your horizontal resistance is generally governed by minor genes. It is polygenic in nature. It controls from so many number of genes. This we can easily explain by gene for gene interaction. Through gene for gene interaction, you can easily explain the vertical resistance. But it is multiple, okay, generalized defense mechanism, your horizontal. It is highly specific, broad spectrum, complete or near complete. There is a complete range of reaction here in case of vertical. Here there is a partial resistance. It will give some partial resistance. Durability, it is unstable and short-lived. Okay, if the new race evolved from pathogen side, then the single vertical resistance gene will be failed. But in case of this, it contains multiple number of genes. Okay, if it got evolved also, there is a less degree of damage. It contains so many number of genes. It's very difficult to evolve that pathogen for the all different possible genes. Okay, that's why the durability is stable. Okay, it will be expressed in all plant growth stages from seedling to adult, but your horizontal will be expressed in adult plant resistance. If its resistance breaks, as I told, there is a possibility of epidemic, the severe epidemic. But in this case, the rate of disease spread will be reduced since it contains more number of gene. It rely upon the rapid deployment of resistance varieties used for gene pyramiding. Okay, gene pyramiding. It is sustainable disease management. Okay, or horizontal will be sustainable. Super race, gene pyramiding, both are. Array. Kavita Q super race, it's regarding the different race, the pathogen. Gene pyramiding is different. That is the mechanism. Okay, we are doing it. Gene pyramiding. Super race is nothing but it is infects. It infects all the other particular resistance gene since it is having virulence for all the genes. Okay, the gene pyramiding is a mechanism. Okay, where we are using the different genes which will be effective for all the races. That is different mechanism. Okay. There are some synonyms: race specific, qualitative, monogenic, oligogenic, major gene, hypersensitive, as I told. Okay, these are different synonyms for vertical resistance gene. Here the non-specific, it is quantitative, polygenic, okay, minor gene, partial resistance. If you know the meaning of these particular terms, okay, you can easily get a concept of vertical resistance and horizontal resistance. Okay. Then this is triangle. It is interaction between host, pathogen and environment. Okay. If the favorable environment and when there is a presence of pathogen and susceptible host, if these 3 are there, your disease will occur. Okay. If you have to have a pathogen first, then the susceptible host and the favorable environment, humidity, temperature. Okay, then there will be development of disease. Now the disease pyramid, the recent one, when the fourth factor, time. Okay, when, again it will matter. Okay, when in the season maybe. Okay, now the time is also important. They have included time also in the disease development. Okay, it can be represented by disease pyramid, from disease triangle to disease pyramid now. Okay. So now we will see what exactly the gene for gene hypothesis. विजय बता रहा है फिफ्थ फैक्टर ह्यूमन। ठीक है, पेंटागन बना दो पिरामिड से। मीन्स सुपर रेस काज डिसीज इन जीन पिरामिडिंग स्पीसीज। अरे भाई, gene pyramiding is nothing but when you will introgress more number of genes into single variety. Okay, whether it is gene pyramiding or not, if you are multilines, in case of multilines, how you can say it is gene pyramiding? Okay, whether it is gene pyramiding or it is a mixture of multilines or whatever it may be. Okay, in farmers field, one farmer is growing the resistance, the variety which is having resistance gene R1, the beside one is growing variety which is having R2, and someone is growing with the variety which is having resistance in R3, then if the pathogen race if attacks all three fields, then you will call it as a super race. Okay, maybe if your variety contains all three genes, at that time also it will attack. If the varieties which are having different resistance gene, that time also it will attack. Okay, why you are confusing, why you are you are introgressing pyramiding genes into this particular super race concept. Okay, as I told, whether it may be your pyramid gene, okay, pyramided variety, or multi lines, or the single pure lines. Okay, whatever it may be, you just what you should know, this in case of super race, it will attack, it is having the virulence gene for all other all is vertical resistant genes. ठीक है, बस उतना होना चाहिए। ठीक है कविता and for Shinchan. Now we will see what exactly gene for gene hypothesis. It is given by H.H. Floor in 1942, flax rust disease, that is Melamopspora leani. What he will exactly tell, what in case of the particular basic definition, for every gene controlling the resistance in the host plant. Okay, for every gene which control resistance in the host plant, okay, the resistance will be dominant in case of host plant, called resistance or gene. There is a corresponding specific gene controlling virulence in the pathogen. Okay, virulence, AVR, called virulence or AVR gene. This is the basic definition. How they will interact and everything, again there will be a different definition. The basic thing should be, for every resistance genes in the host plant, okay, there is a virulence, not virulence. There is an virulence which is dominant again. Okay, there is a virulence gene in the corresponding pathogen. You can see once again, for every gene controlling a resistance in the host plant, there is a corresponding specific gene controlling a virulence in the pathogen. This is the basic definition of gene for gene hypothesis. Okay, now we can see resistance reaction. If the host plant possesses a dominant R gene, if the host plant possesses a dominant R gene and the pathogen possesses the corresponding dominant AVR gene, okay, R gene and AVR gene, what will happen? What will happen? There is a resistance reaction. Okay, the plant will recognize the pathogen. Your resistance gene will produce some chemical and AVR gene will produce some chemicals. They will interact each other or they can recognize each other and they will produce a resistance reaction. Okay, this is when the resistance reaction occur, when it will occur, when capital R and AVR. What about susceptible reaction? If the host plant lacks the specific R gene, okay, small r, if the pathogen lacks the corresponding AVR gene, so that the small AVR, the plant will not recognize the pathogen. Since, as I told, this particular mechanism will be based on the interaction between the chemical produced from resistance R gene and dominant AVR gene, virulence gene. Okay, if they will produce chemical, they will interact each other and they will produce a resistance reaction. In all other cases, okay, there will be 4 cases, right? Capital R, small r. अरे क्यों आ गया? R gene or R gene in case of your post plant, AVR gene or small AVR gene. What we are dealing with, this is first mechanism of gene for gene hypothesis. Okay, when there is an R and AVR, as I told, there is a chemical. There is a chemical, so they will interact each other, they will recognize each other. So there is a resistance. Okay, resistance reaction. But in all other cases, okay, if it misses any, in these cases, there is no chemical for this particular capital AVR chemical. Since it is small AVR, so it is susceptible. Okay, susceptible. Here there is no chemical from this side, small r, so again susceptible. Again here both are missing, susceptible. Only in this case there is a resistance reaction. Okay, can anyone tell what we will call this rule? Anyone from the in the comment box, so that maybe they can get a gift. For this particular rule, we are having some term. Flora rule. There is no flowers. Flora rule. Flowers. अरे क्या डाल रहा है तू? कविता. Yes, anyone? Now you got this particular concept. Okay. अरे कहाँ चला गया? This one, when there is a resistance reaction, when there are all other 3 possibility, there is a susceptible reaction. We are having some term. We are having some term for this particular rule. Floor hypothesis. Yes, good Ratnesh. This is Floor's definition, the original definition. Also we'll call it as a original definition, and we will call it as a also we will call it as a I have expected this. अरे ज़्यादा हो रहा है slide. Also we will call this particular thing as a incompatible reaction. ठीक है, Floor's definition, incompatible reaction and original definition. Good Kavita, incompatible reaction. Then again there is another rule. We will call it as a rule of definition. You can see the rule of definition. Also we will call it as a compatible reaction. The allele for a susceptible are in the host. Okay, what it will do? It will explains the susceptibility. Okay, how susceptibility will occurs. Are in the host, and those for virulence, small AVR, in the pathogen produce specific molecules, which interact with each other and produce the susceptibility response. Okay, susceptibility. But in all, a lack of interaction between these products, okay, there will be a resistance response. Now you can see capital R, small r, capital AVR, small AVR. Only in this case, small r, small AVR. In that particular case, susceptibility. But in all other cases, okay, here it is explaining the susceptibility. In all other cases, there is a resistance. Okay, we will call it as a rule of definition. It is rule of definition. Both are given by Wheeler, Floor's original definition, and the rule of definition. Floor's rule, sorry, not definition. Floor's rule and rule of definition. Both are given by Wheeler. Okay. We will also call it as a compatible reaction. ठीक है, समझ गए ना? Both the rules, the original rule and the rule of definition. ठीक है, in case of original rule, it will explain how the resistance reaction will occur. Okay, that we will call it as an incompatible relationship. Okay, where there is an incompatibility, there is a susceptibility. But here, in case of rule of definition, okay, it will explain how susceptibility. Okay, when there is a absence of any particular chemical from any one gene, then there is a presence of resistance in case of your compatible reaction. ठीक है, समझ गया ना? बताओ. Simple thing. This is about your gene for gene hypothesis. There is nothing. Okay, there is nothing. The basic definition and the first rule, that is your Floor's rule. Then your second rule, that is rule of definition. Okay, what will happen in the both the cases? That's where what's in the slide. This is about your Floor's rule. Then what about vertifolio effect? The important concept again. Okay, the simple thing, there is a German potato variety. Okay, German potato variety, that is your vertifolia. Okay, they have developed this particular variety for bacterial blight resistance. It contains R3 and R4 resistance gene. Okay, whenever the farmers grow in the larger area, okay, the pathotype P3 and P4 got evolved. Okay, then there is an complete breakdown of this particular resistance. Complete breakdown and failure. The complete loss of an particular variety, okay, due to, or we can also tell that the complete loss of an vertical resistance due to the evolve of pathotypes P3 and P4, and we can tell that it is not a complete failure, or we can also tell that this is not due to the vertical resistance gene. Yeah, we are having vertical resistance gene R3 and R4, so P3, P4 got evolved. Okay, but since this particular variety only contain R3, R4, if it contains some horizontal resistance, then if the pathotypes got evolved also, okay, then there is no complete breakdown or failure of the particular crop. Okay, the particular vertifolio effect is not only about the failure of vertical resistance gene. It is due to, yes, Ranjan Kumar, good, continuous breeding for vertical resistance to the loss of horizontal resistance. That's what the important thing. विजय, आप तो साजन तो हमारा नहीं मानिए, ठीक है. विजय and Ranjan, you guys are our students. रिमी तो हमारा स्टूडेंट है. रंजन, मैं पूछ रहा हूँ, आप हमारा स्टूडेंट है क्या? और विजय, दोनों बताओ जल्दी कमेंट में बताओ। गुड, यस, दैट्स व्हाई आप लोग बता रहे हैं चीज, मैं क्लास में भी बताया था। इट इज ड्यू टू लो लेवल ऑफ HR, horizontal resistance. Everyone will think, okay, that's why। अभी देखो सिंचन थोड़ा चुप हो गया, सिंचन तो लाइव क्लास नहीं देखा था। If I ask Sinchan what is the responsible for vertical effect, what he will tell? He will tell that, sir, there is an complete breakdown of vertical resistance. ठीक है, but the thing is, it is not due to complete breakdown of vertical resistance. It is due to low level of horizontal resistance. If there are some similar, if there are some level of horizontal resistance, okay, then there is no, if it is both are got evolved also, then the crop will not complete failure. Okay, since it is a low level of horizontal resistance, when we are breeding for vertical resistance, this is the main cause of vertifolia effect. ठीक है, समझ गए ना विजय? आप तो हमारा कोर्स का स्टूडेंट नहीं है, ठीक है, देन यू आर गुड। यू टोल्ड दैट कंटिन्यूअस ब्रीडिंग ऑफ वर्टिकल रेजिस्टेंस टू लॉस ऑफ हॉरिजॉन्टल रेजिस्टेंस, ठीक है, गुड। विजय, आप हमारा मार्क टेस्ट बंडल का स्टूडेंट है क्या एटलीस्ट? विजय, मार्क टेस्ट स्टूडेंट सर, ठीक है, गुड। Then boom and bust cycle, again it is due to your vertical resistance genes only. Okay, when breeder will produce cultivar with new resistance, when he use vertical resistance gene, 1 or 2 genes, okay, for particular resistance, for example, okay, in case of, in that particular time in Europe, the lead blight will be the major disease. What breeder did? They targeted some resistance gene, vertical resistance gene, that to that particular pathotype. Then area will increase, encourage, everyone will try to go that particular variety in that all over Europe. Then there is a development of epidemic, since again due to the year in the another cycle, increased virulence detected, frequency increases, your both pathotype, and since the area is increased, okay, the acreage increases, then there is a development of epidemic. Okay, then the process, your area of acreage, that acreage, the cultivation area will be decreased again, it will be cycle. Okay, you can here also the pathotype, here it increases, then epidemic over, then again frequency decreases, then your increases virulence detected. Okay, once the breeder produce new cultivar and there is a more acreage of area, if any virulence gene got detected in the system, the cycle, okay, it is a cycle, boom, where there is an area of acreage is also more and frequency of the particular pathogen will increase, then there is an epidemic, then bust, okay, then there is a decrease in area also after epidemic, then decrease in the frequency of pathogen also. Okay, this is like a cycle, it is given by Priestley, okay, the simple phenomenon, boom period, bust period, good, okay. Then clean crop approach, dirty crop approach given by Marshall. Yes, already we discussed about it in the yesterday's, or sorry, in the last class. Okay, the multi-line development. Okay, it is given by Marshall. What will happen in clean crop approach? Okay, or multi-lines. All the lines in the mixture are resistance, okay, to all prevalent pathogen races. For example, as I told, these are your multi-lines. For example, you are having 4 multi-lines. Okay, this particular multi-line contains resistance gene, okay, for all other the prevalent pathogen races. That means R1, R2, R3. These are 3 resistance gene, okay, and it controls all the pathogen races. So your all multi-lines contains these 3 gene, R1, R2, R3. Okay, it is clean crop approach. No single line is missing the any resistance gene. But in case of dirty crop approach, none of the individual lines are resistant to all the pathogen races. Again, here we are having 4 lines. Okay, we have made a mixture of this particular 4 lines. Some are containing R1, some are containing R2, some are containing R1 and R3, some are containing R2 and R3. But none of the individual single line contains resistance for all the genes. Okay, in this particular case, okay, it stabilizes the race composition of the pathogen population. If in this case, if pathogen got evolved, if it evolved to P4, any pathogen, then the complete failure of loss, complete, since it doesn't contain this particular pathotype. Okay, where there are prevalent are P1, P2, P3 pathotypes. They will try to attack, but there is no space for their survival. So pathotype 4 will got evolved here. Okay, we are making the pathotype to get evolved. But in this case, you can see there are P1, P2, P3 pathotype. Since R1, R2, R3. Your P1, P2 will attack here. Your P1 will attack here. And your P3 will attack here. Sorry, P3 will attack. If it is R1 or R2, P3 will attack here. So there will be survival for the pathogen. And again, each pathotype are attacking different multi-lines. Okay, there is survival for multi-lines also. The yield loss will be less. Okay, it will mimic the HR, horizontal response. Okay, there is not complete crop failure here. समझ गया ना? Clean crop approach and dirty crop approach. बताओ अभी बताओ कोई. I hope now you got it. बताओ. Okay. Vijay, dirty crop once again, sir. अरे वही सिंचन आप बताओ, आप 3 वही लगा रहे हैं, वो वही WHYR यस मैं क्या समझना चाहिए उधर से रतन. डर्टी क्रॉप रिपीट. अरे भाई क्लीन क्रॉप अप्रोच में ठीक है सारे R1 R2 R3 है एंड सिस्टम में P1 P2 P3 है. ठीक है सारे लाइन मतलब सब में R1 R2 R3 है. सो उनका चांस ही नहीं है अटैक करने के लिए. सर्वाइवल का भी चांस नहीं है इधर सिस्टम में. तो ऑटोमेटिकली क्या होगा? पैथोटाइप 4 evolve होगा. The evolvement of pathotype 4. But in case of dirty crop approach, first line contains only R1. Your second line contains only R2. Third will contains R1 and R3. Fourth will contains R1 and R2. None of the lines contains all 3 genes. ठीक है इसमें देखो आपका P1 पैथोटाइप है ना, आराम से इधर सर्वाइव हो जाएगा सेकंड में. आपका P2 इधर सर्वाइव हो जाएगा since there is no R2 here. आपका P3 will survive here since there is no R3 here. सारे पैथोटाइप सर्वाइव हो जाएगा. So there is no requirement for evolvement there in another race evolvement. ठीक है सर्वाइवल तो हो गया और उसका इन्फेस्टेशन भी कम है ना. सारे pathotypes will not infect all the races. So there is a less reduction in the yield. ठीक है इसलिए it mimics the horizontal resistance. समझ गया ना डर्टी क्रॉप अप्रोच में. बताओ रतन और कोई पूछा था? विजय बताओ, दोनों बताओ. यस सिंचन यस ठीक है. यस KSML 3 KSML 8 कल्याण सोना मल्टीलाइन वैरायटी. Okay, those are mixture of different lines. Sir, if the crop is clean, then everything will be clean, sir. When the pathogen evolves, then everything will be off. Yes, good Pooja. If in the case of clean crop approach, ठीक है, when कहाँ तक only एक मिनट, कहाँ तक P1 P2 P3 रहेगा सिस्टम में, ठीक है, वहां तक क्लीन है, ठीक है, सारे क्लीन. इफ there is a P4 then सारे अगेन क्लीन. ठीक है, या क्लीन एंड क्लीन, पूरा क्लीन हो जाएगा. In case of dirty crop approach, there is no situation, okay, for the complete loss of a crop. Dirty crop approach better for decreasing epidemic rate. सही है. Our genes are not present together in dirty crops. Clean crop में सारे are present होता है. वही है. After a few days, the pathogen will definitely evolve in the clean crop. Yeah, वही बता रहा है मैं. Clean crop approach creates selection pressure on the pathogen for evolve new virulence gene. अरे यार, पूरा ब्रीडिंग लैंग्वेज में बता रहा है. सिंचन तो ब्रीडर हो गया. मल्टीलाइन फॉर डर्टी एंड प्योर लाइन फॉर क्लीन क्रॉप अप्रोच. बता सकता है. एंड ऐसे नहीं, आपको क्लीन क्रॉप अप्रोच में सारे प्योर लाइन है, ठीक है. एज वी आर टेलिंग ऑल द लाइन्स इन द मिक्सचर. Okay, if you are telling mixture, then you are referring it as a pure line. Okay, since you are R1, R2, sorry, all 4 lines are having same resistance gene, that doesn't mean that they are pure line. Okay, maybe they are having difference in their genomic level for different traits, right? Okay, that's why we're calling it as a multi-line. Both are multi-lines only. Okay, not your clean crop will be like not pure lines. Maybe they are differing for some other genes, some other resistance gene, some other disease resistance, for some other disease or some other any traits for difference. Okay, for Chaitanya. Okay, then difference between self fertilization and inbreeding depression, tell after the class, sir. Arey Mohan, difference between self fertilization and inbreeding depression. Self fertilization will be in self pollination crops. In case of cross-mated crops, we will not call it as a self-fertilization. We will call it as a inbreeding. Okay. In case of cross-pollination, sometimes, okay, we are not able to do the self-pollination. That means the flowers, the male flowers from the same plant to the same female flowers of the same plant. Okay. When there is a dioecious nature, we can't do like that, right? Okay, in that case, as it is like a sister sib mating. We will call it as a sib mating. Okay, in case of cross-mated crop plants, your inbreeding will be the intense form of self-pollination. Okay, intense form of self-pollination in case of cross-mated crops will be your inbreeding. ठीक है, उतना difference है, कुछ नहीं, दोनों same ही है. And depression, that is consequences of inbreeding in case of cross-mated crop plant. ठीक है, वो inbreeding depression. ठीक है. Exploitation of vertical resistance. How you can exploit vertical resistance? One is your varieties with individual major genes. Okay, some are having XA5 and some lines are XA13, some are XA15. You can exploit like this, XA21. Varieties with individual major genes, multi-line development. Okay, you can go for multi-line development which contains, again, maybe you can go for clean crop or dirty crop. In case of multi-line development, you can exploit your vertical resistance like this. Then gene pyramiding in a single line, XA13, XA21 and your XA5. You can introgress all the genes, the resistance gene of a particular single disease. You can introgress in a single variety. That is your gene pyramiding. Then gene deployment. What about gene deployment? It is nothing but spatial or temporal management. Gene deployment is nothing but spatial or temporal management. Okay. When in case of wheat, for any rust resistance, we are growing in Haryana, Punjab. In Haryana, we will try to push the varieties which are having different resistance gene. In Punjab, we are trying to push different resistance gene varieties. Okay, in case of Delhi and Uttar Pradesh, we are pushing different resistance gene. This is spatial isolation, the spatial management. Okay, that so that we can avoid the epidemic. If any attack, if any evolvement of races, okay, it will not completely spread all over the wheat growing regions, since those contains different resistance gene. What about temporal? In one season, we are trying to grow another resistance genotype. In next season, we are trying to grow some other resistance gene genotypes like this. Okay, the management between spatial means over the space and over the time. We are trying to rotate the different resistance gene. That is your gene deployment. ठीक है क्या बताओ जियोग्राफिकल स्पीसिएशन अरे क्या बता रहा है जियोग्राफिकल स्पीसिएशन plant strategy, yes, it is a plant strategy. Spatial, temporal, good. Okay, this is about your gene deployment. These are the different types how you can exploit the vertical resistance. What is the breeding status regarding in the major crops? In case of rice, we already know that improved Pusa Basmati 1, XA13, XA21. Okay, the first MAS variety in case of rice, that is your improved Pusa Basmati 1, developed from ARI. Then improved Samba Masuri, XA13, we have introduced one more gene, XA5 and XA13, XA21. Both are for bacterial blight disease. Then recently, Pusa Basmati 1847, 1885, 1886. Okay, for the blast and BLB. Then Dweep Dhan 7 and Dweep Dhan 10, the recently 2025 varieties for BLB, Rani Dhan, BLB plus submergence. These are the recent varieties which are released in rice for disease resistance. Okay, these are very important. Okay, whatever the varieties which are mentioned in case of your BD Singh book or any books, any textbook, they will not ask about those things. Okay, any milestones like improved Pusa Basmati 1 and improved Samba Masuri, maybe they can ask, and they will concentrate on recent things. Okay, not about which are released 10 or 20 years before. Okay, इसलिए थोड़ा classes important है. Then in wheat, 2NS, 2AS translocation. That is nothing but in second chromosome. Okay, they have translocated some part of the chromosome from the Aegilops ventricosa. This is the wild relative of wheat. From that particular donor, they have took particularly the second chromosome. Okay, then they have transferred that some of the part of that second chromosome to the wheat crop in the second chromosome itself. You can see the translocation 2NS, 2AS. Okay, chromosomal translocation. It contains LR37, YR, leaf rust, yellow rust, and stem rust. All three rust. Okay, this is the recent development. DBW 371, 372, highly resistant to stripe rot and leaf rust. Malvia 838, blast resistance variety. Then in green gram, Deep Mung 1 and 4, the recently for yellow mosaic virus, and Shika APM 4103 for yellow mosaic virus resistance. ठीक है. न्यू दिल्ली 1989, क्या बता रहा है तू? पूजा, what you are going to tell? न्यू दिल्ली 1989 मतलब? अरे हो पूसा बासमती 1 which is released in 1989. It is not Pusa Basmati 1, it is improved Pusa Basmati 1. Okay, your Pusa Basmati 1 is an parent variety. Then they have introduced XA13 and XA21 using marker rested selection. Okay, that is your improved Pusa Basmati 1. ठीक है. अरे थोड़ा शेयर कर दो सबको, आपके ग्रुप्स में, फ्रेंड्स को सारे शेयर कर दो ना, उसको भी थोड़ा बेनिफिट्स हो जाएगा. चुप के मत प्रिपेयर करो. ठीक है, सबको भेज दो, मतलब ये लिंक यूट्यूब ए क्लास का. हाँ पूजा, दैट्स व्हाट इम्प्रूव्ड पुसा बासमती 1 इज रिलीज्ड इन 2008. ठीक है. Okay, this is all about your disease resistance. Next is your insect resistance. Property of a variety of a host crop due to which it is attacked by insect pests to a significant lower degree. Okay, than the varieties other than the same crop. Stress will be same. It will be impact on your plant growth, development and reproduction. What about resistance? It is a property of a variety of a host crop due to which it is attacked by an insect pest to a significant lower degree than other varieties of the same crop. That is resistance. Okay, some of the genotypes are giving some fight back nature so that there will be a lower degree, than the other varieties. That we will call it as a resistance. Approximately there will be 16% loss due to insect attack. Again, here also you should know there are mechanism. In case of there, the mechanism will be susceptible, tolerance, immunity, that case. In case of insect, there will be a mechanism, again, some mechanism. The degree of resistance, okay, it is given by painter. Immunity will be insect will not consume the plant under any condition. Okay, it is safe. We will call it as a non-host. So immunity, high level of resistance. The host plant possesses such traits that are result a small level of damage by a specific insect under given environment. So that is high level of resistance. Then low level of resistance. The level of resistance is such that the host suffer less than average damage. Then you are susceptible, the complete failure. The level of damage or infestation is more than average, that is your susceptible response. Highly susceptibility is completely damaged, okay, greater than average for the crop, and there is a death of a particular host crop. That is highly susceptible. Okay, the painter has classified like this. And there are some mechanism, okay, how we can see the resistance, that are the three things: non-preference, antibiosis, tolerance and avoidance. Okay, in case of insect, these are important terms: non-preference, antibiosis, tolerance and avoidance, which is given by painter. A resistant variety may have more than one of these mechanisms. Okay, maybe it is having non-preference, maybe it is having antibiosis, or the combination of any of these mechanisms. Okay, in case of insect. Before that, you should see what are chiromones and alamones. In case of plant biochemical, okay, with respect to plant, okay, in case of chiromones, affect insect behavior and physiology in such a way that it leads to the adaptive advantage to the insect pest. Plant will release some chemical, or plants are having some chemical, okay, which will attract the insect, advantage to insect. That is your chiromones. Okay. Then what about alamones? It affects the insect activities in such a way that result in adaptive advantage to the plant. Plants are having some chemical. Okay. For example, in case of cotton, there is a gossypol. What it will do? It will advantage to the crop plant. Okay, where there is a gossypol, it is a toxic. If gossypol, it will be toxic for the insect. That is your alamone. But plants having some chemicals which are advantage to the insect, they will attract the insect. Okay, then it is advantage to the insect. Those are chiromones. Okay, Vijay, chiromones are nothing but plants will release some chemicals which attracts the insect. That means it will be advantage for the insect. That is your chiromones. Okay. First mechanism will be non-preference. In this case, host varieties exhibiting this type of resistance are unattractive or unsuitable for food, shelter and reproduction. Food, shelter and reproduction, for that means oviposition. Okay, there is some mechanism. It is due to unattractive or unsuitability for food, shelter and reproduction by an insect pest. We will also call it as a non-acceptance or anti-xenosis. Okay. Another is antibiosis. This is anti-xenosis or non-acceptance, non-preference. Okay. In this type of resistance, insect will not accept a resistance host variety, even if there is no alternative source of food. Okay, for insect there is no alternative source of food. In that case also, okay, insect will not accept a resistance host variety. So there are no chance, they will not adjust itself. Okay, include color, light penetration, airiness in host, leaf angle, odor, taste. Okay, these are responsible for your anti-xenosis. And it is not an effective mechanism for resistance against polyphagous pest. You know polyphagous pest. Okay, if you are not giving preference as a cotton, I will go for some other bendy. I will go for some other crops. Since I am polyphagous, I don't have any problem. Okay, that's why it is not that much effective. Okay, your anti-xenosis. You can see this particular table. For different crops and different insect pests, there are some non-preference thing. In case of wheat, for stem sawfly, your non-preference will be solid stem. If there is a solid stem, there is no attack from the stem fly. Okay, and they will prefer hollow stem. And in case of rice, okay, BPH, they will low aspergine. Means they will prefer high aspergine. Then that means your aspergine is nothing but chiramon. In case of with respect to plants, okay, if the plants will produce more aspergine, that means that will be an advantage for the insect. You can see high aspergine will be preferred by BPH. That's why your aspergine will be chiramon. Then toughness of an husk, in case of maize, it will be non-preferred by the corn earworm. So that based on that, you can target those traits for your breeding program. You can try to breed for toughness of an husk so that you can avoid the corn earworm. Okay, this is the table which is very important. Okay, you can take a screenshot and you can see this particular table. It will be available in your textbook also. Okay, the important thing again, in case of bollworm in the cotton, okay, they will not prefer smooth leaves and they will also not prefer nectary leaves. Okay, they will be prefer nectarid and airy leaves. Okay, like that there are different examples for different crops. Airiness of leaf and stem is non-preference for jacids in case of cotton. In case of potato, Colorado potato beetle is related to odor. In maize and sorghum, resistance to brown hopper related to taste, like that. Okay, your non-preference will be nothing but the mechanism will be based on the non-preference for their reproduction, their shelter. Okay, this is the mechanism. In case of antibiosis, what will happen here? It refers to an adverse effect of feeding on a resistance host plant. If it feeds on resistance host plant, then there will be an effect on the development and reproduction of the insect pest. In severe cases, it may even lead to the death of an insect pest. Okay, in case of antibiosis, if the insect feeds on that particular host plant, resistance host plant, then it will be affect for the development and reproduction of the insect pest. Maybe it will be, as definition tells, there will be a death of a insect also. The true form of resistance. Okay, this is the true form of resistance. Antibiosis may involve morphological, physiological, and biochemical feature of a host plant. Anything. Maybe it is morphological or physiological or biochemical feature. For example, in case of wheat, solid stem. For stem sawfly, if it is solid stem, if it eats on that, then it will be death for an insect. High silica content. In case of rice, it will be effective against rice stem borer. Again it is. And also high dimbova in the leaves. In case of maize, then it will be resistance for European corn borer. Like that. In case of brassica silicon, okay. That means your silica and your dimbova are nothing but allomones. These are advantage for the plants. Okay, if our plants having high silica content, that means it will be advantage for the plant, since it is effective against rice stem borer and European corn borer respectively. Okay, this is anti-biosis. That is anti-xenosis. Okay, and you also know about at this particular time about chiromones and allomones regarding to plant. Again, there will be allomones and chiromones with regarding to insects also. If insects are releasing, then it is a different story. If plants are releasing, the classification will be like this. Okay, it is very important. Again, it is your non-preference or anti-xenosis and the anti-biosis. It is a feeding behavior. If it feeds on that particular chemical, then there will be a effect, okay, on the development and reproduction. But in case of non-preference, okay, they will not prefer at all for the growth, for the oviposition, for the shelter and food. Okay, you can, again, there is a table available in the textbook and you can take a screenshot here also, so that you can revise it. Then tolerance, again it's the same thing, the same story. Okay, regarding tolerance, whether it is drought, whether it is heat, whether it is disease. Okay, what exactly the tolerance is? There will be an effect, there will be an infestation from the stress factors, but when we are talking about tolerance, it will be related to your yield. Okay, it will experience each and everything, but the level of yield loss will be minimal. Okay, you can maintain the yield. Insect tolerant variety is attacked by the insect pest has the same degree as the susceptible variety. Your reaction, the range of reaction will be the same as your susceptible variety. But at the same level of infestation, the tolerant variety produces larger yield than a susceptible variety. Okay, that means larger yield than susceptible variety means there will be minimum loss of yield when there is an attack by the insect. Minimum loss, not a complete loss. Complete loss will be in case of susceptibility, but in case of tolerance, there will be minimum loss of yield. Tolerance is measured in terms of capacity to repair and replace the damaged parts, healthy leaf growth, and high plant vigor. Okay, how much capacity it is having to repair that infestation, that is your tolerance. Tolerance cultivars have greater recovery of damaged parts than the susceptible ones. Okay, then avoidance or escape, the complete immunity type of a reaction here, refers to escape of a variety from insect attack either due to earliness. In case of sorghum, okay, for sorghum shoot fly, if you sow the sorghum well before the season, it will be escape from the sorghum shoot fly. Okay, and in case of your example, in case of wheat, there will be a problem with the terminal heat stress. That means in case of April, in case of at the end of March and April, there will be a heat stress will come in case of northern India. If you sow the wheat in the earlier October or in the earlier days of November, your crop will mature before March or April itself, so that you can avoid the terminal heat stress. That is again your avoidance or escape mechanism. It will either due to earliness or the cultivation in a season where insect population is very low. Early maturing cotton varieties are escape from the pink bollworm infestation. Okay, in case of sorghum also, okay, early sowing of sorghum will be escape from the sorghum shoot fly. This is your avoidance or escape mechanism. In case of insect, there are 4 mechanisms. What you should know: one is non-preference, we will call it as anti-xenosis. Then your anti-biosis, okay, it is completely dependent on the feeding behavior of a plant of insect. Then the tolerance, your R is equal to 1, but compared to susceptible variety, there is a high yield, minimum loss of yield. Then your avoidance. Okay, this is escape. These are 4 mechanisms in case of insect. What is the status? Breeding status of insect resistance for insect resistance. TKM6 in case of rice, a traditional variety widely used as donor for stem borer resistance. The recent source. Okay, and PUSA 1612, PUSA 1701 resistance against brown plant hopper. Okay, in case of wheat, there are no prominent varieties which are developed in the recent for the insect resistance. VL616, land races from the Himalayan region, resistance for the cereal leaf beetle. In case of cotton, Okay, again important. Your in 2002 BT1, that is Bolgard 1, Cry 1 AC, BT2, Bolgard 2, Cry 1 AC and Cry 2 AB. Okay, they have modified it and so that there is a broad spectrum of resistance. In case of recent 2015 to 2026, okay, the triple gene hybrid, as I told in the beginning class also, Cry 1 AC, Cry 2 AB, and the third gene WIP3A, that is vegetative gene. That means it will be effective on all other stage of the particular insect. Okay, WIP3A. This is about Bolgard BT1, BT2 and the triple gene hybrids. In major crops, again in pigeon pea, ICPH 2740, significantly resistance to the sedative mosaic virus and moderate borer tolerance. PUSA 00 mustard, okay, PDZM 3536, okay, the recently released varieties in case of brassica, okay, which will avoid the aphid colonization. Okay, this is about insect resistance. Okay, clear? Under same condition, tolerance can give more yield than susceptible. Yes, good. अभी हो गया आपका डिसीज हो गया, इंसेक्ट हो गया, मतलब कुछ क्या इंपॉर्टेंट होता है ना, मैं बस वही बता रहा है, ठीक है. सारे बाकी है ना आपको बुक्स में, वो सारे उतना इंपॉर्टेंट नहीं है, ठीक है. बस आपको इतना याद रखना है, ठीक है. Then drought resistance. Again the same story, again the same definition, ठीक है. Krushin, this series, thank you so much for giving this series. Thank you, Kushi. Ability of a crop plants to grow, to develop and reproduce normally under the moisture deficient condition, okay, referred to as a drought resistance. And drought hardening will be like improvement in drought tolerance ability of a crop plant. How we can improve the drought tolerance? That will be drought hardening process. There will be different mechanisms, different techniques we can use to improve that particular drought tolerance. Okay, resistance is a thing, but again the same thing, it is having some ability, okay, to grow under different moisture deficient condition. Okay, then again there will be different mechanism for drought resistance. Okay, you can see here mechanisms of drought resistance. One is your escape, how your plants, crops can escape from the drought stress. Plants complete their life cycle before severe drought occurs. The same story, early flowering and maturity and short life cycle. These two will help in the escape, complete escape from the drought. Okay. Then avoidance, ability of a crop plant to maintain higher internal water balance. Okay, how they can avoid the drought stress? Okay, the stress which is caused by moisture condition. The crop, the genotypes having some ability to maintain high internal water balance. How they can maintain high internal water balance? They can be through the reduced water use. That means they can like less transpiration, like the less loss of water from the leaves, or increased water uptake. Either of these cases, okay, they will maintain higher internal water balance so that there is no word for the stress. It is like avoid the drought. Two mechanism, like as I told, reduce water loss through the transpiration. We will call it as we will call those genotypes as a water savers, or the maintain water uptake during drought period through the like water spenders. That means they are taking more water content to maintain. They can spend normally. Okay, those are water spenders. Water savers are, they will reduce through the transpiration. Water spenders are, they are taking more water. so that they can maintain. Okay, these are the two mechanisms in case of drought avoidance. Drought avoidance is nothing but they will maintain a year internal water balance. Okay, water spenders and water savers, good. Next, drought tolerance. One thing here, how genotypes will maintain water uptake by having deeper root system. Okay, the root system will be important in case of water spenders. Okay, if they are having good root system and they are having some good root hairs, okay, primary, secondary, each and everything, so that they can uptake more and more water from that drought deficient stress. Then drought tolerance, again here ability of to withstand the low tissue water content. There anyway they are not having any mechanism for more uptake or less transpiration. Okay, they are having some ability to withstand that low tissue water content and continue their metabolic activity normally. How they can do this? Through osmotic adjustment. Okay, based on the salt concentration in case of particular internal tissues. Okay, they will try to withstand that low tissue water potential. And proline accumulation. When there is an accumulation of proline, okay, there will be a osmotic imbalance, so that they will try to uptake the water to maintain that particular osmotic potential, so that they can maintain that pressure, turgor pressure of that particular cell. Okay, it will be through the proline accumulation. Increased ABA production, again the same thing. These are responsible for the osmotic adjustment. Where there is a accumulation of proline or ABA, so it will try to maintain that particular water potential in case of tissues. Okay, then drought resistance, this will be tolerance, ability to withstand the low tissue water content. Then resistance, sum of drought avoidance, drought tolerance. Okay, both the mechanism will be act together and they will give somewhat more fight back nature. That is your drought resistance. Okay, it will be measured in terms of various mechanisms: germination percentage, seedling growth, if and stomatal futures. Okay, if it is having the good germination under the stress and they are having good seedling growth in case of stress, okay, we can select those genotypes. They are having that, we are considering that they are having good resistance or tolerance mechanism. Okay, these are the different selection measures. And if I want to talk about breeding for stress, again the story will be same. Okay, how we can do the breeding? You can introduce the variety from outside, you can go for selection, you can go for pedigree breeding, backcross breeding. The methods will be same again. That's why I'm not talking about the breeding methods. Okay, methods are the same, but before that you should know the traits which you want to target. Okay, that's why I am discussing all about these things. Okay, anyway breeding will be same. You can use whatever breeding method you want. Okay, you can use genetic engineering to introgress some genes which are having the resistance or tolerant mechanism. Okay, you can use the marker set variety. Okay, like you can introgress some genes through using markers. Everything breeding procedures and mechanisms, different methods will be same for each and every stress. But you should know the principles or basics before that, so that you can choose the traits whichever important for particular stress. That's why we are seeing the different mechanisms. Okay, Palmira क्या है विजय वो? Okay, the traits as I told in case of drought, morphological traits, leaf rolling. Okay, when there is a leaf rolling, that means it is trying to giving some tolerance to the drought. It will reduce leaf area exposed to sun, so that it will reduce the water loss. Okay, it is trying to avoid that drought. Okay, the leaf rolling is important trait in case of drought. You should know. Where there is a leaf rolls, so that means there is an resistance mechanism. Waxy cuticle. Again, if there is a waxiness in the leaf, it will reduce the transpiration rate. Okay, it is one of the trait which is having drought resistance. Stomatal control, your stomata, reduced leaf area, and deep root system. These are the morphological trait. Okay, the airiness, again, the airiness and the waxiness, both are important in case of drought. Physiological traits, again, stomatal closure, high water use efficiency, osmotic adjustment. Like osmotic adjustment is nothing but the water potential adjustment, and maintain cell membrane stability in case of drought. These are the traits which are having some physiological background. These are the important traits again. And biochemical, proline accumulation. Okay, again it is act as an osmoprotectant. Your proline and ABA, sorry, yes, ABA, and antioxidant defense. Okay, your proline, ABA, and stress responsive genes. Okay, these will be comes under your biochemical traits. Okay, these are the traits you can target for the drought resistance. Okay. Then that is about drought. What about salinity then? Okay, it is also one of the stress. pH above 7.5. Ability of a plant to prevent or reduce or overcome the injurious effects of soluble salt present in their root zone. Plants having some ability to escape, to overcome that particular salinity. So it is, we will call it as a salinity resistance. What are the ways to overcome that problem? There are two different ways to reclaim that salt affected soil. Again, it is very difficult to reclaim. The reclaimity of salt affected soil will be very difficult procedure. So we can go for development of salt tolerant genotypes. Again, what is the mechanism here? Here there are two mechanisms. One is exclusion and compartmenting. Exclusion means the plants or genotypes are trying to exclude that salts in their root zone itself. They don't want to take it. That is your exclusion mechanism. And compartmenting. Anyway, they already took it. Now they want to do something. What they will do? They will try to compartment. They will compartment means they are trying to push those salt to any of the particular tissue or any of that in the within cell only. They are trying to push to any as a junk material. They are trying to compartmenting these particular salts. That is your second mechanism. Exclusion is process of excluding entry of higher amount of salts in the root zone itself. Compartmenting is nothing but some parts of resistance genotype accumulate the more salts than the others. More minerals store in leaves than the shoot. They will try to store in the leaves. Okay, that is your compartmenting. These are the two different mechanisms in case of salinity. Okay, so salt tolerance. Then the mineral resistance. In case of mineral resistance, okay, the two types, deficiency and toxicity. There will be a deficiency of some minerals or there will be a toxicity of some minerals. Okay, mineral deficiency, genotypes that show lower detrimental effect of mineral deficiency than the other genotypes of the same species are called mineral deficiency resistance genotype or mineral efficient genotypes. Okay, when there is a mineral deficiency, they are having some lower detrimental effect. Okay, that means they are giving some good yield compared to other genotypes of the same species. Okay, we will call those genotypes having that mineral efficient genotypes or mineral deficiency resistance genotype. Okay, again there are two mechanisms: avoidance of mineral deficiency, tolerance to mineral deficiency. How we can avoid the mineral deficiency? Observe more quantities of the deficient minerals and transport to the appropriate sinks than the other genotypes. It prevents the deficiency of the concerned minerals in relevant tissue. Okay, they will observe more and more quantities. That is your avoidance mechanism. Tolerance mechanism, low level of injury. Okay, when there is a deficiency, the low level of injury. Okay, than the other when the tissues contains comparable and lower than the critical concentration. Avoidance is nothing but they are not into that mechanism itself. Okay, but in case of your tolerance mechanism, so that in avoidance they will try to accumulate more and more. They will try to absorb more minerals so that there will be no proper deficiency. But in case of tolerance, what will happen? In case of tolerance, there will be a mechanism. Okay, anyway, deficiency is occurred, but there will be low level of injury. Okay, they will try to cope up with that particular deficiency. That is your tolerance. Okay, this is about mineral deficiency. There is a toxicity also. How they can escape from that particular toxicity. Okay, before that, the mechanism of mineral deficiency resistance. What are the mechanisms? How they can cope up with that? Mineral redistribution, first one. Minerals present in older and senescing leaves mobilizing to younger leaves. Okay, your NPK and Mg. They can mobilize from the older leaves to younger leaves. Mineral, we will call it as a mineral redistribution, so that the deficiency we can escape or we can avoid the deficiency. That is your mineral distribution. Then efficient mineral uptake. Roots of iron efficient genotypes releases H ions in the rhizosphere, which reduces the F3 to more available Fe2. Okay, the efficient mineral uptake. It is one of the mechanism. Then increased mineral transport, increased root and shoot ratio, and increased root air density, so that they can uptake more and more minerals. Okay, these are the different mechanisms to escape from the mineral deficiency. Okay, redistribution, efficient uptake. Okay, these are the mechanism. Now, as I told, toxicity. Your aluminium and manganese, these are the two major elements. They can make a situation where the toxicity will appear. Toxicity stress arises due to the presence of toxic concentration available minerals in the root zone. In acidic soil, it is aluminium and manganese are most important elements of toxicity. Okay, aluminium toxicity resistance. The resistance mechanism we are talking here. How we can maintain that resistance? Increased pH of rhizosphere, which reduces the aluminium solubility and uptake. If there is a pH increase in case of rhizosphere, so there will be a less solubility of aluminium and uptake. And exclusion of from the root itself. Your Atlas 66 wheat variety, this is one of the older source for the aluminium toxicity resistance. Okay, in the roots itself, there is having some interaction, so that it will exclude in the root itself from entering the root. Aluminium compartmentalized in the root, older leaves within the cells. This may also occurs due to the chelation of aluminium by organic acids in the soil. Compartmentalization in the root itself or in the older leaves, so that it will not enter into the growing regions. In some aluminium resistance plants, growing tissues is accumulated high level of aluminium. Okay, but they are having some resistance reaction. Okay, that is having some resistance reaction. Then manganese toxicity, again reduced transport of manganese from the root to shoot. Okay, manganese is oxidized in the root to Mn4, so that the rate of oxidation is higher in manganese resistance genotype, so that they can be avoided in the root stage itself. In apple, manganese distribution is such that different concentration accumulate in the different parts of the shoot. In apple, manganese distribution. Again, that simple thing, the simple basic thing what you should know, how they can get an resistance mechanism. In case of deficiency, they will try to uptake more. Okay, and they will try to redistribute from the older leaves to the younger leaves. But in case of toxicity, they will try to exclude, like your salt exclusion. They will try to exclude in the root zone itself, and they will try to accumulate in the older leaves. Compartmentalization as seen in the salt. Okay, these are the basic mechanisms of mineral toxicity resistance. In this particular thing, again, as I told, they will not definitely ask you that how the mechanisms will work out. As I told, the important things will be your disease and insect and your drought. Okay, and heat. Okay, in case of drought, as I have told some traits which you can target. Okay, based on that only they can ask. your leaf rolling, your vaccinase, okay, they will give a tolerance or avoidance mechanism. In case of heat stress, avoidance again, ability of a genotype to dissipate the radiation energy, thereby avoid the rise in the plant temperature. But in case of tolerance, ability of a genotype to withstand that stress better than their internal temperature are comparable and in the realm of the heat stress. In case of avoidance, they will try to avoid that stress itself. They will try to avoid this radiation energy. But in case of tolerance, okay, yes, they will experience that stress, but they will try to cope up with that. As I told from the beginning of the class, okay, this will be the mechanism. You can see avoidance will be through transpiration, so that there will be a cooling. It will avoid the particular radiation energy. And leaf reflectance due to pubescence and glucosinase. That means pubescence is due to airiness and glucosinase is due to the vaccinase. It reduces the light interception by the leaves, so that there is no heat stress. In case of tolerance mechanism, again, your stability in photosynthesis too after the experience with the stress, photosynthetic translocation, your osmoregulations. These are the mechanisms with related to tolerance. But avoidance through the pubescence and glucosinase, vaccinase. Okay, these are the mechanisms in case of your heat. What about cold? Again, chilling is more than 0 degree. If any stress that is more than 0 degree, that we will call it as a chilling. Freezing, it is less than 0 degree. In plant level, what will be the effect of stress, chilling stress? It will reduce germination, root growth decline, okay, AB accumulation, abnormal seed set and failure of seed set. But in case of cellular level, there is a membrane damage, protein change conformation, decline in photosynthesis, and it will also interfere with the chlorophyll synthesis. This is the effect of stress in case of plant, chilling stress in case of plant level and subcellular level. So how we can do the resistance? Okay, what are the mechanisms here? Freezing, avoidance, tolerance. Again, two terms, avoidance. In avoidance case, they will completely avoid that particular stress. Ability of a plant tissue or organ, okay, to avoid that ice formation at sub-zero temperature. How they can avoid that ice formation? Supercooling mechanism. Okay, lack of ice nucleators. Ice nucleators are responsible for the ice formation, but that lack of those ice nucleators will avoid the ice formation. Small cell size, little intercellular space, low moisture content, presence of anti-nucleators. Okay, this will responsible for supercooling mechanism. That means it will avoid the ice formation. That is your avoidance. But in tolerance, there is a formation of ice crystal and everything, but they will survive once after the particular stress. Okay, ability of a plants to survive the stress generated by extracellular ice formation. There is a formation of ice crystal, but they will recover after thawing. There may be some having cold responsive proteins, so that they can try to get that recovery after thawing. Okay, that is your tolerance. Same words, same definition and same mechanism. What are the achievements? What are the achievements in case of abiotic stress breeding? Okay, whether it may be drought, whether it may be salt, okay, whether it may be heat stress. Okay, there are some achievements. In case of rice, Sabagi Dhan, one of the most successful drought tolerance rice variety released in India. Okay, Sabagi Dhan. DRR Dhan 42, DRR Dhan 54, DRR Dhan 55. Okay, which are released from Indian Institute of Rice Research, Hyderabad. Drought prone areas are for aerobic cultivation. Pusa DST1, okay, drought and salt tolerance. Recently released, it is gene edited rice cultivar. Okay, Pusa DST1 from IRI. CSR1, a mass derived NIL of Pusa 44 possessing bacterial blight genes and also the saltal QTL for salt resistance. CSR101, which is for salt resistance. Then Porteracia correctata, the recently found that it is an highly salt tolerant wild relative found in coastal region, offering unique genes for extreme salinity tolerance in case of rice. This is all about rice. Submergence tolerance, your Swarna sub 1. Okay, the sub 1 gene, it will give submergence tolerance. We have introduced that sub 1 gene into Swarna background. Then salinity tolerance, saltal QTL, CSR10, CSR23, CSR36. Okay, these are salinity tolerant rice genotype. Okay, these are the recent ones and the prominent the important achievements in the initial phases. What about wheat? Karchia 65, famous donor for salinity and drought tolerance. Okay, Karchia variety. Pusa Prabhat, drought and heat tolerance along with the good protein content. SD3385 from the IRI Pusa in 2022-23. Okay, beat the heat variety. It is for majorly for terminal heat tolerance. Okay, SD3385, famous variety. Then Aegilopsis tosti is the source for drought and heat stress in case of wheat. Okay, there are so many wheat genotypes like the wild relatives, Thinopyrum species, Paticum, Intermedium and Elongatum. These are like for the sources for salinity, drought and extreme temperature. This is about abiotic stress spreading in wheat. Next, abiotic stress spreading in Bajra. Dhanashakti, this time they asked in ARS prelims, in ASRB net. Okay, about Dhanashakti. From which crop they are? Simply they have asked. Okay, since it is recently a biofortified variety in case of Bajra and also having some drought tolerant. Okay, Dhanashakti. HHB 67 improved, the first MAS variety, marker-sted variety, which is drought and downy mildew resistance. In case of abiotic stress spreading in maize, DHM 117 drought tolerant and drought guard. This is a transgenic variety which is not released in India. Yeah. Okay, but this is transgenic variety. For it contains bacterial cold shock protein gene, so that it is containing some drought tolerant mechanism. Okay, in case of maize, drought guard, transgenic variety. Okay, then breeding for quality. Breeding for abiotic stress, breeding for biotic stress completed. Now, what about breeding for quality? What are the important things? Maybe they can ask in case of your exam. Quality trait is nothing but a trait that defines some of aspects of the produce quality. Okay, it will express the quality of a produce. Okay, there are some aspects. We will call them as a quality traits. We are classified them into 5 groups: morphological, size and color, easily observable. Based on that, we have classified some traits as a morphological traits: organoleptic, palatability of produce, taste, aroma and smell and softness. Okay, taste, aroma, smell, softness. Based on that, we have classified some traits as a organoleptic. Okay, nutritional, protein content, quality, vitamin and mineral content. Based on that, we have classified our quality trait. Biological, protein efficiency ratio, biological value, body weight gain, and other quality like cooking quality, fiber strength of the cotton. Okay, these are the different groups we have classified in case of quality. Okay. What about breeding for quality? What is it? How we can explain that? Breeding for quality is the genetic improvement of a crop plants to enhance the desirable quality character, such as someone want to increase protein content, someone want to increase oil content, vitamin, fiber. Okay, in case of rice, someone want to increase the basmati quality, the grain length. Again, it is a quality trait. In case of brassica, okay, the oil quality by reducing your glucosinolates, okay, and particularly in groundnut, you should increase oleic content. Okay, these are the different traits where we can target. Okay, by using genetic mechanism or genetic improvement to enhance this desirable quality. Okay, so that we can do some reading. Aroma, keeping quality in case of your tomato again, shelf life. Okay, these are the different examples for breeding for quality. In case of rice, this is the table where you can see the different traits. Okay, hulling and milling recovery, preferred type will be high. There should be high milling recovery, right? Once you are going for milled rice or hulling, okay, you should get some more percentage. Preferred is higher. Endosperm, there will be transparent and non-waxy. Okay, transparent is opaque due to loose packing of starch and it is polygenic in nature, and non-waxy due to dominant gene. Less the length and shape of grain. We always prefer long and slender rice. And amylose content should be intermediate. In case of our Indian condition, your amylose content should be intermediate. Okay, if amylose will be less, then will be more amylopectin. Okay, if there is more amylopectin, your rice will be like waxy. Okay, so sushi rice, the Japan and Chinese, they will prefer that sushi rice. Okay, it will be very waxy. Okay, in the bowl they will eat the waxy rice. It will be like milky nature. Okay, where there is an less amylose, then there will be more amylopectin. Okay, that will create that waxiness. The sticky rice, good. Yes, that's what. Okay, the grain elongation, greater elongation, the greater preference. The basmati is having the more grain length. Okay, though there where the consumers will prefer. And these are different thing. Aroma, that should be presence. Okay, it is oligogenic in nature. Prefer, the consumers will prefer aroma, right? Okay, in case of basmati, they will prefer aroma. Okay, can anyone tell which is responsible? The aroma in case of basmati, which is due to which chemical and which gene responsible for that? बताओ, gift, ले लो gift. किसको चाहिए? The gene which is responsible for aroma in case of basmati rice. देखते हैं अभी गिफ्ट. हाँ, पैरील और सिंचन, आपको तो नहीं मिलेगा गिफ्ट. आप सही से बताओ, देखते हैं. अरे डाल दिया कोई, मधु हेगड़े. BADH2 सिंचन, आप बस BADH बताया था. BADH2 हाँ, पहले डाला था मधु हेगड़े. ठीक है, मधु हेगड़े आप हमारा कोर्स का स्टूडेंट है क्या? मधु हेगड़े, पेंडा मारी लगते ना. क्या है विजय, क्या लगा रहे तो? ठीक है, if you are our student, then you know how to contact me so that you can send your address so that you will get a gift. ठीक है, रतन आपको ऑलरेडी गिफ्ट तो मिल गया यार. BADH क्यों लगा रहा है फिर से? दो बार. ठीक है, बीट बाउंस आपको नेक्स्ट टाइम ट्राई करो. आज तो अभी मधु हेगड़े आंसर कर दिया ऑलरेडी. ठीक है, that is due to BADH2 gene and the chemical which is responsible for that is 2-acetylene. लगा दो chemical, लगा दो कोई. The gene which will produce some chemical. अरे मैं इसमें ऑलरेडी बताओ. देख लिया ना सारे. Aroma in rice. Aroma is very important quality trait in basmati rice. The main aroma is due to 2-acetyl-1-pyrroline, the chemical, but the gene responsible for that fragrance is BADH2 gene. Okay, the locus which is responsible for that particular chemical is BADH2. Fragment rice varieties usually have mutation in the BADH2 gene, which leads to an aroma development. Normally in BADH2, if it is in dominant condition, then there is no fragrance. When there is a mutation, in that particular loci, then you will get an chemical. Okay, yes, 2-acetyl 1-pyrroline. Good, good. Then breeding for aroma. Your Basmati 370, the traditional, the first Basmati variety in case of India, the traditional aromatic Basmati variety. Then Pusa Basmati 1, 1989, semi-dwarf. We have intra-aggressive semi-dwarf gene by crossing. I have told in pedigree breeding. Okay, by using pedigree breeding, they have developed Pusa Basmati 1. Then by using Pusa Basmati 1 sister lines, okay, it took again some 15 years after 1989. In 2002 or 4, they have released Pusa Basmati 1121. Okay, by crossing 2 sister lines continuously, and they have selected for the longer, extra long length, extra long slender grain, high elongation. That is your Pusa Basmati 1121. Okay. My tab was getting eat and automatically closed at that time. Sinchan. That is about your aroma. Then golden rice. Again, it is a quality aspect. If I want to ask, if I want to tell, what is golden rice? There is a major rice growing area in case of Philippines, Indonesia. They will prefer rice more and more. That in that region there is a deficiency, vitamin A deficiency. So there are so much problems with the vitamin A deficiency. So that what they have did, Ingo Patricus and Peter Bayer, they are having some vision that it should be a vitamin A. We should target that vitamin A deficiency. Okay. Beta carotene is a pro-vitamin A and gives the grain golden color. Okay. It will also help to reduce vitamin A deficiency. Okay. So we need to produce this particular beta carotene, which is not natural available pathway in case of rice. We should do something extra. Okay. Why normal rice lacks it? The normal polished rice, endosperm doesn't have an active carotenoid pathway. But in case of golden rice, we are having that pathway. We are making it. How we are making it? It is a pathway in the golden rice. Okay. GGPP. It will start from GGPP. It is normally not having in case of rice. By using transgenic, we are introgressing some genes from external so that pathway will occur. Your GGPP is the primary chemical where your pathway will start. We have given phyto and synthase. Okay, that is PS by gene from the daffodil in the golden rice 1. Okay, that will convert the GGPP into phytoin. The responsible here phytoin synthase PSY. That gene we have introduced from the daffodil. Then this phytoin will convert into lycopene. How? We are having CRT1. Okay, this CRT1 is from the Erwinia iridivora. Now we are calling it as a Pentoia ananitis. Okay, we took that particular gene from Erwinia. Then lycopene to beta carotene. Your beta carotene is precursor for vitamin A. This particular step, we need the gene that is lycopene B cyclase. This lycopene B cyclase, again we took it from from the daffodil. Okay. This is the complete process. We have introduced two genes, from two organisms, from daffodil, okay, that is your PSY and lycopene cyclase, and CRT gene, which is from Erwinia, so that the pathway will occur in case of rice, so that we will get a beta carotene. But in case of golden rice 2, we replaced this CRT gene that we took it from, the first from daffodil in earlier golden rice, that is this PSY gene. Okay, we took it from maize. Maize having the natural mechanism, the natural pathway. In case of maize, we are having the natural pathway for beta carotene production. So that we replaced this PSY gene from the maize. In case of first, we took it from daffodil. But in case of golden rice 2, we replaced the PSY gene, which is from maize. Okay. And this particular lycopene B cyclase, okay, we have searched in the rice itself, so that we got the endogenous activity in rice itself in case of golden rice 2. So that only the gene with the CRT we used in the golden rice 2 from the bacteria, okay, and the PSY1 from the maize in case of golden rice 2. This is all about your golden rice. Okay, in 1990 it started from Ingo Patricus and Bayer. In 2000 your golden rice 1 is developed. In golden rice 1, as I told, it is daffodil PSY, the daffodil PSY and bacterial CRT and daffodil lycopene B cyclase. Okay, these two are from daffodil and this is from bacteria, that is your Erwinia rudevora. Okay, in case of in 2005 the golden rice 2 developed with the maize PSY and bacterial CRT. And anyway, this third gene will be endogenous in the rice itself. That is your golden rice 2. Okay, in Philippines in 2021 it is officially released for the cultivation, golden rice 1. Recently they have promoted this as a Malasag rice in Philippines. Okay, it is famously labeled as Malasag rice. Malasag means hope. There is an hope. We will call it as like that, na? That means Malasag means there is an hope in their language. Okay, they have released it for the cultivation so that they can tackle this vitamin A deficiency. Okay, this is all about golden rice. Gene taken from daffodil and maize. Yes, that's what I told. PSY, in case of your golden rice 1, it is took from daffodil. In case of golden rice 2, that same PSY gene from the maize. Okay, yes, good. Then rice ho gaya. Then wheat. In wheat, the major quality trait I can say that is your gluten content and strength. Okay. Anyway, grain protein, high protein, we prefer high protein. In chapati quality, bread quality, we need soft texture and good palatability, and again duff strength and elasticity. Biscuit quality again requires soft grain, low protein and weak gluten. Okay. Micronutrients, zinc and iron biofortification in case of wheat. In case of wheat, zinc and iron biofortification is very important. Okay. There are different quality traits. You can take a screenshot or you can see the recording. And as I told, the gluten strength is very important in case of wheat. Hard wheat, medium wheat, soft wheat. There are 3 types of wheat. Okay. And it is based on the gluten. If it is strong gluten, it is hard in structure. If it is moderate, then medium. If it is weak, then soft. Endosperm will be compact in case of hard wheat, intermediate in medium wheat, soft in the flory mealy wheat. Okay. Water absorption, high in case of hard, moderate in case of medium, and low absorption in case of soft wheat. What you should know in hard, medium and soft, that is your gluten strength. The strong gluten, the wheat will be hard, so that if where there is a strong gluten, you can go for bread and chapati making. Okay, the strong will be mainly for bread making, so that it will having good water absorption capacity, and the duff strength will be strong, the duff strength, so that there will be maximum expansion in case of like your bread. Okay, in case of soft wheat, you can see the gluten strength will be weak and it is flory, so that your duff strength will be weak. There is no expansion of your duff, so that you can use it for biscuit and cookies, your soft wheat. That's what you should know. Hard wheat for bread, since it contains good gluten strength and good expansion, and your soft wheat weak for biscuit purpose. Okay. Recent achievements: Karan Vandana WB02 for high zinc content, And this is important. Pusa Tejas, good for, that is durum wheat, good for pasta and semolina. And your Pusa HSP, good quality for bread making. Okay, these are some recent achievements in case of wheat. Okay, we are talking about quality. Okay, you should remember. Then your maize, Surinder Vasal and Evangelia Wellingus in CIMIT, what they have did, they have developed QPM, quality protein maize. That's why they got World Food Prize in 2000. What is quality protein maize and normal maize? In normal maize, maize protein is poor in lysine and tryptophan, and it contains more zein protein. Where there is a zein protein, where there is more zein protein, there is less lysine and tryptophan. Okay, where there is more zein protein, less lysine and tryptophan. That's what will happen in normal maize. But what about QPM? In QPM, you can see there is an opaque 2 mutation. Mutation will happen. Since this mutation, which is in recessive condition, it will reduce this zein protein. It will reduce this zein protein, so that the non-zein will increase. Automatically, your lysine and tryptophan will increase. Okay, the key gene is opaque 2. Okay, the normal opaque 2 will increase zein protein, but it is mutated opaque 2, so that there will be less zein protein. I will be lysine and tryptophan. The simple mechanism. But the original opaque 2, the originally they have found originally was found by someone else. Okay, so that what happened in the original opaque 2 gene maize, it is having soft and chalky, low grain weight, poor storage quality, more susceptible for pest and disease, and lower yield. Okay, the thing is soft and opaque kernel. We will not prefer those things. So Surendra Vasal and Evangelina, what they have did, they have introduced some modifier genes to that opaque background, so that odd endosperm, better yield, better storage quality, and high lysine tryptophan. This is before soft and opaque kernel. After they have used modifier genes, odd and vitreous kernel. Okay, this is about your QPM. The loci will be your opaque 2, that is mutated opaque. Okay, the important QPM will be your Shakti. The release first QPM is composite, which released is Shakti. Then your high quality protein maize, that is QPM hybrid, HQPM, HQPM5, HQPM7. Okay, these are the next version of your QPM. Then Vivek QPM9. This is early maturing, the first QPM hybrid. Vivek QPM9. You should remember, first QPM hybrid. Okay. Now recently Pusa HQPM1 improved. Okay. What they have did for HQPM1. Okay. Now they have introduced one more gene, that is your beta carotene. Okay. Now they have included that provitamin gene also through MAS, marker selection, to the background of QPM. Okay, that is your recent one, that is Pusa HQPM1 improved. Pusa HQPM1 improved. Okay, this is all about your QPM story. Then sweet corn, waxy corn. There are pathway in case of maize. Okay, based on that there will be a different corn. Okay, the sweet, normal, sweet, and waxy corn. In case of normal starch maize, what will happen? The sucrose will convert into glucose 1-phosphate, then glucose to ADP glucose, starch and amylase and amylopectin synthesis. This is the normal pathway, the normal starch maize. Okay, where you will end up with amylase and amylopectin. From sucrose to glucose 1-phosphate, there is a need of sucrose synthase. Then glucose 1-phosphate to ADP glucose, there is an enzyme we need, that is AGPase. It will convert glucose 1 to ADP glucose. Then ADP glucose to starch, that is through the starch synthase, so that you will get a normal amylase and amylopectin. Most sugar will convert into starch in case of normal starch maize. Okay, it will end up with the starch, that is amylose and amylopectin. Result will be hard and starchy kernel, the normal pathway. But in case of sweet corn, what will happen? This sucrose will not convert into the starch, that is your amylose and amylopectin. What will happen here? Reduced conversion of sugar into starch, so that this sucrose, that sugar will accumulate, so that your kernels will be very sweet. The gene which is responsible is your SH2, small SH2. Okay, where there is a small SH2, it will reduce this particular AGPS activity, so that your sucrose will not convert into starch, so that there will be accumulation of your sucrose in the kernels, so sweet kernels. The genes involved as SU1 and SH2. Okay, these are the higher sugar, lower starch in the kernels. That is your sweet corn. Okay, it is also not the quality breeding. In case of maize, you can target this particular SH2 gene, so that there will be accumulation of sugar. Sweet corn तो पता है ना, बेचते हैं ना सब, आपका थिएटर के सामने, मार्केट में, सब जगह में मिलेगा ना स्वीट कॉर्न ले लो, उसका पाथवे. ठीक है. In case of waxy corn, okay, there is a waxiness. Okay, what will happen? How that procedure particular? Again, the pathway will be same. If it is normal, then sucrose to glucose 1 phosphate, then ADP glucose, then ADP glucose to starch, and there will be accumulation of amylose and amylopectin. But as I told, waxy, in case of rice also same. Waxy means more amylopectin, accumulation of more amylopectin. What is the pathway here? Sucrose will convert into glucose 1 phosphate, no problem. Then glucose to ADP glucose through AGPS, no problem. Then ADP glucose will convert into starch synthesis. It will happen. But there is an mutation. In case of waxy, if it is normal WX, then normally it will convert into amylose and amylopectin. But there is a mutation in this particular loci, so that the GBSS will be absent. The GBSS will produce amylose normally. In normal case, your GBSS, that is granule bound starch synthase gene, will produce amylose. This waxy loci will be responsible for granule bound starch synthase, that is the normal gene. But when there is a mutation in this particular loci, you will not get amylose. If you are not getting amylose, that means automatically it will increase the amylopectin. Okay. When there is a mutation in the waxy locus, there is no GBSS. There is no GBSS. That means there is no amylose. There is no amylose means there is a amylopectin, so that your grains will be waxy. Okay. Starch is almost entirely due to amylopectin, so that you can get a waxy corn. Seratina, good. Arey, popcorn is the specific variety of a maize. Okay, it is not that any pathway which is regulating. Okay, it is a completely specific variety of a maize. Okay, where you will, whenever you will get a, in case of normal maize also, if you give the heat, okay, what it will, there will be your kernel will burst out. In case of normal maize also, but not that much extent, as you can see in the popcorn. Okay, so that is a based on the selection, we have particularly developed some of the varieties which are popcorn variety, where there is a maximum bursting and expansion of your kernel. Okay, if you see, in case of sorghum, in case of your chickpea, okay, wherever you will give yield, so there will be expansion of your kernel. Okay, burst and expansion. But in case of maize, there will be at the more extent. It is based on again selection. Okay, there is no particular pathway. Okay. Primitive ancestor of maize is pod corn. Yes, pod corn. That is your teosinte. Okay, it is one of the form of pod corn. Teosinte. Yeah, that's what. This is all about your maize. What about pulses? In pulses, the mostly will target high protein content, improves nutritional value, better amino acid balance, improves protein quality, high iron and zinc. Okay, high iron and zinc is a important trait where we are targeting now. Okay, helps reduce micronutrient deficiency, low anti-nutritional factors. It is very important. Okay, you should target these anti-nutritional factors. Improve digestibility and mineral absorption. If where there is an anti-nutritional factor, then there will be a problem with the digestion, or it will give the problem with the mineral absorption. Okay, breeding for anti-nutritional factors, low anti-nutritional factors is a very important thing. They will definitely ask in case of your exam. Okay, better cooking quality, reduces cooking time, so fuel requirement, better dal recovery, important for processing industry, better seed size and color. Okay, better seed size and color, improves market preference, improve taste and texture, consumer acceptance. In the pulses, these are the very important thing. Okay, where you are trying to improve the protein, there will be a negative impact on the yield. Okay, where you are trying to improve the protein, then you will get a less yield. It is an actual negatively correlated traits, both are. But up to some extent, we can improve. Okay. Regarding our courses in between, okay, now you will get this mock test bundle only at 399 for this particular exam. There will be a more 15 days more. You can just exploit this course. Okay, you will, if you get enrolled, then definitely you will get an good mock test and information. Okay, and for next year or for 365 days, we are having ASR Net and PhD course for 6,999, where we have covered entire syllabus of your ARS and ASR Net. Okay, from different mentors. Okay, including me. Okay, this is about your ASR Net and ICAR PhD for plant sciences. Okay, for next year, for 2028, there will be a course available for 8,999, and for exam for PG for next year, it is available for 4,999. Okay, for the starting students, there will be a price of, you can go and purchase. Okay, and get most benefit from our platform. You can just imagine, okay, how much information we are giving. We are already given in as a free in YouTube. Okay, you can imagine, just imagine what about our courses. Okay, how much content we are having in that particular courses. You can just ask your feedback from our students, the immediate seniors. Okay, based on that only you can enroll. No problem with that. Okay, you can just inquire about your immediate seniors or your friends. Okay, one are already enrolled at our courses, you can purchase. And one thing I should tell you guys here, in our already the mock test series course, we are having almost 2,000 students, which they are enrolled already for this particular this time course. That means, as I told in the last class also, only there will be 3,500 students which are writing ICAR PG plant science. Out of that, already 2,000 students are enrolled to do this particular courses. If you are not yet enrolled, then already what I thought are, you are out of the competition, out of the syllabus. So that you should enroll, so that you will get an inform. You will get most benefit. अरे यार 399 तो एक मूवी का टिकट का प्राइस है बस. आप कितना मूवी देख सकते हैं एक बार. यार एक बार JRF होने के बाद ना, आप जस्ट इमेजिन कितना मूवी देख सकते हैं. व्हाट अबाउट बायोफोर्टिफिकेशन इन पल्सेस? ठीक है, क्रॉप lentil, पूसा गेती मसूर, iron and zinc. This particular variety lentil L471, they have asked in the previous exam, in ICAR PhD exam. Okay, iron and zinc, iron and zinc in case of lentil. In chickpea, Pusa chickpea 10216, recently released, high protein. Pusa chickpea 20211, high protein again. Field pea in IPFD, and mung bean IPM 0203, improved quality for protein and yield. These are biofortified pulses. What about biofortification? To increase the mineral and nutrition or any vitamins. Okay, any quality traits through the breeding mechanism. That is your biofortification. Okay, these are the recently released varieties. एंटी न्यूट्रिशनल फैक्टर, अरे ये तो ले लो, स्क्रीनशॉट ले लो, ठीक है, पक्का एक क्वेश्चन आएगा एग्जाम में, ठीक है. मे बी दे विल आस्क इन योर मैच द फॉलोइंग क्वेश्चन, और मे बी दे विल आस्क in case of your single MCQ question. Okay, like they can ask the different attenuation factors in different crops, they will definitely ask. Okay, these are the important things in BOAA in Lathria sativus. Okay, what it will affect? It will do the paralysis for lower limbs, erucic acid in brassica, gossipol in cotton. Okay, phenolic glycoside in case of groundnut, phytic acid in pearl millet, trypsin inhibitor in soybean. Okay, these are the anti-nutritional factors which are very important. Okay, you should target these factors to reduce. Okay, this is one of the major targeted area in case of quality breeding for low anti-nutritional factors. ले लो फटाफट स्क्रीनशॉट ले लो, ये टेबल से पक्का एक क्वेश्चन आएगा, ठीक है, पक्का 100%. Then quality breeding in mustard, as I told, low erucic acid, low glucosinolate. These are the two major we are targeting in case of mustard. Erucic acid should be less than 2% and your glucosinolate, it should be less than 30 micromole per gram. Why it matters? Better edible quality, better meal and feed value, better consumer acceptability, better export value. How we can do the simplified breeding here? Your oleic acid, that is 18:1. Okay, mono unsaturated fatty acid. There is a FAE1 enzyme, okay, fatty acid elongation 1. Due to that enzyme, your oleic acid will convert into eicosanoic acid. Then FAE1 enzyme, again that fatty acid elongation enzyme, it will convert this into erucic acid. Okay, C22:1. Your normal oleic acid will convert into erucic acid with the enzyme FAE1. Okay, this is, we don't need it, this is erucic acid. Okay, the mutated FAE1, we should mutate this particular FAE1, fatty acid elongase 1 gene. Okay, fatty acid elongase. We should target that fatty acid elongase gene so that there is no erucic acid. In case of glucosinolate, your amino acid will convert into glucosinolate biosynthesis pathway, then glucosinolate in the seed meal. So we have to select for the low glucosinolate accumulation so that you can also reduce glucosinolate. Okay, these are the two mechanisms. What about the recent examples? Your PD, ZM 31, 32, 35, 36. These are the recently released varieties from the PUSA campus, from IRI, PUSA 00 mustard. Okay, low fiber 000, good Vijay. PUSA 00 mustard, PUSA 000 mustard, that means for low fiber also. One more trait we have included. This is for mustard. Okay. Why quality breeding is difficult? Yes, your quality breeding or stress breeding, both are very difficult. Why? Quality traits are polygenic in nature. Sometimes they are controlling by more number of genes. It is very difficult to target those particular genes. Okay, if their trait is quantitative, then it is very difficult. Strong environmental influence and IgE interaction. IgE interaction. G interaction is nothing but, okay, there is an interaction with the environment. In some of the environment, they will perform better. In some environment, they will not perform well. Okay, there is an interaction with the environment. Okay, in quality traits, there is a problem with that G interaction. Quality testing is costly. Trait measurement is itself costly. Okay, quality traits are difficult to judge visually. Negative association with the yield, as I told, if you are trying to increase protein content, there is a reduction in quality. Okay, consumer preference varies by region. Different end users need different quality. As I told, in case of rice, as a South Indian, for me, I need somewhat waxy, and the rice grain should be smooth, okay, somewhat sticky. But in case of North India, they prefer basmati. Your rice should be not sticky, okay, that should be spread in their nature. Like that, each and every consumer having their preference. Okay, in Japan and China, they want sushi rice. They need sticky, very much sticky rice. Again in Tamil Nadu, they will prefer sticky rice. Okay, and there it is a consumer preference. Okay, so that it's very difficult to breed for quality. And linkage rank with the undesirable traits. When you are wanting to increase some of the traits, again there will be some undesirable traits are also linked with that particular trait. It is very difficult. Okay, quality traits may have low irritability. One of the major problem, low irritability. We have discussed about exactly what is irritability in the previous class. Okay, if anyone don't know about it, just go and watch that particular part. Okay, when there is a low irritability, it is very difficult to breed for those things. ठीक है, आपका स्ट्रेस ब्रीडिंग हो गया, बायोटिक एबायोटिक, क्वालिटी ब्रीडिंग हो गया. ठीक है, अभी हम we will see regarding the mutation breeding these things. I will take some 2 to 3 minutes break. I will have some energy drink and I will come back to you guys. Please be in the online. Okay, don't go anywhere. If you guys want to drink or anything, you can just go and finish it off. Okay, I will come in 2 minutes. अरे आ जाओ जल्दी आ जाओ, वापस आ जाओ सारे। SK मुत्तुस्वामी एंड D हुसैन, क्या विवेक QPM? SK मुत्तुस्वामी कौन है भाई? सिंचन, विवेक QPM कोई बता सकता है क्या? विवेक QPM कहाँ से डेवलप हुआ था? आई एम टॉकिंग अबाउट विवेक QPM, बताओ। विघ्नेश मुत्तुस्वामी, SK मुत्तुस्वामी लगा है तू। Okay, we are not going to waste time now. So now we will Almora. Good Shruti. It is from Almora. सिंचन बता रहा है विघ्नेश मुत्तुस्वामी और फिरोज हुसैन, वो तो साइंटिस्ट, दे आर फ्रॉम IRI. Okay, that is improved PUSA HQPM1. For that it is from IRI. ठीक है सिंचन? VPKS, yes, Almora. So what about mutation breeding now? In mutation breeding, distant hybridization or your another breeding, which one? Polyploid breeding. Okay. Anyway, everyone knows about it now, each and everything. Okay, wherever the different, as I told, in biotic stress, in abiotic stress, okay, in quality breeding, okay, the varieties which may come from mutation or which may from polyploidy or distant hybridization. Okay, and these are the methods, the special breeding methods. Okay, the mutation breeding. I will not talk into too much about this. Okay, as already you guys know what exactly the mutation and how we can use in the breeding program. I will just give a glimpse. Okay, in mutation and distant hybridization, polyploid breeding. Okay, we will see. Okay, genetic improvement of a crop plants of various characters through the use of induced mutation. Okay, we are improving the genetic improvement of a crop plants through the using mutations. Okay, by using induced mutation. Okay, we will use these in self-monitored and clonal propagated crop plants. Okay, since our crop cross method are they are segregating and it will be in heterozygous condition. Okay, majorly in case of clonally propagated crop plants, okay, we will use this particular mutation where we want to create some variation. Okay, in case of other cross method, we are highly targeted about hybrid breeding, right? There is already somewhat variation is existing. Where there is a no existing variation in that particular crops, we will try to do this mutation breeding. Okay, in the following situation where desired variability is not present, where high yielding varieties have oligogenic defect, where we need to break the linkage drag. Okay, where there is a variability creation, where we want to create some variation. Okay, these are the for perennials, it is a shortcut way for genetic improvement. Okay, where we can easily create some variation. This is about mutation breeding. How? What is the procedure of mutation breeding? I will not talk about the mutagens. Okay, anyway, it will be covered in your genetic classes. But by using those mutagens, we will create mutation. What is the procedure for that? First, we will see the material, the best for a commercial variety or already adapted variety. You will see. Okay, then you will use the choice of mutation. Depend on plant part to be treated, whether it may be floral part, whether it may be seed. Okay, based on that you will select some the mutagens. Okay, for if it is seed, you will go for chemical mutagens. You can use the chemicals to treat the seed so that there will be a mutation will be appear. UV rays, pollen grains. Okay, for pollen grains you can use UV rays, and any other vegetative parts you can go with the radiations based on the based on the different part. Okay, you can use the different mutagens. Okay, what about treatment? LD50, which kills 50% of the treated individuals. Okay, dose differ from the species to species. Now we are not giving LD50. I am telling that what exactly LD50. LD50 is kills 50% of the treated individuals. We will not go up to that much extent. Okay, well before within the 5-10% of LD, we will try to go for treatment. Okay, it will species to species, it will change about the concentration of mutagen. Seeds, how we will do? First we will soak the seeds, desired dose of chosen mutagen, then we will do the washing. Then there are phases. Okay, M0, M1, M2. The treated seeds will be your M0. Then if you grow one more generation, again it will be M1. Then in the next generation it will be M3, M2 and M3, so on. For dominant mutation, you can select in the M1 itself. Okay, since your dominant mutation will be expressed in M1 itself, you can easily select. But for recessive mutations, you should be begin with M2. Okay, अरे कहाँ चला गया सारे? बस 20 लोग, आ जाओ सारे. ब्रॉडकास्ट ಮಾಡಕ್ಕೆ ಅವನಿಗೆ एक बार फिर से ब्रॉडकास्ट कर लो. ब्रेक लिया था मैं, सारे चला गए. फिर से ब्रॉडकास्ट कर रहा है मैसेज. अरे भाई सारे लोग चला गए क्या? इधर आ जाओ अभी. आई एम हियर सर. ओके, if it is you can see here, if it is dominant, if it is recessive, you will treat the seed. Okay, M0, the generation will be M0. Okay, your seeds, the first generation, whenever you will treat the treated seeds, we will call it as M0. Okay, like all are having small a. In case of dominant mutation, okay, what will happen? In any of the loci, there is a mutation from small a to capital A, so that in M1 you will get capital A, small a. So that you will see there are so many seeds. In some seeds there is a mutation, in some seeds there is no mutation. When you will grow M1 generation, this particular genotype, there are mixture of small a, capital A, small 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 a. This is a mixture in case of M1. Once you grow those seeds, okay, but you can start the selection from M1 itself since it is dominant mutation. ठीक है, थोड़ा कट ऑफ के बारे में बता दीजिए सर. मोटिवेशन इज गेटिंग लो. अरे भाई कट ऑफ, what for which college you need cut off? If you want IRI, then you should be get within 10. Out of 3,500 students which they are writing, out of them you should be top 10. Then you will get IRI. Simple thing. If you want to get JRF, then you should be within 60, 50, 60 rank. If you want genetics and with JRF seat. ठीक है, बस आपको सीट चाहिए कहाँ भी. अरे भाई वो तो आराम से मिल जाएगा, बस मैराथन देख के चलो आपका सीट मिल जाएगा. But if you want JRF seat, then you should have put some efforts. Okay, and if you want IRI again, then as I told, you should be within 10 for genetics. For pathology, maybe you can take up to 15, 16 or 20, up to 20, within 20. For any other minor subjects, up to 30, 40 if you are general category. ठीक है, बस नॉर्मल कैंपस. एक्सपेक्टेड नंबर ऑफ कॉम्पिटिटर्स कितने? अरे भाई आप तो अभी मोटिवेशन के लिए आ रहे हैं क्या? हाँ, एक्सपेक्टेड नंबर ऑफ कॉम्पिटिटर्स कितना है वो सब कुछ क्यों चाहिए? आप आपका लगा दो एफर्ट्स. ठीक है, as I told, there will be 3,500 students are writing this exam. Okay, there will be only in plant science for JRF, there will be 400 maybe seats. बस मैं इतना बता सकते हैं। सर आई गेव द मॉक टेस्ट ऑफ Agridict and got रैंक ऑफ अराउंड 4500। वो तो 3-4 बैच का स्टूडेंट्स है ना हमारे साथ, इसलिए 4500 आए। ठीक है, you can start it from M1 itself. You can, then you will go up to some M6 or M7, so that the plant will get homozygous capital A mutation, so that you can release as a variety. This is in case of your dominant, but in case of recessive, okay, the base material is capital A. When you are treat with the mutagen, okay, there is somewhere in the any of the one plant where A will get mutate, so that in case of when you will grow in the next generation, so maybe you will get capital A, small a, capital A, small a, and mixture of capital A, which are not get mutated. Here in case of M1, you are not able to differentiate these two, so you will go to M2. In that case, maybe we will get capital A, small a, capital A, capital A, small a, like that mixture, so that you can start this selection, and you will go till M6, M7, then you will release this particular small a as a variety. Okay, you will start from M2 in case of recessive gene selection, but in case of dominant, you can start it from M1 itself. Okay, ये तो basic है. The basic breeding in case of dominant and recessive, what you should know. Okay, you will start with M0, then M1, M2. Based on that selection, dominant and recessive, you can select in M1 or M2. Then you will go till M6, M7. Then once it got homozygosity, then you can push it for releasing chain. Okay, this is about mutation breeding. Simple. Okay, there is no need to think too much about it. The procedure, as I told, okay, we will start with M0. Okay, treatment, the mutation treatment, then M1, M2. Okay, you will start selection for dominant here. You will start selection for recessive in M2. Then you will go up to M6 or M7. Then you will go for yield trial, and again you will push for varietal trial. This is the procedure for mutation breeding, where we will use most properly asexually propagated majorly, but we will also use in self-pointed and cross-pointed crop plants. Okay. What are the limitations? Low frequency of desirable mutation. When you treat the seeds, it's not always you will get a desirable mutations. It is very, if you are lucky, then you will get out of some when you treat 1,000 seeds, out of 1,000 seeds, maybe in 1 or 2 seeds, maybe there is an desirable mutations what you are expecting. It is very low frequency. Need for quick screening techniques. You have to start your screening M1, M2 itself, so that you can save time. Otherwise, then your treatment and your resources, everything will be waste. Associated with undesirable side effects always. Recessiveness makes detection difficult in case of polyploidy crops. Okay, when there is polyploidy, for example, 6 sets. Okay, A1, 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 and there is a mutation, so that you are getting A2 allele. It is your polyploidy condition. So if you want to identify this particular mutation in polyploidy crops, then it should be comes in like this: A2, 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 A2. That is your like nulliplex condition. You can see how much selfing it needs, how much the probability of getting this homozygosity, this particular allele in homozygous condition. Okay, it is very difficult to get this particular combination in case of polyploidy. So that mutation breeding will be very difficult to detect in case of polyploid breeding, the recessiveness. Okay, these are the limitations. Can anyone tell where we can do the mutation breeding and the treatment, everything in case of India? In Indian perspective, which institute majorly working on mutation breeding? बताओ. Mutations are mostly harmful. अरे कुछ नया बताओ. Mutations is usually beneficial. अरे 1 out of 10,000. Rimi, yes, bark. Where it is located? Baba Atomic Research Center. Okay, from where we are. Okay, whether you are from ICAR Institute, whether you are from different institute, okay, we will just provide our seeds. Those that they will do the mutation treatment, then they will give back. Okay, the one and only institute where we can go for mutation treatment. That's what Baba Atomic Research Center, where it is located. Your bark. In case of earlier days in 1980s, okay, but the first it started in West Bengal, the Atomic Research Center, not this bark. Okay, the mutation breeding station. Now and then in IRI also there is a mutation treatment building was present, but now it is currently not available in case of IRI also. Only the institute which is having that facility is bark. Okay. No, not in IRI. Inchan. There is no mutation breeding facilities in case of IRI. Okay, we'll just provide our seeds from IRI. Then they will do their treatment. Okay, then they will give back. Yes, Mumbai, Trombay, Maharashtra. That's आप इंटरनेट में देखता है क्या सिंचन? इंटरनेट ने फटाफट देख के ट्रॉम्बे लगा दिया क्या? Good. It is in Maharashtra, Mumbai. The region is Trombay. Polyploidy breeding, genetic improvement of crop plants through the manipulation of chromosome number. Okay, your polyploid breeding is nothing but manipulation of chromosome number. Okay, the numerical aberration, anyway, there are structural aberrations also. We don't need it. Okay, you will discuss about everything will be dealt in genetics chapter. In numerical aberration, euploidy and aneuploidy. Okay, in case of euploidy, it is complete manipulation of chromosome sets, complete set modification. In case of aneuploidy, only 1 or 2 chromosomes. Okay. In case of euploidy, monoploidy, diploidy, polyploidy. Okay. In polyploidy, again auto and allo. In case of aneuploidy, monosomy, nullisomy, trisomy, tetrasomy, double monosomy and double trisomy. In case of monosomy, 2n minus 1, 2n minus 2, nullisomy, trisomy, 2n plus 1, tetrasomy, 2n plus 2, double monosomy, 2n minus 1, double trisomy, 2n plus 1 plus 1. Okay, this is the condition of your aneuploidy. And in case of monoploidy, can anyone differentiate between monoploidy and haploidy? Batao, comment mein batao. Sir, to check whether the mutation has happened or not, you will have to put it in the field. Yes, Nutan. Okay, you will give some, for example, as a breeder, I will give 1,000 number of seeds for the treatment for the bark. What they will do? They will just treat your seeds and they will give back that 1,000 seeds, mutation treated seeds. Now, whether that mutation is occurred or not, how you will identify? There will be some mutation. Okay, there will be some mutation, or maybe that's not happened. You have to grow those 1,000 seeds, and you have to see whether your targeted mutation has happened or not. Again, it is very, if you are lucky, then maybe there is a possibility again. Okay, that you are, sometimes you want to do the mutation for seed coat color. Okay, then you will give those 1,000 seeds, then they will give back, then you will see, you will sow the seeds, 1,000 seeds in the field, then you will see, you will look in that amount generation for the change in the seed coat color. As I told, it is, if you are lucky, then maybe in 1 or 2 plants there is a different seed coat color, so that you will get your targeted mutation. Okay, it is very difficult. That's what I told, low frequency. Sir, now the gamma garden is not available in the IRI. Yes, As I told already, it is closed now. Okay, whatever we are doing, which is everything from the bark. Okay, basic number of chromosome is monoploidy. Good, monoploidy is basic, but haploid is half of the somatic chromosome. Good, Vijay. Haploid is the number of chromosome in the gamete, but monoploid is the basic chromosome number. Yes. If you take example of wheat, okay, 2n is 42. Okay, what about n? This is your 21. This is the haploid chromosome number. Okay, but your 2n is equal to 6x, 42. What about x then? What about x then? 7. This is your basic chromosome number. Okay? Yes. This is difference between basic and monoploid. Sorry, haploid and monoploidy. So, autopolyploidy and allopolyploidy. We will talk about these two. Okay? In polyploid breeding. History. Geiger's mutant. You know the first discovery by Lutz in 1907. It is autotetraploid. The first polyploidy which is discovered is autotetraploid with chromosome number changed from 2n is equal to 28 to 2n is equal to 56. Geiger, it is Geiger's mutant in Inothera. This second will be globe mutant. You should remember these are the two important things in case of polyploidy. First one is Geiger's mutant in Inothera, then globe mutant in Datura. It is discovered by Blacksley. Okay? Trisomic mutant it is. That is, it is 2n plus 1. This is autotetraploid. Okay? That is 4x. Tetraploid. It is first reported aneuploid. Okay? It is explained by later explained by Belling. First artificial synthesized species, which is triticale from Rimpo in 1890. Okay? 1890. Then chromosome doubling by colchicine was demonstrated by Blacksley and Avery in 1937. Okay? What is the mode of action of your colchicine? How it will make the doubling of chromosome? बताओ जल्दी. Except rice. What? सिंचन तो कुछ बोलने के लिए जा रहा है. Spindle fiber disruption. Okay, inhibit spindle fiber formation. How it will happen? Anyone knows about it clearly? Okay. Before that, I will ask something else. Yes, it will affect the spindle formation, spindle fiber formation. That is, the thing is, it will affect on tubulin, microtubules. Yes, good. Someone, Pooja, good. Okay, it will affect on microtubules, which are responsible for the spindle fiber formation. So there is no spindle fiber formation. Okay, in case of anaphase, okay, before that metaphase, your chromosomes will arrange. Okay, your chromosomes will arrange. Then there is a separation of your centromere. Then there is no spindle fibers to move those two from one chromosome. There will be two sister chromatids. Then they will not move to the poles, so that they will stay there itself. There is no separation. So what will happen? Your chromosomes, anyway, there is a, in case of rice, there is a 24 chromosomes. Then there is a separation of centromere. Then there will be 48. Then they will not separate. Then it will convert into 48. Doubling of chromosome. ठीक है, हाँ ठीक है, छोड़ दो. Yes, it is no separation of chromosome. Next, my question is, okay, which is this particular colchicine, which is extracted from the which organism? Colchicine is discovered in which plant, or it is extracted from? बताओ. Metaphase will happen, but there is a no separation. That is, anaphase will not happen. ठीक है? Autumn crocus, colchicine, autumn nele. My question is, that particular colchicine chemical is present in that particular plant. Then again, there is no separation. Again, anyway, it is present, that chemical which is present in that particular plant. Then for each and every minute or each and every generation, there is a doubling of chromosome in that particular crop. There is no separation of that chromosomes, spindle fibers. Okay, the plant itself contains that colchicine. Okay, then what will happen? In every generation, there should be a doubling of that particular thing. But why it will not happen? It is resistance against colchicine. Why? How it is having resistance? Anti-colchicine. अरे स्लाइड में देखा था क्या सारे? गुड विजय, गुड रतन. Anti-colchicine. ठीक है. Alkaloid obtained from seed or corn of a plant, Colchicum autumnale, okay, itself has anti-colchicine, so that there is no effect on that particular plant. Okay, if you treat this colchicine in any other than Colchicum autumnale, then there is a inhibition in the formation of spindle fibers. Okay, they will directly attack on microtubules, then there is no spindle fiber formation. So your chromosome will get double due to the non-separation of your chromatids after the centromere division. C22H25O6N. This is the chemical formula of your colchicine. Okay, this is about colchicine. Black C and noble. No. It is discovered by Blacksley and Avery. ठीक है, सिंचन मैं बताया था ना, Blacksley and Avery. If it is mentioned as Blacksley and Noble in case of your books, then you just make a correction. Okay. What are the effects of polyploidy? Thick stem, larger cell size than the diploid. It is like luxuriance, as we discussed in the while we are discussing about the different heterosis phenomena. The luxuriance, okay, which is by large guard cells, stomata, leaves are fleshy, thicker and larger. Your roots are stronger and longer. Okay, flowers, pollen, seeds are larger than diploid. Water content higher than diploids. Water content is the major one trait. Okay, it is based on the effect of polyploidy. Slower in growth and later in flowering. Show reduced fertility. Okay, these are the different effects of polyploidy. Then application of in the crop improvement. Your auto polyploidy, for example, your seedless watermelon. Okay, it is an triploidy. Okay, triploidy. That is your seedless watermelon. Seedless banana, most of your Cavendish variety. Okay, which are triploid in nature. So the seedless will be, we can see in case of banana. Triploid sugar beet, higher sugar. Triploid tea, TV29. Pusa giant berseem, okay, which is 4x, higher green fodder yield. Okay. Then allow polyploidy. Tracing, what are the application here? Allow polyploidy. You can trace the origin of natural alloploid. Okay, chromosome pairing study. Based on the chromosome pairing study between the donor parent and the your allopolyploidy, you can easily trace the origin of the natural polyploidy. जेनेटिक मेड इजी में ब्लैक्सली एंड नोबल दिया है सर. ब्लैक्सली एंड नोबल, अरे मैं एक बुक रेफर किया था, ठीक है, कौन सा, शायद अलार्ड है क्या, ब्रीडिंग उसमें तो दे दिया है ब्लैक्सली एंड अवेरी. ठीक है, मैं एक बार वो कंफ्यूजन करके मैं एक पेपर में भी सर्च किया था. ठीक है, अवे उसमें भी ब्लैक्सली एंड अवेरी था. ठीक है, मतलब कुछ बेसिक्स बुक में शायद बीडी सिंह और इसमें ब्लैक्सली एंड नीबल है शायद, ठीक है, एक बार कंफर्म कर देंगे फिर से देखे, ठीक है। जेनेटिक में और डिस्कवरी ऑफ कॉल्चिसिन, ठीक है, मैं एक और बार कंफर्म कर देंगे, ठीक है, मतलब मेजरली वो ब्लैक्सली एंड अवेरी है, ठीक है, वो नेबल कुछ बुक्स में ऐसे दिया है शायद। Creation of new species like triticale, it is the use of hollow polyploidy. Okay, triticale is the cross between wheat and rye. Okay, your rye sickle cereal, that is your triticale. Interspecific gene transfer from wild related species as a bridging species. You can use hollow polyploids as a bridging species. Okay, first you will cross with that particular species so that it will be helpful in the breeding program. These are some application in case of crop improvement. The most one, the important one we can tell, okay, which is in the market now, that is your seedless watermelon. Okay, you will cross diploid watermelon. First you will treat with colchicine so that you will get a tetraploid watermelon. Then you will use this tetraploid and you will cross with the diploid. Why we will always go with 4x, your tetraploid as a female and your male as a diploid. Can anyone? If you reciprocal this cross, you will then there is a problem with the triploid seed. Why we will go with female tetraploid and diploid male? Yes, Sinchan. Why we will go like this? What's the need of this particular combination? Not like 2x into 4x for the triploid seed production. We will not use this. Why? बताओ। क्रोमोसोमल बैलेंस, अरे कुछ अच्छे से बताओ कुछ वर्ड। anyone else? अरे बताओ। Because triploid have 3 sets of chromosome, the chromosome cannot pair up evenly during meiosis. अरे SN कोचिंग इंस्टीट्यूट, अरे भाई मैं पूछ रहा हूँ why we will use 4x into 2x cross, not 2x into 4x. I'm not asking about triploids. I know triploids having 3 sets of chromosome. Okay, to prevent from somatoplasmic sterility. अरे पता नहीं आप कहाँ से वर्ड लेके आ गया। EBN, एंडोस्पर्म बैलेंस नंबर। अरे सारे उतना ज्यादा डीप मत जाओ, ठीक है। इधर देखो, when there is an production of your female spore, you know na, megasporogenesis. Okay, what will happen? There is a production of 4 megaspore and your 3 will degenerate. Since this 4x, okay, since your 4x is having 4 different sets of chromosomes, while it is producing gametes, maybe there is some aberrations. Okay, out of 4x, what will your gamete will be 2x. Due to some aberration, maybe it will produce some 1x, 3x, like some aberrations. Okay, while it's producing gamete. In this particular megasporogenesis, since 3 will degenerate, if any aberrations in producing this particular 2x gametes, they will degenerate with this particular 3 gametes. You are the most 2x functional. The 2x gamete will be survival. It will be functional. Okay, all other aberrations will be gone through the degeneracy. So your 2x again, anyway, it is normal. Your x will come and they will fuse and you will get triploid seed. But if you make that reciprocal cross, 2x into 4x. Okay, the male, the pollens. Okay, there may be some aberrations in each pollens, so that there will be mixture. The pollens will be mixture of 2x, 1x, 3x, like that aberrations. Okay, the numerical aberrations, so that it is very difficult to produce the uniform 3x triplet 3x seed. ठीक है, समझ गए ना? बताओ. After triplet seed, okay, you will get a triplet plant 3x. Then you need pollination with diploid pollen again. Okay, anyway, we will give this triploid seeds to the farmer. Then they will grow triploid plants, and also they have to sow some diploid pollinizer plants in between, so that from that, anyway, the triploid 3x, they will not produce functional pollens. Okay, it is completely sterile 3x. Then they need normal diploid parent as a male parent to stimulate the fruit development. Okay, there is no, anyway, your triploid will not produce any proper functional pollens or any proper functional egg cells. Okay, they just need some stimulation from normal pollens, so that your fruit will develop and your fruit will be seedless. Okay, since again there will be some seeds, those are rudimentary. Okay, not fertile seeds, and also they will not feel like a seeds. Okay, it is your seedless watermelon. Okay, pseudogamy, yes, they need some stimulation. Okay, rudimentary seeds are nothing but you will not feel those are seeds. Okay, that's what the phenomenon in case of seedless watermelon. What you should remember, the cross, it should be 4x into 2x, and you need pollination with the diploid pollen. Okay, need for the fruit set. Okay, diploid pollinizer. Undeveloped seed. Okay. Then distant hybridization, that is all about polyploid breeding. Okay, there are not that much achievements in case of polyploid breeding. That's why I'm not going into too much about it. Then distant hybridization is cross between individuals belonging to different species or genera. Effective means of transferring desirable genes into cultivar plants from this related species. Okay, any biotic or abiotic stress genes which are having in the wild relatives, in the wild germplasms. Okay, how we can transfer? That is through the distant hybridization. Okay, interspecific or intergeneric. In case of interspecific hybridization between different within species, okay, between different varieties. Sorry, that is intra. It is interspecific between different species of the same genera. Okay, between different species of same genera. That is your interspecific hybridization. What about intergeneric hybridization? Between different genera, completely different genera. That is your intergeneric hybridization. Okay, bulbosome technique in barley. Example for which? That is example for interspecific or intergeneric. bulbosome technique in barley. बताओ. Chinchan बताओ. What about triticale? Wheat into rye. That is interspecific or intergeneric? बताओ जल्दी बताओ. What are the difference between interspecific and intergeneric? Two different species of the same genus, two different genera of the same family. Such hybrids are vary from completely fertile to completely sterile, but here always sterile in intergeneric. Here there is a chance of some fertility. More than intergeneric crosses, seed set, here it is too much low. More than the interspecific we will use in case of breeding program, okay, in crop improvement, but intergeneric will be very much less. Okay, we have released some possible hybrids also in case of interspecific, but in case of this it is not possible at all. बताओ, मैं क्वेश्चन पूछा था एग्जांपल्स फॉर इंटरस्पेसिफिक एंड इंटरजेनेरिक. Triticale is intergenic. Good. Triticale is intergenic. What about interspecific? Intergeneric is as इंटर स्पेसिफिक. हाँ अरे भाई दोनों डिफरेंट है, इंटर generic and inter specific. I have told the differentiation also. Why you are having the doubt? Jayalakshmi, Varalakshmi. अरे वरलक्ष्मी क्या हुआ आप लोगों के लिए? हाँ ठीक है ठीक है ठीक है, कार्टन में बता रहे हैं, कुछ ऐसे नाम लगा रहे हैं ऐसे सोचा था. इंटर स्पेसिफिक जयलक्ष्मी वरलक्ष्मी, ठीक है, that is between Gossypum hirsutum and Barbadans. Good example. Yeah. Okay, your critical, that is between wheat and rye, that is intergeneric. ठीक है. Any doubts? Then what are the barriers for this distant hybridization? There will be pre-fertilization barriers and post-fertilization barrier. Before fertilization, there will be some barriers. Okay, cross incompatibility barriers, failure of zygote formation, lack of that is pre-fertilization is failure of zygote formation. Post-fertilization is failure of zygote development. Okay, zygote will form, then the development will not happen. Okay, what about the pre-fertilization barrier? Lack of pollen germination, insufficient growth of the pollen tube. Okay, the pollen tube may burst in the style of other species if it is too much distant. You can see lack of pollen germination, insufficient growth of pollen tube burst in style or other species, inability of male gamete to unite with the egg cell, long style, small pollen tube, maize, female, tripsacum male. If used like that, then there is an long style and small pollen tube, so that there is no fertilization. We'll call it as cross incompatibility. Okay, these are the different reasons. Then what about post-fertilization barriers? Anyway, the zygote will form, then but it will not develop. First one is your hybrid inviability. Okay, what it will happen? Your hybrid will inviable. Fertilization occurs, zygote form but unable to grow into normal embryo. What are the factors? Some lethal genes. In Agileps ambulata, there are some genes which are lethal. Okay, unfavorable interaction between chromosome of two different species. It may leads to the hybrid inviability. Disharmony between cytoplasm and nuclear genes of two different species, and endosperm abortion will happen. These are the different things. Okay, the different reasons where your hybrid will inviable. Inviable is nothing but there is a formation of zygote, but it will not develop. It is inviable. That is your hybrid inviability. What about hybrid sterility? Your hybrid will form, zygote will form, it will develop, but in the next season there is a sterile. That is your hybrid sterility. Decay inviability, inability to produce viable offspring, lack of structural homology between chromosome of two species. It will leads to the hybrid sterility. No pairing. Okay, there are so many reasons. Okay, you can see here. No pairing or reduced pairing. It may leads to the gametes which are not proper. There will be so many aberrations. Okay, so that there will be sterility in the upcoming generation. Okay, your F1 will come, but that hybrid will not produce functional gametes, so that there is a sterility will occur. Irregular anaphase separation, it is completely related to your meiosis, so that you can observe sterility in case of F1. The third one, that is your hybrid breakdown. Okay, your hybrid will form, zygote will form, it will produce fertile male gametes in the next, male and female gametes in the next generation, but it will not be not that much durable. Okay, it will break down in one or the other generation, maybe in F2 or maybe in F3. That is the breakdown. Major issue in the interspecific crosses, F1 is vigorous and fertile, but F2 progeny is weak and sterile, maybe in F2 or F3. Okay, maybe due to the gene combination or structural differences in case of your F1 hybrid, so it may get breakdown. Okay, these are the different post-fertilization barrier. Okay, pre and post-fertilization barrier. These are the very important. Okay.
जूस पी ले भाई, मार दोगे क्या सबको? कितना टाइम हो गया? 9:32. गाइस, क्या हो रहा है? खत्म करना है नहीं करना है? ब्रेक लिया था बीच में?
लिया था 10 मिनट।
अच्छा 10 मिनट।
हाँ।
अच्छा ओके, अभी 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 भी
नहीं, अभी आधा घंटे हो गए।
ओके। अभी भी 50 स्टूडेंट्स है, ये सॉलिड 50 स्टूडेंट्स है जो लास्ट 1 वीक के लिए बहुत कंसिस्टेंट होंगे। सर सीइंग फ्रॉम लास्ट all the marathons कि almost like 50 students are very consistent। बाकी बाद में आई नो लाइक 1.5x में 2x में स्टूडेंट्स, सारे स्टूडेंट्स देख रहे हैं। यहाँ से आपको कंपटीशन का अंदाजा लग जाएगा यहीं से कि इस वीडियोस में कितने व्यूज आ रहे हैं ना, उतने लोग एक्टिवली देख रहे हैं। उसमें अगर तुम 10% ऑफ स्टूडेंट्स को निकाल भी दोगे जो थर्ड ईयर के होंगे या सेकेंड ईयर के होंगे, बाकी लोग आपके कॉम्पिटिटर्स ही है। सो दे आर ऑल योर कॉम्पिटिटर्स। सो इसका एक आइडिया मिल जाएगा। हम एक और चीज़ भी सोच रहे थे ना, सो वी आर ऑल्सो, सर सीटेक का बुक का वेट, अच्छा आ जाएगा ना, बुक तो आ गया ना। देखो मैं इतना इसलिए रिलैक्स्ड हूँ क्योंकि बुक आ गया, वरना तो मैं पागल जैसा घूम रहा था बिना एयर कट करा के। सर क्रैश कोर्स फॉर ये अच्छा। कितना एग्जाम के लिए पढ़ोगे भाई? So like I was telling one thing, what we are planning ना for the next last 1 week or 10 days, हमने एक चीज प्लान कर रहे थे। I don't know like how, whether it will be useful or not, but we want to conduct like one mega mock test for all of you, just to know like how you people are prepared and what is the competition among yourself, जिससे कुछ लोग ऐसा भी बोलेंगे कि सर इससे हमारा कॉन्फिडेंस डाउन हो जाएगा रिजल्ट से। फ्यू स्टूडेंट्स विल से सर यस वी नीड इट। सो व्हाट डू यू थिंक, चाहिए या नहीं चाहिए? कितने लोग बोलोगे चाहिए, कितने लोग बोलोगे नहीं चाहिए? व्हेन विल द जेनेटिक्स मैराथन स्टार्ट्स? जेनेटिक मैराथन्स कल से स्टार्ट करेंगे, कल या मैक्सिमम परसों से। प्लेस द ऑर्डर। गुड। सो जिन्होंने भी ऑलरेडी सीटेक का बुक ऑर्डर किए हो, थैंक यू सो मच। सो इट विल बी लाइक योर लाइफ लॉन्ग फ्रेंड। ऐसा नहीं कि सिर्फ पीजी के लिए यूजफुल होगा। सी रियलिटी सबको पता है, आपको भी पता है कि आप क्या करोगे आगे जाके, लाइक व्हाट यू आर गोइंग टू डू आफ्टर दिस एमएससी। एमएससी एक इनिशियल स्टेप है। ऐसा मान लो कि एक पहला स्टेप है प्रोफेशनल डिग्री के बाद हायर स्टडीज के लिए। उसके बाद व्हाट यू आर गोइंग टू डू, एक माइंडसेट तो सबका होगा ही। अगर क्लैरिटी नहीं है, क्लैरिटी बना लेना। यू आर एट द एंड ऑफ द डे you are going to prepare for any of the government exams. That will be the major priority अगर आपको job लेना है। So the best option that you can get in front of you being a MSc graduate will be preparing for the government exams. First will be the ARS, other than the SMS, STO. Other than that, there are central government exams to which you will be preparing. But you take any of the exam, you have to cover all the general agriculture except ARS exam, which is subject specific. So no matter like whatever time you are investing in learning these concepts, understanding the things, writing the notes, taking the textbooks, everything will count, okay? Everything will help you in some point of time। ऐसा इतना कैलकुलेटिव मत बनो कि हर एक सेकंड पे सोचने लग जाओगे इतना मेहनत करके क्या फायदा, क्या फायदा, क्या फायदा। ऐसा कुछ नहीं है। जितना आप अभी मेहनत करोगे तो कभी ना कभी फायदा आ ही जाएगा। कभी ना कभी क्या, अगले 2-3 साल से लेकर आपका इसका आंसर मिल जाएगा, आपको ये आंसर मिल जाएगा। यू विल ऑल्सो गेट दिस आंसर। यस, करंट अफेयर्स 2 चीजें मेरे साथ प्लान्ड है, एक जनरल एग्रीकल्चर, एक करंट अफेयर्स। हमने 2 चीजें प्लान करके रखे हैं। लाइक वी हैव प्लान 2 थिंग्स। वन इज we will be discussing all the general agriculture questions which have been asked in the previous year ICAR PG exam, and not only for the one subject, from all the subjects। क्योंकि इसमें प्रोबेबिलिटी ज्यादा बनते हैं कि जनरल एग्रीकल्चर का क्वेश्चंस जो बाकी सब्जेक्ट्स में पूछा गया है उस क्वेश्चंस आपके में आ सकता है। सो दैट्स व्हाट वी आर गोइंग टू डू। वन सेट विल बी कीपिंग इट all the general agriculture questions from the PYQs of all subjects, we will be discussing it, and the other part is like current affairs, agriculture current affairs with respect to the data, varieties, institutes, awards, schemes, like we will be discussing in one session. That current affairs section I will be discussing, and the general agriculture part, our new general agriculture mentor Vasundhara ma'am, she will be discussing it after genetics and seeds and marathon mock, but after genetics and seeds and marathon। ठीक है, ठीक है, हो जाएगा। एग्री बॉय सेशंस फॉर करंट अफेयर्स, हाँ मैंने बोल दिया ये वो क्रैश कोर्स। वन शॉट जनरल एग्रीकल्चर मोस्ट एक्सपेक्टेड क्वेश्चंस, ओके दैट विल बी ग्रेट। बस काफ़ी है भाई। स्टूडेंट्स लाइक इससे ज़्यादा कंटेंट कुछ मत कंज्यूम करो, दिमाग फट जाएगा। ठीक है, हम देने के लिए क्या, हम तो रोज क्लास कर सकते हैं, रोज कुछ ना कुछ बोल सकते हैं। इतना हम पागल है कि हम एक बार सब्जेक्ट स्टार्ट कर लिया ना तो मन करेगा पूरा बता दे। बट इस राइट टाइम पे क्या राइट डिलीवरी देना है वो हमें भी सोचना चाहिए ना। अगर बोलो तो मैं पूरा जेनेटिक्स पढ़ा सकता हूँ फिर से, लेकिन दैट शुड बी यूजफुल एट दिस लास्ट पॉइंट ऑफ टाइम। इतना ज़्यादा कंटेंट पे लाइक रिले मत होना, जितना कंटेंट मिल रहा है उसके अलावा गिव मैक्सिमम टाइम इन रिवाइजिंग व्हाट यू हैव ऑलरेडी लर्नड। ठीक है, जितना आपने ऑलरेडी पढ़ लिया है उस पर ज़्यादा फोकस करना। फोकस आपका इसमें 30 to 40% होना चाहिए, और 60% तक आपका फोकस सेल्फ स्टडी में। और सेल्फ स्टडी का मतलब जो नोट्स आपने बनाया है, जो बुक से आप ऑलरेडी पढ़ रहे हो, रिवीजन पे होना चाहिए। ठीक है, इतना ज़्यादा देखते रहोगे देखते रहोगे देखते रहोगे, अगर आपको याद करने के लिए खुद के लिए टाइम नहीं हो ना, तो एग्जाम में प्रॉब्लम होगा। So keep the mock after all the marathons 1 day revision so that we can at least go through it once. Yes, we'll keep it in the 1st of July या 2nd of July को ही रखेंगे। ठीक है, उसी टाइम पे रखेंगे। सर आई एम ओनली रेफरिंग द हैंडबुक एंड अग्रीड नोट्स, नथिंग एल्स। मोर देन एनफ है। लाइक आप हैंडबुक से जा रहे हो, एग्रीड मास्टर कोर्स के क्लासेस से जा रहे हो, या मास्टर क्लास के क्लासेस फॉलो कर रहे हो, प्लस उसके साथ एग्रीड नोट्स फॉलो कर रहे हो, प्लस उसके साथ मॉक टेस्ट अटेंड कर लिया आपने, एंड फाइनली यू आर रिलाइंग अपॉन दिस मैराथन। उससे पहले अगर मोस्ट एक्सपेक्टेड सीरीज देख लिया, सो दिस थिंग्स आर मोर देन सफिशिएंट। इतना ही है। अगर मैं आपको दोबारा एक चीज अच्छे से बता दूं, so what you have to like what you can rely on this. First one is about master class. Second one is aggregate notes जो uploaded है. Third one, all the crisp notes which are uploaded in the short form. Fourth, you attend all the topic wise and full length mock test. Fifth, you have gone through the most expected. अगर मोस्ट एक्सपेक्टेड में ऐसा हुआ कि आपने वीडियोस देख नहीं पाए, बट एटलीस्ट गो थ्रू द पीडीएफ्स तो सबके पास होगा ही, क्योंकि हमने सारे पीडीएफ्स को गूगल ड्राइव लिंक में डाला है। मोस्ट एक्सपेक्टेड, अगर आपके पास अभी तक मोस्ट एक्सपेक्टेड क्वेश्चंस का पीडीएफ नहीं मिला है, तो डिस्क्रिप्शन बॉक्स में एक गूगल फॉर्म रहेगा, उसको फिल कर दो। उसके बाद आपके पास डायरेक्टली ये मोस्ट एक्सपेक्टेड क्वेश्चंस का पीडीएफ आ जाएगा। पीडीएफ प्लस वीडियोस जो आपको सही लगे, मैं तो कंपलसरी नहीं बोलूंगा, जो टाइम है बचा हुआ उसमें जो आप कर सकते हो उसको कर लो। एंड द सिक्स्थ पार्ट, द लास्ट एंड द फाइनल क्लाइमेक्स पार्ट इज अबाउट मैराथन। लिख के ले लो, अगर इतना सॉल्व कर लोगे ना, इतना बहुत ही अच्छे से, लाइक इफ यू डू इट विद द 100% डेडिकेशन एंड कंसिस्टेंसी, कि इसमें आपने, मैं कभी 100% का बात नहीं करता हूँ, इफ यू कवर लाइक 70 to 80% ऑफ इट, इससे आपने 70 to 80% भी आपने अच्छे से कर लिया। मतलब करना तो पूरा है, बट करने के बाद अगर इस पे से आप 70 to 80% भी आप याद रख लिए, so if you are able to remember at least 70 to 80% of all these things, still you will definitely get JRF seat. JRF तो पक्का लेके रहोगे। जैसा मैंने बोला भी था, ऑन एन एवरेज AR1 का रैंक कहाँ पे आके पड़ता है प्लांट साइंस में, इट इज समवेयर अराउंड 400। 480 में 400 होगा जो AR1 का रैंक आएगा। सो ऑल इंडिया 1 विल बी समवेयर नियर टू द 400। उसका मतलब क्या हुआ? सो दैट्स दिस डायरेक्टली मींस 100 क्वेश्चंस तो सही होने ही होने हैं, जो गलत नहीं होना है। सो यू हैव टू मेक 100 क्वेश्चंस राइट इन द एग्जाम्स। नाउ इफ यू आस्क योरसेल्फ, सर योर बिहेवियर इज नॉट राइट, इज इट? ये क्या है? क्या हो गया बिहेवियर को? ये नया सवाल आ गया। तबियत इंग्लिश में ट्रांसलेट हो गया। लाइक तबीयत ठीक नहीं है, तबीयत तो ठीक है, बट वही है ना, लाइक इट्स बीन लाइक मोर देन 16+ आवर एवरी सिंगल डे फ्रॉम लास्ट 1 or 2 weeks। सर गूगल फॉर्म का लिंक मिल जाएगा। सॉरी कि क्या बोल रहे हैं, और आस्किंग अबाउट योर हेल्थ। अच्छा, समझ गया। ओके, तो लास्ट 1 week से थोड़ा ड्रास्टिकली बहुत कुछ देना पड़ा, जो आपने प्लान भी नहीं किया था कि इतना देना पड़ेगा। दैट्स व्हाट। सो कमिंग बैक, लाइक व्हाट आई वाज डिस्कसिंग, कि ऑल इंडिया 1 के लिए, ओके थोड़ा पिच ज्यादा कर लेता हूँ। ऑल इंडिया 1 के लिए 400 होगा, आसपास के स्टूडेंट को ही ऑल इंडिया 1 रैंक मिलेगा प्लांट साइंस में। ठीक है, उसके लिए आपको कितना क्वेश्चन सही करना होगा? यू हैव टू मेक लाइक 100 क्वेश्चंस राइट। नाउ आस्क योरसेल्फ लाइक हाउ मेनी यू आर मेकिंग इट राइट इन द फुल लेंथ मॉक टेस्ट। जितना भी फुल लेंथ मॉक टेस्ट है, इतना कर रहे हो। मैं लिख के दे देता हूँ, इतना तो मिनिमम कर रहे होंगे, मैक्सिमम नंबर ऑफ स्टूडेंट्स आर स्टकिंग समवेयर अराउंड 60 to 70 क्वेश्चंस, यस और नो? इतना मिनिमम करेक्ट कर ही रहे हो, यस और नो? हाउ मेनी ऑफ यू से यस कि यस सर मिनिमम तो ऑन एन एवरेज 60 to 70 क्वेश्चंस कर रहे हैं? थम्स अप लाइक इफ इट इज यस दैट यस सर वी आर डूइंग एटलीस्ट 60 to 70 क्वेश्चंस हमारा सही हो रहा है सारे मॉक टेस्ट में. हाउ मेनी ऑफ यू से यस? देखो अराउंड 80 to 85 ग्रेट. सी 70 to 60 to 70 एंड अबव, ठीक है, 60 एंड अबव, मान लो कि 60 एंड अबव. आई नो लाइक सारे लोग तो आंसर नहीं करेंगे यहाँ पे, बट मेजॉरिटी जितने लोग भी एटलीस्ट 3 महीने से प्रिपेयर कर रहे होंगे ना, इफ यू आर एटलीस्ट गिवन लाइक डेडिकेटेड 3 to 4 months of your time, even if you're not given the 3 to 4 months of your time, but still you have some good residual knowledge from the 4 years of basic agriculture, still you will be able to get 60 to 70 plus. If you are securing like 95 plus, it's well and good, you will be definitely a topper if you do it same in the main exam. सिंचन तुमने तो नाम नहीं बताया तुम्हारा, एटलीस्ट रिजल्ट के बाद बता देना, ठीक है? Okay. So 60 to 70 questions, like I guess like most of you who are preparing seriously, you are pushing it. देखो रैंक 1 में और तुम्हारे में कितने डिफरेंस है? सी डिफरेंस 30 क्वेश्चंस में होंगे बस मैं बस इसलिए बोल रहा हूँ कि 60 to 70 questions which you are making right, which will be like 100% of the things that you will be knowing. इसमें 100% वही क्वेश्चंस होंगे जो आपको पता होगा, ठीक है? Now the next part is like you have to make a guesswork, use the proper elimination method and ensure कि you push next 30. ऐसा होना चाहिए एग्जाम में आप 110 115 तक अटेंड करते आओ, इसमें आपका लगभग गलत होने का चांसेस 10 से 15 होना चाहिए, बाकी आपको करेक्ट करके ही आना है. एंड anywhere if you are pushing around like 110 to 115, if the paper is medium to hard, देखो मीडियम टू हार्ड होगा तो अगर एक्सट्रीमली पेपर डिफिकल्ट आ जाएगा या एक्सट्रीमली मीडियम टू डिफिकल्ट लिख देते हैं अगर पेपर या ये भी याद रखना लाइक इफ द पेपर विल बी लाइक एक्सट्रीमली डिफिकल्ट, तो एवरेज नंबर ऑफ क्वेश्चंस दैट यू शुड अटेंड विल कम टू 90 to 100 एक्सट्रीमली डिफिकल्ट केस में. बट मेजॉरिटी ऑफ द केसेस जितने भी पेपर्स हमने देखे हैं 2020 से लेकर 2025 तक, सारे इसी लाइन पे पढ़ते हैं और जितने भी टॉप रैंकर्स आए हैं उस टाइम से, सो दे हैव स्कोर्ड अराउंड 390 to 406. AR 1 का बता रहा हूं मैं, इतना स्कोर किए हैं 2021 से लेकर यहाँ तक, ठीक है? तो इफ सेम ऑब्वियसली लाइक सेम पेपर विल कम, सिमिलर डिफिकल्टी लेवल विल कम, बट एट अ वेरी रेयर केसेस इट माइट गेट डिफिकल्ट. एक्सपेक्ट करेंगे कि ऐसा नहीं होगा, तो फिर तो आपका टारगेट यही होना चाहिए कि 110 115 इतना तो एवरेज करके आना चाहिए अगर इफ यू वांट द JRF सीट, इफ यू वांट द ऑल इंडिया टॉप रैंक, इफ यू वांट द IRI. फोकस सबका मैं अभी भी बता रहा हूँ, जितने लोग भी लाइक 60 to 70 पुश कर रहे हो ना, ईच वन ऑफ यू आर केपेबल टू गेट द JRF सीट. JRF फेलोशिप लेने के लिए आप सब लोग एलिजिबल हो अगर आपका 60 to 70 क्वेश्चंस अभी मॉक टेस्ट में सही हो रहे तो. सो बट मे इंश्योर कि नेक्स्ट 30 क्वेश्चंस को कैसे टारगेट करोगे, कैसे टैकल करोगे एंड हाउ डू यू प्रैक्टिस इट, दैट विल गिव यू द रैंक. इसको बाद में सोचोगे ना, एग्जाम के बाद लगेगा कि अरे 5 क्वेश्चन ही थे, 10 क्वेश्चन ही थे, थोड़ा एक्स्ट्रा एफर्ट लगा देता तो आ जाता. लाइक इफ आई वुड हैव मेड लाइक 1 डे एक्स्ट्रा एफर्ट, इफ आई गॉट लाइक 1 डे एक्स्ट्रा, या मैं इस सब्जेक्ट को थोड़ा और पढ़ लेता, थोड़ा 2 बार और पढ़ लेता तो आ जाता. ऐसा रिग्रेट्स डेफिनेटली दिमाग में आएगा एग्जाम के बाद. तो एटलीस्ट आपको बोलने वाला एक इंसान है, लाइक यू हैव समवन हु विल से दिस दैट दिस इज व्हाट इज गोइंग टू हैपन, अ स्ट्रिक्ट वार्निंग टू ऑल ऑफ यू. तो एटलीस्ट जब भी आपको लगे ना कि हाँ मैं एग्जॉस्ट हो चुका हूँ, पुश इसलिए करना, इस सारे चीज को याद करके थोड़ा और एफर्ट डालोगे, थोड़ा और एफर्ट डालोगे, डेफिनेटली बहुत डिफरेंस आएगा. एक बार आप JRF की कैटेगरी में आ जाए ना, the way you look yourself, the way you treat yourself and the way people look you will be entirely changed. बहुत change हो जाएगा उसमें. You will start feeling confident about yourself the day you know that you got a JRF fellowship. यही motivation होना चाहिए last point के लिए. इस बहुत ही realistic way में बता रहा हूँ क्योंकि इस सर्कल में majority of the students तो पढ़ रहे होंगे, right? 60 to 70 question, it's like too much easy for majority of the students. ठीक है, 50 एंड अबो हो देन दिस इज एप्लीकेबल फॉर ऑल ऑफ यू, सबके लिए बोल रहा हूं मैं, जितने भी लोग 50 से ज्यादा सही कर रहे हो, ठीक है। सो फोकस ऑन दिस, गिव दैट एक्स्ट्रा मिनट, एक्स्ट्रा आवर, एक्स्ट्रा डे। जब भी आपको लगेगा कि बहुत ज्यादा एग्जॉस्ट हो चुका हूं मेरे जैसे, जैसा मैं पुश कर रहा हूं, आप भी पुश कर लेना। इट्स अ मैटर ऑफ जस्ट अनदर 10 डेज, बहुत ही कम डेज बचे हैं। कम डेज इसलिए बचे कि ज्यादा मेहनत नहीं करना पड़ेगा, कम डेज के लिए मेहनत करना पड़ेगा, इसलिए बोल रहा हूँ कम डेज बचे हैं। अगर पढ़ने के लिए हो तो ज्यादा दिन बचे हैं आपके पास, बट ऐसे सोचने के लिए कि कितना दिन और मेहनत करना पड़ेगा, सिर्फ और सिर्फ 10 दिन के लिए मेहनत करना पड़ेगा। यू हैव टू ओनली फॉर नेक्स्ट 10 डेज। पीएयू में जेनेटिक्स में सीट चाहिए, मिल जाएगा आराम से, 90 प्लस 95 प्लस क्वेश्चन सही करके आ जाओ, आराम से मिल जाएगा। विल यू ऑल डू इट? करोगे सब लोग? मैनिफेस्ट करो अंदर से, सिर्फ मेरे बोलने से नहीं। यू विल पुश फॉर 90 प्लस क्वेश्चंस मिनिमम इन द नेक्स्ट मॉक टेस्ट दैट यू आर गोइंग टू अटेंड। अभी से मैं बता रहा हूं, नॉट इन द फाइनल, नॉट ओनली इन द फाइनल एग्जाम, बट फ्रॉम द नेक्स्ट फुल लेंथ मॉक टेस्ट दैट यू आर गोइंग टू अटेंड, push yourself for 90 plus, and I don't know like how many of you are dedicating like 2 hours of time. मैं लिख के दे देता हूँ, 99% ऑफ स्टूडेंट्स दो घंटे का एग्जाम दिया ही नहीं होगा अभी तक जितने भी मॉक टेस्ट आपने अटेंड किया है ना, आई डोंट थिंक लाइक आप लोगों में ज्यादातर स्टूडेंट्स 2 आवर्स पूरा यूटिलाइज किया होगा। एटलीस्ट इसके बाद मैं 2 घंटे पूरा नहीं बोलूंगा, एटलीस्ट 1.5 आवर्स या 1.45 मिनट्स पूरा यूटिलाइज कर लेना। एटलीस्ट मैं बता रहा हूँ 145 मिनट्स या 1.5 घंटा जो भी बोलोगे यूज कर लेना, मिनिमम इतना 1.3 या 1.45 मिनट्स यूज कर लेना, 1 आवर 45 मिनट्स इतना तो मिनिमम यूज कर लेना। और अच्छे से जो क्वेश्चंस आपको बिल्कुल पता ना हो, लाइक द क्वेश्चंस व्हिच यू डेफिनेटली डू नॉट नो, यू डू नॉट नो एनीथिंग अबाउट इट, still try to attend them. At least make sure that if you attend such kind of question 10, at least 5 should be correct. 5 should be correct. ये प्रैक्टिस के साथ डेफिनेटली आएगा। इफ यू यूज लाइक एलिमिनेशन मेथड, इफ यू प्रॉपरली यूज योर ब्रेन, यू कैन डू इट। ऐसे AR1 को भी जाके पूछोगे, इसका मतलब नहीं होगा कि he knows she knows everything in the exam. They have like utilized some category, like some trick, and it has worked for them. It will also work for you. Ultimately, hard work matters. ठीक है? देखो 50-55 मिनट्स, 40-45 मिनट्स, देखो यू हैव टू आल्सो ट्रेन योर ब्रेन्स सो दैट इन द रियल एग्जाम you should be comfortably able to sit for 2 hours where your brain is continuously working. अगर इसका प्रैक्टिस आपने एक बार भी नहीं किया है तो एग्जाम प्रेशर में बैठ तो लोगे, आपके पास कोई ऑप्शन नहीं है, उठ के तो नहीं जा सकते, बट कितना इफेक्टिवली आपका ब्रेन काम करेगा उस पर थोड़ा इफेक्ट पड़ेगा। ओके, सो दैट्स व्हाई एटलीस्ट 1 और 2 एग्जाम के लिए पूरा 2 घंटे बैठ जाना, बाकी एग्जाम्स के लिए एटलीस्ट 1 आवर 30 मिनट्स या 1 आवर 45 मिनट्स तक देना। एंड कोशिश करना, पुश करना नंबर ऑफ क्वेश्चंस को बढ़ाते जाने के लिए। अगर फर्स्ट पेपर में आपने 120 में से 100 किया या 90 किया तो पुश इट टू मोर एंड मोर। अगर 120 से 120 भी कर रहे हो, जिसने ये बंदा जो पढ़ा रहा है ना वो 120 करके आया था, so that's also good, like he secured all India 20 and 21st rank in ICAR PG exam. I attended 113 questions in which like 94 were correct, so I secured all India 48. See the difference, बहुत difference है। So he got genetics, I got C tech, बहुत आगे आ चुके हैं, पीछे भी नहीं जा सकते अभी। So सोच लो, एक्स्ट्रा पुश कर लो, हो जाएगा, ठीक है। सो कंटिन्यू करे। ब्रेक अच्छा था, मेंटर के लिए ब्रेक था, आपके लिए तो ब्रेक नहीं था, बट फिर भी कितने स्लाइड बचा है भाई अभी? हाउ मेनी स्लाइड्स आर देयर?
20-25।
सो हाउ मच मोर टाइम विल टेक?
1 आवर।
ठीक है, सो जस्ट बी विथ हिम फॉर 1 मोर आवर, उसके बाद पूरा रेस्ट कर लेना कल सुबह तक। कल पूरा दिन रेस्ट करना हो तो मेंटली रेस्ट कर लेना। मैराथन से हम परसों स्टार्ट करेंगे, ठीक है। ओके गाइस, गुड नाइट। ये 1 और घंटा भी इफेक्टिवली बैठ जाना इसके साथ। दोस नंबर्स आर द मोटिवेशन एक्चुअली। जो नंबर्स वहां पे दिख रहे हैं ना 50 to 50, अनलेस एंड अंटिल दे आर कांस्टेंट, मेंटर विल बी वेरी हैप्पी टू डिलीवर हिज बेस्ट। ओके, देखो मेरे आते ही व्यूज बढ़ गए अभी।
कहा है यार,
800 था, अभी 900।
अरे वो कंसिस्टेंटली वो बढ़ रहा है, ठीक है। 50 था मैं जाने समय, अभी भी 50 ही है, ठीक है। 100 हुआ तो मैं बोलता था, ठीक है। नाउ वी हैव ऑलरेडी सीन व्हिच आर pre-fertilization barrier, then post-fertilization barrier. Then what are the techniques to overcome those barriers? How we can avoid those barriers so that we can successfully get some distant hybridization products. Okay. What are the techniques? Choice of parent. अरे ये तो बहुत छोटा करके गए है ब्रश, मुझे थोड़ा बड़ा चाहिए. हाँ, इतना बड़ा चाहिए. Choice of parents. Okay, based on that, which are the parents you are choosing, so you can overcome the barrier. You will try with different parents so that maybe there are some crosses, you will get some fertile hybrids. Okay, reciprocal crosses. As I told, 4x into 2x, it is not happening. You are not getting fertile. Then you will go for 2x into 4x. Okay, if it is 2x into 4x is not happening, then 4x into 2x. Okay, you will just shuffle the parents so that maybe you will get some success in case of wide cross hybridization, that is your reciprocal crosses. Okay, manipulation of ploidy or chromosome doubling. Okay, when you will cross two different species, any two different genomes, for example AA into BB, these are completely two different genome. Maybe you can take the two different genera. Okay, then you will get F1, which is AB from A and B. This is your F1, though it is completely sterile. There is no pairing among the different genomes. What you will do? You will go for chromosome doubling, AABB, so that you will get some fertile product. Okay, so manipulation of ploidy level or chromosome doubling is one of the technique to overcome. Then bridge crosses. Sometimes in case of wheat, you can't directly transfer your any wild gene from wild progenitors. What you will do? First, you will transfer into the intermediate, that is your AABB genome, that is your tetraploid wheat, Triticum turgidum. First, you will cross to this Triticum turgidum. Then this Triticum turgidum you will cross with normal wheat, AABB hexaploid wheat. Okay, so you will use this particular Triticum turgidum as bridge species so that you can go for wild cross. First, you will cross your wild progenitor with this Triticum turgidum. Then you will cross with normal hexaploid wheat. Okay, this is bridge crosses. Use of pollen mixture. When you are crossing with different genome as like this, okay, using these pollens with the normal AA pollens, so that there is more chance of that pollen germination and effective fertilization. Okay, these are the pre-fertilization barriers. How to overcome pre-fertilization barriers? There are some things. Okay, this is about post-fertilization. After fertilization, if you double the chromosome, so you can get some fertile inbreds. Okay, those are some post-fertilization barriers. Then manipulation of pistil. ठीक है, क्या कर सकते हैं manipulation of pistil मतलब? You can remove the stigma. Okay, that will be barrier sometimes. You can remove pistil so that you can directly transfer your pollens to the embryo sac. Okay, these are some techniques. Manipulation of pistil, use of growth regulators. It will improve your germination and fertilization. Okay, large number of crosses, so there is a chance of getting fertile hybrids. Protoplasm fusion. This is the one of the pre-syngematic, that is pre-fertilization barrier. Where there is an problem with the fertilization, you can directly we can directly hybridize two protoplast so that you will get an hybrid, distant hybrid between potato, tomato, protoplasm fusion. Okay, between brinjal and tomato. Okay, what is the example for protoplasm fusion? The best example which is tried in case of 19th century. बताओ कोई protoplasm fusion. Then embryo culture. This is to avoid post fertilization barrier. एम्ब्रियो तो फार्म हुआ है, उसका डेवलपमेंट नहीं हो रहा है अच्छे से. ठीक है, you can go for embryo culture, you can rescue that embryo and you can grow in the cultural media. ठीक है, grafting, these are the different techniques where you can overcome the wide cross, okay, so that you will get some distant hybrids. Ointment प्रोटोकॉल किसके लिए आंसर बता रहा है? Now the weaves increases even at the end of the head. ऑइंटमेंट किसके लिए बता रहा है? बताओ। टोमेटो प्रोटोप्लास्ट फ्यूजन, ऑइंटमेंट क्या है वो पूछ रहा है हमें। अरे सारे लोग ऑइंटमेंट मैं पूछ रहा हूँ क्या है वो ऑइंटमेंट। वही बताओ ना, इट इज पीजी, ठीक है, पॉलीइथिलीन ग्लाइकॉल। what I have asked is what is the successful story that you should tell tomato. It is not, anyway it is not successful, but they have tried between protoplast between potato and tomato, ठीक है। These are some techniques to overcome this particular barriers. Now what are the role of wild cross? Okay, you can improve disease and insect resistance by crossing from the wild progenitors. Okay, then quality, you can improve the quality in case of cotton, okay, in case of different crops, okay, where you can take some genes which are responsible for quality traits or disease and insect resistance traits from the wild progenitors. If you are taking from wild progenitors, it is normal, it is not a normal cross, it is completely distant hybridization between different genera, different species, okay, adoption, for yield, for mode of reproduction, sometime developing commercial hybrids, okay, we can use this wide hybridization, creation of new species like Nicotiana digluta between tabacum and glaucum, okay. These are some role of wide crossing in crop improvement, majorly for disease and insect pest resistance. Since our wild progenitors are wild relative contains these genes, okay, at this particular time we can't go for simple crosses, you should go for distant hybridization, okay. If I want to talk about some achievements, example in cotton, okay, there are some different varieties which are developed through MCU 2, 5, okay, from the distant hybridization. Hybrids like Varalakshmi, Jayalakshmi, okay, which are interspecific between your Eastern and Barbadans. Napier grass, there are some hybrid Napiers. Evolution of new crop species like wheat, rye, triticale. In tobacco, Nicotiana diglota. In radish and cabbage, that is your Raphana brassica. Between radish and cabbage, okay, these are some achievements. And if I want to talk about the distant hybridization achievements, it's not like the successful achievements, okay. Yeah, yes, in case of experiment, these are achievements, but in case of commercial scale, okay, it's not that much efficient achievements, okay. This is all about your distant hybridization. Now we have completed our special breeding techniques also, some like mutation breeding, polyploid breeding, and distant hybridization. Okay, what are the important thing you should know? Okay, I have covered those things. In case of your mutation breeding, like dominant and recessive, where you have to start your selection. Okay, these are the important things. In case of polyploid breeding, okay, the different thing which is like applications in case of polyploid, auto and allo. Okay, in case of your distant hybridization, pre and post fertilization barriers, how to overcome those things. These are important, not each and everything which is provided in textbooks like BD Singh, Kundan Singh. Okay. In next, now we are left with some genetic engineering approach and some markers and their applications. Okay, and we will see some the latest topics maybe they are targeting nowadays. Okay, what is RDNA technology? It is now, and anyway, before that there will be some biotechnological tools like tissue culture. Okay, your tissue culture covers your embryo culture, ovary culture. These anyway, these are not that much having some deep thing. Okay, it is like self-explanatory topics. You can go, you can just see. Anyway, for our course students, we are already uploaded those classes, okay, whichever I took in the live sessions, and also there are notes which are uploaded for different tissue culture methods. You can go according to that. Okay, apart from RDNA technology, we will discuss what exactly it is. Okay, genetic engineering technology, we will also call it as a genetic engineering technology, where we can do some engineering in the DNA level. Okay, we will cut DNA and we will introduce some DNA. Okay, and we will try to upload those DNA to your plants. Okay, this is one of the beautiful technology. Okay, the recombination will intentionally we will do some recombinant DNA molecule. Okay, recombinant DNA molecule is nothing but molecules created, the DNA molecules created by combining genetic material from different sources. Okay, now we are having some rice genome. For that, we will try to combine some other any bacteria, anything from maize. We'll take some genetic material from that and we will introduce introgress to this particular rice genome. We are just recombining the DNA molecule. That molecule we will call it as a recombinant DNA molecule, and technology will be recombinant DNA technology. In vitro introduction of different segments of DNA. Okay, in vitro means inside the controlled environment. Inside in vitro is inside glass. Okay, we are doing it. It is like an controlled condition. One being the vector, other normally unrelated DNA sequence that are capable of replication in the host cell either autonomously or an integral part of the host genome. What are the steps in case of recombinant DNA technology? Okay, if you want to see the steps, you can see here. First, what we will do, we will isolate genetic material. For example, I am working on wheat, so I will take the wheat genome first. Then I will cut that particular wheat genome, okay, like it is donor. We will, for example, any genome, okay, any genome which is you are having some target gene. I am having some genes which are present in wheat. Okay, I will target those genes. I will cut the complete genome. Okay, then that will be donor DNA. Then I will insert that donor DNA into vector. What is vector? Vector is nothing but self-replicating autonomous DNA. We can use it as a vehicle. Vector is nothing but like a vehicle. Okay, to carry that particular donor segment. Okay, vector we can use for multiplication, rapid multiplication to produce multiple copies, or sometimes vector we can use to transfer from one organism to another organism, or sometimes vectors we can use as a expression. Okay, expression vector. Cloning vector is to produce multiple copies of our target gene. Okay, we will introduce our target gene to that particular vector, so it will multiplicate autonomously rapidly, so that we will get multiple copies of particular our gene. That is your cloning vector. Okay, cloning vector. Next, second one, your expression vector. What will expression vector? We need to express our gene in particular vector molecule, so that we will produce some chemical, we will produce some enzyme from that particular gene in the artificial condition. So you will introduce this gene and you will give some condition, favorable condition. Your gene will produce particular chemical. For example, insulin. We will artificially we will produce insulin by inserting our insulin gene into the vector. So it is expression vector. So then vector will become expression vector. And third one, your expression vector, sorry, your vector will be act like a vehicle. Okay, like from one genome to another genome, we want to transfer our DNA. Can anyone tell which is the example for vector which is used as a vehicle? You know this one, okay? The vector molecule which we were using to transfer our DNA molecule from plasmid. Vectors will contain your plasmids, yeast artificial chromosome, cosmids. Okay, there are so many vectors molecules. Okay, there is particular vector, okay, which is we will use as a vehicle, we will use as a vehicle to transfer gene from one organism to another organism. Plasmid, yes, as I told, yes, we will use plasmid, but there is some plasmid, one plasmid, okay, Agrobacterium tumefaciens. क्या वो प्लास्मिड है क्या? वो उसके अंदर रहते हैं ना, वो पूछ रहा है मैं. एग्रोबैक्टीरियम इज बैक्टीरिया भाई, उसमें एक प्लास्मिड रहता है ना. नेचुरल जेनेटिक इंजीनियरिंग क्या बोलते हैं उसको, प्लास्मिड को? आफ्टर प्लास्मिड वो कुछ वो नहीं आ रहा है वो कमेंट्स में शायद. ओके, रिमी लक्की पटेल आप कुछ लगा रहे हैं, वो रोमन में आ रहा है शायद. टीडीएन की बात है इधर, टीआई प्लास्मिड. ठीक है, we will use this particular TIA plasmid as an vehicle vector. Okay, we will use some vector for cloning purpose. We will use some vectors for expression vector as an expression vector. We will use some vector as a vehicle. Okay, to transfer as a transfer vectors. ठीक है, tumor inducing TIA plasmid. Good. Now the general steps. Okay, now we will introduce that our gene into vector. Then there is the amplification. If you want to do through the PCR or the naturally cloning vectors will be, what you will do, you will target gene introduced into the vector by using DNA ligase. DNA ligase is like a gum. Okay, it will joint this target gene with the vector molecule. Then you will transfer this particular vector into any bacteria or any host cell. Okay, we will transfer into the host cell. We will provide some good condition for that. Then what will happen? There is a multiplication of the several multiple copies will form. Okay, so then we will, after we are introducing into the host, we will select the host cells which are having this particular recombinant DNA molecule. This is recombinant DNA molecule. Okay, after introducing some genes to vector, now it is recombinant DNA, means we are merged two DNA into a single DNA vector. Now we are introduced into the host bacteria. Then we will select which are the bacteria having this particular recombinant molecule. How we will select, which is the different story, maybe during by using selectable markers. Okay, by using selectable markers which are having transformation. Okay, we will select those host cells. Then we will give some congenial condition for those host cells through the bioreactors, so that maybe it will produce large quantity of particular gene product which is responsible. This target gene will produce some proteins, right? Okay, by using bioreactors in the large DNA level, we can produce the chemical. That is your expression. This is the for the expression vector. If it is only for cloning purpose, okay, we will introduce into the host. Then we will select the host recombinant cells, okay, which are having the recombinant DNA. Then we will grow in the large number of copies, so that your particular with as your plasmid is autonomously replicating, okay, wherever it is replicating, your target gene also replicating. So you will get a multiple copies like a PCR. That is your expression vector. Okay, but whenever you want to transfer your target gene into the different species, so for different organisms, then also you can use vector. After this particular step, after your selection of recombinant host cell, okay, then you will use those bacteria, like if it is agrobacterium, then you will just infect those agrobacterium bacterials for the plants, so that automatically that agrobacteria, the TA plasmid, will transfer this particular tDNA to the host plant. Okay, that is as a vehicle. These are the basic things what you should know. We are waiting for that only same, but I think it was getting translated. Okay, no problem. Nathan and Smith. Yes, Nathan and Smith, restriction endonuclease. Which there are different types of restriction enzymes. These are the restriction enzymes. Molecular scissors. Okay, they will cut where there is an this recognition sequence. Okay, they will cut at that particular sequence. We will call them as a restriction enzymes. Okay, type 2 into अरे आप लोग डाल रहे हैं, मैं कुछ नहीं पूछ रहा है, ठीक है, मैं तो कुछ पूछने के लिए जा रहा है, आप लोग कुछ कुछ लगा रहे हैं. सर अपलोड 2022 और 2023 PYQ PDF. ठीक है, नहीं होगा अपलोड, मैं बताता हूँ. ठीक है, दिस इज अबाउट your recombinant DNA technology. What is recombinant DNA molecule? It is nothing but combination of two different DNA molecules. How we will do? By using your molecular scissors and DNA ligase. Your molecular scissors are nothing but your restriction endonucleases. Okay, type 2 restriction endonuclease we will use, since they will cut at particular recognition sequence. Your type 1 and type 3, type 4, they will cut too much distance from the recognition sequence. Okay, or into is the first endonuclease which is isolated from the Haemophilus influenzae. Then what are the genetic transformation? The genetic transformation are nothing but how you can process that particular foreign DNA into the genome of an organism. Okay, it is a process by which foreign DNA is introduced into the genome of an organism, resulting in the alteration of its genetic makeup. Okay, we'll call it as genetic transformation. In this case, anyway, we have multiplied our trait of gene, okay, our targeted gene, into multiple millions of copies by using recombinant DNA technology. Okay, or sometimes we are also use as an expression vector, we are using that particular chemical in the large amount, particular enzyme. Okay, and as a vehicle, as a transfer vehicle also, you can use for a to transfer as a different molecule. It will include in this particular case. Okay, what are the different other transformation mechanism where you can transfer the DNA? Direct and indirect. In case of direct method, physical. What are the physical methods? Gene gun. Then electroporation. Then micro injection. Your gene gun is nothing but particle bombardment. What we will do in this particular gene gun method? Okay, by using high velocity gold particles. Okay, we will coat that gold particles with the DNA molecule. Then we will throw them in the high velocity to the cells, so that the DNA will get inserted into the cells. Then it will go into the nuclear genome. We will use high velocity gold or tungsten particles. Okay. Then in case of electroporation, we will give high voltage. Okay, we will give high voltage to the cells, so that there will be a pores formation in the outer cell membrane. so that they will take up the DNA which is available in the outside. Okay, it is due to voltage manipulation. It is like gene gun are, this is velocity, high velocity, gold or tungsten particle. Okay, what about micro injection? We will use hypodermal syringe. Okay, where that is like 0.5 mm syringe, and we will directly transfer to the nucleus. It is like a syringe. Anyway, you have saw syringe na, when you will get injection. That is macro injection. Okay, but here it is micro injection. Your needle will be too much. That is like 0.5 mm. Okay, this is about your different physical method. What are the chemical method? What are the chemical method for transformation? You can use, yes, boilistic is nothing but again the same. We will call it as boilistic technique also. What about chemical? Which chemical we will use for the transformation? Calcium chloride. Okay. Then any other method? PEG. One is PEG, okay, polyethylene glycol, where again there will be pore formation in case of your protoplast. Then there is an cytosis, endocytosis, so that the DNA which is around the cells, it will get introgressed into the cells. Then calcium chloride precipitate. Okay, that is also precipitate. It will also form endocytosis. That means your cell will take from the outer the DNA which is possible. Yes, the liposome mediated. Liposome. Okay, where the lipids, when we use the lipid molecules, okay, your DNA will be coated. DNA will be coated by the lipid molecule. That lipids will be endocytosed then into the cells. Then your lipid will get dissolved in the cell, within the cells. Then you are like this. This is your DNA are negatively charged. Then your lipid, these are positively charged. Okay, what happens? This will introduce into the cell. Then your lipid molecule will dissolve. Then your DNA will release into the nuclear genome. Then it will get introgress into the cells. That is your liposome mediated thing. Okay, make competent to the cell to take up. Yes, good. Liposome. Good. Next one, Agri ब्लॉगिंग वाला पूछ रहा है सर हाउ टू कवर ओवरऑल प्लांट ब्रीडिंग इन 1 डे. ठीक है, मेरा मैराथन है, तीन क्लासेस है मैराथन. ठीक है, बस आप क्या करना है, सुबह से चालू करो 1 by 1, 6 घंटे का है, शायद 18 आवर्स में कवर हो जाएगा. मैं और क्या बता सकते हैं, एक दिन में कवर करना हो तो बस यही करना है. ठीक है, मेरा मैराथन देखना है बस. कुछ और डाउट है क्या, बताओ. देन वायरस मीडिएटेड are nothing but when you will introduce your gene into virus, okay, whenever your virus will attack bacteria or any organism, it will transfer its own DNA, so that your gene will also get introduced. Then agrobacterium mediated, it is nothing but indirect method. Okay, the natural engineering method where you can introduce your DNA into the plant system. Okay, your agrobacterium tumefaciens contain TA plasmid. In TA plasmid, if you introduce your gene, naturally it will pass TA plasmid into the crop plants to produce tumors, so that your DNA will also get introduced into the crop plants. Okay, Maharashan King Abhi sir, right? So, bus sir, this is what happened. BS sir, this is what happened. क्या बता रहे हैं आप, Agri? Logging wala, batao. Agrobacterium mediated, how it will happen? It is natural TA plasmid which is present in bacteria. Okay. What is plasmid again? Plasmid is one type of a vector. Okay. In vector there are too much like plasmid, cosmid, yeast artificial chromosome, bacterial artificial chromosome. Okay. There is no need to think too much about it. When you will enter into MSc, you will get to know each and everything. Now, what is plasmid? Plasmid is simple self-replicating, okay, double-stranded circular DNA molecule which is present in bacteria. TI plasmid is which is particularly present in Agrobacterium tumefaciens. What? This is the structure of your TI plasmid. tDNA region. It contains left border and right border. Then auxin, the genes responsible for auxin, cytokinin and opine. What will do naturally, your TI plasmid, that like agrobacteria, when it will inflect the cell, root cells, okay, when there is an there is a wound in case of your plant's roots, if there is wound, then there is a formation of acetosyringin. Okay, it will automatically trigger the bacteria to transfer this tDNA region. Naturally, this tDNA region will transfer to plant cells. Then what happens? It will produce tumors. Where there is a tDNA, since auxin, it contains auxin, cytokinin and opine. Okay, so automatically there is a formation of tumors. Means the replication, the continuous division of cells, and it will form the tumors in the plant cells. Okay, that's why you will get a disease. This is tumor inducing plasmid. So what we will do? We will capture this natural mechanism. Okay, we will replace this particular tDNA region, with our gene of interest. So what will do Agrobacterium? Okay, whenever it wants to transfer its tDNA region, it will transfer our gene of interest. That's what we needed. We need to transfer to our plants our transgene, so that we will get some genetically modified organisms. Okay, this is the basic mechanism where we can target. What are other genes? Open catabolism genes also it contains. Okay, origin of replication, it will have origin of replication. That means the autonomous self-replicating DNA. It will not dependent on bacterial nuclear genome. Okay, and virulence gene. These are the genes responsible for this particular tDNA transfer. It contains again your virulence genes like virulence, vir A, vir B, vir C, vir D. It is a complete vir box. Okay, again for you guys there is no need to think too much about it. Okay, it will start from vir A. Okay, then it will it will stimulate vir G. Then all other your virulence genes will activate. So those will be responsible for to transfer this particular tDNA segment. Okay, at this point of time, what you should know. Okay, this is the thing. Okay, it will be around, normally it will be around 25 kb your tDNA region. Okay, this is what you should know. And your virulence genes are responsible for the transfer of that particular tDNA region. Then transgenics. Now we have saw that, okay, how we can create the recombinant DNA molecule. Okay, once we are done with recombinant DNA molecule, how we can transfer that recombinant DNA molecule through the different chemical or physical methods, direct or indirect method. In indirect method, we will use Agrobacterium tumefaciens, that is TI plasmid, so that naturally we can transfer our gene into organism. Then after that, whatever product we will get, we will call them as a transgenics. Okay, introduction of exogenous DNA into the genome, okay, stably maintained in a readable manner. That we will call, that process is we will call it as a genetic engineering process, and the product what we get is transgenic. Okay, before we should know what exactly the transgenic, there are different terms. GMO. genetically modified organism, a broad word covering the all methods which are alter the genetic material. Okay, if you are using mutation breeding, if you are using some distant hybridization or any breeding methods, okay, where you are trying to modify organism, okay, it is broad term, genetic modified organisms. But what about genetically engineered organisms, GMOs? Okay, through the recombinant DNA technology, where you are using recombinant DNA technology to produce GMOs, then it is genetically engineered organisms. Then what about transgenics? This is genetically engineered technology using foreign gene, belongs to different species. Then only we will call it as a transgenics. Okay, your GMO will first come. Okay, broad-based term, broad word will cover each and everything where you are making some manipulation of genetic material. Maybe your normal traditional breeding methods also will come in genetically modified organisms. In mutation, it will come your mutation, polyploid, everything will come. Then your genetically engineered organisms, where you will use recombinant DNA technology. Then you will use recombinant DNA technology for different genes, maybe from the same species gene transfer, different genes, species gene transfer. Okay, where you are transferring gene from the different species, okay, which is not cross compatible. We can't use normal crossing program. Okay, so that's why we are using recombinant DNA technology. Those we will call it as a transgenics. Okay, those organisms we will call them as a transgenics. Okay, golden rice is transgenic. It is transgenic. Since we took genes from bacteria, bacteria is from different species, right? Okay, but where, again, I will tell the three different terms now. This anyway you got it. GMO, then GEO, then transgenics. Okay. Then what about transgenics, intragenics, and cisgenics? Your transgenics, the coding region, the gene you took from any source which is incompatible, sexually incompatible. You are not able to make crosses between among themselves. Like you are taking from different any bacteria or any virus or any other different species. Okay, then you will call them as a transgenic. Whenever you are using the sexually compatible species, like you can cross maize and wheat sometime. You can cross wheat and rye. Okay, if you are taking the gene from rye or if you are taking from the gene from maize and you are transferring for wheat, okay, then you will call the product as a intragenics. Okay, in case of cisgenic, again, sexually compatible in the both the cases. Regulatory sequence. There are some regulatory sequences right, like promoters to promote that particular gene expression. Okay, like in case of your Barnes gene, TA29 promoter. Okay, those are the regulatory sequences. In transgenic, it may be from any source again, like your coding gene. But in case of intragenic, any gene from the sexually compatible. If you are taking your coding gene from the maize and you are taking the promoter from the rye and you are transferring these two into the wheat, then your product will be intragenic. But in case of cisgenic, the promoters and coding gene, they should be both from the same species. Okay, for like same gene, and they should be sexually compatible. Then you will call cisgenic. The difference between transgenic and intragenic and cisgenic, first one is coding gene. If it is from any source, then transgenic, like sexually incompatible. If they are compatible species, then intragenic and cisgenic. And your promoter, if it is any source, then transgenic. Any gene from the sexually compatible, then intragenic. And if it is from the same gene, then it should be cisgenic. Okay, this is the basic differentiation. Okay, this regulation is strict in case of transgenic. It is less stringent in case of intragenic, but in case of cisgenic, it is very much less stringent. Okay, and marker gene may be present here, but in these cases absent. Okay, Marker gene means we will use those genes to select the our gene of interest. Okay, but in case of introgenic and cisgenic, there is a complete absence of that marker gene. ठीक है, ये तो डिफरेंस बिटवीन ट्रांसजेनिक, इंटरजेनिक एंड सिसजेनिक. ठीक है, ये भी पूछ सकता है. गोल्डन राइस इज ट्रांसजेनिक एग्जांपल आर बायोफोर्टिफाइड. आप क्वेश्चन पूछते हैं कि मतलब sometimes you will ask questions in Chan which are like I don't know what I have to say. कोई बता सकते हैं क्या? ये पूछ रहा है गोल्डन राइस इज ट्रांसजेनिक एग्जांपल और इट इज बायोफोर्टिफाइड. इसको आंसर बताओ कोई. बताओ जल्दी. सिंचन पूछ रहा है गोल्डन राइस इज ट्रांसजेनिक और बायोफोर्टिफाइड. उसको आंसर क्या बता सकते हैं, बताओ. बायोफोर्टिफाइड ट्रांसजेनिक. अरे भाई, इट इज बायोफोर्टिफाइड ट्रांसजेनिक क्रॉप. उसमें बायोफोर्टिफाइड किया है बाय यूजिंग ट्रांसजेनिक. क्वेश्चन कैसे, ये एक क्वेश्चन ऐसे पूछ रहा है, आप लोग आंसर भी ऐसे कर रहा है. कोई बता रहा है बायोफोर्टिफाइड, कोई बता रहे ट्रांसजेनिक. यस मधु, बोथ बायोफोर्टिफाइड एंड ट्रांसजेनिक. ठीक है. नेक्स्ट, व्हाट आर द एप्लीकेशंस इन of your genetic engineering technology? I am not covering too much, as again I am telling you guys are not writing plant biotechnology, you guys are writing plant sciences. Okay, in that they will not ask too much about it. Okay, you should know what is the recombinant DNA technology, what are the methods where you can transfer your DNA molecule, and what are the applications. Okay, as I told, in vectors there are too many. Okay, and again in the transfer methods there will be too many. Okay, but what you should know, the basic things. चिंचन, अरे रो रहा है क्या? अरे मैं बस ऐसे मजाक किया था. आप भी मजाक कर रहा है. आप पूछ रहा है बायोफोर्टिफाइड है या ट्रांसजेनिक है, दोनों है ना. ठीक है, छोड़ दो, अभी आ जाओ, फिर से आ जाओ इधर. Application, insect resistance, like your BT Carter, Bolgard 1, 2, 3, I already explained it. Okay, first generation, second generation, third generation Bolgard. Okay, what they will do, they will produce BT crystalline, which is toxin. Okay, when insect will feed that particular leaves which are having that BT toxin. Okay, the cry protein in alkaline condition, it will turns to toxic, so that your insect, the normal gut bacteria contains alkaline condition, so that your insect will feed, it will become toxic, and your insect will it will die, the death of the insect. Okay, this is the basic application. Okay, in India 2002, which is approved for commercial cultivation, your BT cotton one, that is Bolgard one, the only one crop which is commercially approved for the cultivation in case of India. Okay, then virus resistance, papaya ring spot virus coat protein mediated. What they did, okay, they have transferred some genes in the papaya plants, okay, which will produce virus coat protein. When your virus will attack to papaya will, what it will do? It will produce virus coat protein. It is your virus. It contains genome and coat protein. Okay. Then whenever it will attack to the papaya will in turn, it will produce this virus coat protein. Whenever it will try to remove that coat protein and it will infect, it will try to transfer that genome, virus genome into papaya will immediately produce this virus protein. Again, it will encapsulate. There is no encapsulation. Again, it will encapsulate. Then there is no transfer of this particular virus into the papaya. Okay, this is one of the application. Okay, where they have used the genes which are responsible for virus coat protein. Okay, they have developed papaya ring spot virus resistance. Okay, the resistance genotypes, rainbow variety of papaya. Okay, and they, again after this there are so many applications with respect to different disease resistance, virus resistance, for quality, like your as I have already discussed about it. That's why I'm not discussing here it again. Your biofortified rice, that is your golden rice, that is also one of the variety. And your in PYQ discussion we have already discussed about it. Flavor saver. Okay, again that is an transgenic variety. Okay, which is approved, which is released commercially, the first transgenic crop. Okay, flavor saver in 1994 by using antisense RNA technology. Okay, these are the things I have already discussed about it in PYQ and in the different marathon sessions. So I will not discuss about it anymore. What are the biosafety concerns in case of your transgenic? Environmental concerns, gene flow and outcrossing. Maybe your gene which you took from bacteria, maybe it will, whenever you get natural crossing, maybe they will transfer to another varieties. Again, it is a problem. Super weeds and super pest development. Okay, you took the genes from the bacteria, so they will try to get into the normal environment. So it will be, there is a formation of super weeds in case of your croplands. Non-target effects, sometimes they will also form the non-target effects. You are targeting for something and they are giving some different phenotypes. Okay, biodiversity loss, that is also one of the concern. Okay, wherever now we can see, you will try to grow the crops which are tolerant for any particular insect pest. For in that particular process, maybe they are having some linkage drag, and they are having some genes which are also inhibit some beneficial insect. Okay, then whenever the farmer grow that variety, anyway, your BT cotton will avoid bollworm. But if it is contains any other genes which are also affecting the beneficial insect, then maybe loss of that particular beneficial insect from the biodiversity. Okay, that's what the concern. And ecosystem disruption, that's why it will disrupt the ecosystem. What are the human health concern? Allergicity, allergen, toxicity. Maybe when you eat that, maybe there will be some toxicity. Anyway, in our Indian case, we don't have any transgenics. But in case of like in BT cotton, okay, we will use that cake for the feeding purpose sometimes after the extraction of that seed meal. Maybe that time there is a problem. So these are some, and antibiotic resistance and pesticide residues. Okay, these are the concerns with the human health. Socio-economic and ethical concerns like corporate monopoly. Okay, like for example, if BT cotton is very famous, then everyone will try to grow that BT cotton, and it is produced from any one company, for example, Myco, okay, or any other company like Syngenta. So they will take monopoly. It is one of the socio-economic problem. Farmer dependency, they will completely dependent on that particular BT cotton. Biopiracy, okay, it is also one of the concern. If anyone knows that particular gene idea or any particular thing, they can use that particular technology for their commercial activities. That is also again problem. Okay, ethical tampering and your regulatory gaps. In India, there are, that's why we are having, anyway, we are only having one crop, that is BT cotton. Okay, so we are not that much about in the regulatory mechanism. Okay, if any more than so many crops are having so many varieties and everyone is growing, so we should have to target the regulatory mechanism. But since our system only contains one crop, so we are not that much into the regulatory mechanism. So there are gaps. There are so many gaps in case of transgenic. So that we need some guidelines for these concerns. Okay, India's regulatory framework for rDNA research. Okay, there are so many regulatory guidelines to maintain these different things. Recently, if I want to tell, in 2022, the recent regulatory guidelines which are related to your gene-edited crops, okay, they have excluded SDN1 and SDN2. These two from the, they are exempted, okay, from the transgenic. Okay, these two are not transgenic anymore. The SDN1 and SDN2 technology, which are gene editing technologies. Okay, these are non-transgenic. That's why India have released 2 recent varieties. Okay, and those are non-GM, not genetically modified, non-GM rice varieties, which are through the gene editing. And your SDN3 will comes under regulatory framework as a transgenic. Okay. This is all about your recombinant DNA technology. Now we have passed that era also, and there is a growth. There is a too much growth in the markers. Okay, so to avoid those different regulatory mechanism, there will be too much concerns. Okay, now we are into the marker-assisted breeding era. Okay, once we develop the markers, we are using those markers to identify the individuals by using markers. Anyway, without phenotyping, sometimes you can, using markers, you can select the individuals, so we can save the time. We can go crossing in the same generation. Okay, there are so many advantages. Okay, anyway, for you guys, it's not that much needed. You should know some basic things regarding markers. What is markers? It is nothing but your tag or any identity or mark. Okay, wherever you are not able to see the sequence, or wherever you are not able to see the genes, by using markers, okay, you can identify the genes, or you can locate the genes. Genetic markers are known variable DNA locations used to navigate the genome and identify the differences. These are like landmarks, as I told in the video also. Okay, and I have already covered each and everything in the live class. But here, due to the time restriction, okay, I am not able to explain each and everything regarding marker, but I will give some glimpse. Okay, your marker are nothing but the landmarks. Okay, for simple layman language, okay, if anyone of your friend want to comes to your home, okay, you don't know the address. Okay, so what you will tell, अरे वाह इधर आ जाओ, ये इधर एक बैंक है, HDFC बैंक, ठीक है, उसके बगल वाला मेरा बिल्डिंग है, ऐसे बता सकते हैं ना, ठीक है. फॉर एग्जांपल, फॉर इन केस ऑफ Agrict, we are having office, okay, no one knows where it is. If anyone come you guys for Delhi and you will ask sir address, then I will tell, अरे भाई इंद्रापुरी में आ जाओ, उधर हमारा ऑफिस है, ठीक है. इंद्रापुरी में कहाँ है? इंद्रापुरी is one of the area which is located near to the Pusa campus. उधर कहाँ आना है? Then I will tell, okay, in Indrapuri there is an HDFC bank. Okay, before बगल वाला हमारा building है. Beside that HDFC bank, it is our Agri Academy. So what you will do? You will book a cab to Indrapuri. Okay, anyway, no, we are having some live location, each and the location we can easily come to our point, but generally you will land up in Indrapuri, then you will ask someone here, what kaha hai HDFC bank kaha hai? You will come to HDFC bank, then you will see our Agri Academy. Means one who don't know Agri Academy, okay, now we have recently established here. Okay, like that, the gene is Agri Academy. Okay, everyone doesn't know about it sometime. The recently identified or recently developed or any whatever it may be, or it is very difficult to locate. But HDFC बैंक वो तो पहले 20-30 साल से इधर ही है, उसको तो सबको पता है. HDFC bank is a marker. Okay, that's what the definition of marker. It is a landmark. Okay, using those landmarks, okay, what we can do, we can say about our gene. वो तो पूरा नियर है मार्कर, मतलब जीन से उसको सब कुछ पता है, ठीक है. क्या पता है वो जीन कैसे सेग्रीगेशन हो रहा है. Since your marker is also segregating in the same way, your gene also segregate. Since they are like they tend to together, like there is a linkage between them. So wherever your marker is going, your gene also will go. You will know the segregation pattern, and you also will get to know the heterozygous or homozygous, whether it is your gene is heterozygous or homogeneous, and how it is transferring, how it is inheriting, everything will get to know, and your location. Okay, by using your marker location, you can easily locate your gene also. This is the simple definition. Okay, those are like landmark for specific genes. Yes, here HDFC Bank is a marker. Yes, those are landmarks for identifying what genes we want. Yes, okay. This is about DNA. So marker. You can see here. In individual A, okay, it is C. In individual 2, it is having T. Okay, it is polymorphic marker. Okay, using these markers, okay, you can identify the individuals. Yeah, this is individual A, this is individual B, by seeing the basic differences in the single nucleotide. This is SNP. If you are able to differentiate in single nucleotide polymorphism, that is SNP. Then your short tandem repeats, we will also call it as a SSRs. Okay, in one individual, the repeats, the AG80 repeats are 3. In another individual, the AG80 repeats are 5. So you can easily differentiate two individuals. Okay, this is what exactly the marker is. Okay, what is the genetic marker now? The definition, that is layman language. The trait that is polymorphic, we will call it as a trait only. Okay, maybe it is like your morphological trait, sometimes it is biochemical trait, sometimes it is DNA trait. Okay, we will see. Morphological, easily and reliably identified. You can easily and reliably identified and readily followed in the segregation generation. It will also segregate like a normal gene and indicates the genotype of the individuals that exhibits the trait. Okay, it will also indicates what's the genotype, whether it may be homozygous, whether it may be dominant homozygous or heterozygous dominant, or it is recessive. Okay, it will give all other information. Polymorphic exists in the multiple forms within the population. Polymorphic is nothing but it is able to differentiate between the two individuals. That is your polymorphic. What about multiallelic? It enables the classification into multiple groups. Multiallelic, matlab that allele, the nature of that particular marker allele is having multiple copies. Okay, in case of our SSR, if your repeats are 3 year, 5 year, in some other individuals, your repeats will be 6, your repeats will be 8, your repeats will be 10, like the multiple copies, the multiple forms of a marker. That is your multiallelic. And what about codominant? It will distinguish between capital AA and capital AA. Okay, if it is band here in this case, okay, there may be a 2 bands here in case of this particular individual. Okay, this is the how we can differentiate. But in case of dominant markers, okay, you are not able to differentiate between these two. Okay, in the both the cases, you will get a band like this. How we will get bands, what is the difference? There is no need to think too much about it. इसके बाद भी आपको कुछ ज्यादा चाहिए हो तो मेरा मतलब हमारा कोर्स में ये लाइव क्लासेस उसमें आप जाके देख सकते हैं. मैं अभी तो इधर सारे तो कवर करने के लिए थोड़ा मुश्किल हो जाएगा. ठीक है. नॉन एपिस्टैटिक, there should be no interaction between the different markers. Neutral, it will not affect any fitness and it will not affect any individual fitness. It is abundant and evenly distributed all over the genome so that we can exploit those markers. Non-pleiotropic affects only the marker trait. Okay, it should not be pleiotropic effect. It should not control too many functions. Environmentally stable phenotypic reflects the genotype regardless of the environment. Okay, these are the ideal features of a marker. Okay. Then if you want to classify morphological, biochemical, morphological markers are nothing but you can see through your eyes. Okay, for example, Pusa Basmati 1, okay, PB 1 having ons in their grains. Okay, your SD 3086, okay, it contains glooms in case of your wheat, and there is a pigment, okay, the purple pigmentation in case of like anthocyanin pigmentation in some varieties of wheat. Okay, that is morphological marker. By seeing your eyes, you can identify. Okay, and biochemical, which is your protein, like isozymes and allozymes. Okay, those are protein based markers. You can differentiate based on the protein product. Okay, then your molecular. In molecular markers, like hybridization based, PCR based, and DNA sequence based markers. These are the different three types markers which are available. Okay, your DNA based markers are very important. Those are your like molecular markers. Okay, that's why if I want to talk about classification of your DNA markers, then you can see based on chronology, means when they got developed. First generation, your RFLP, RAPD and their modification. Then second generation marker, your SSR, AFLP and their modification. Then after that, now we are having the recent markers, which are your SNP. Throughput, high throughput meaning is nothing but in some of the markers at each step you have to see only, you can see the individual. At a time, you can only see one marker, okay, or one individual. But high throughput, in case of high throughput marker, like your SNP markers, at a time you can go multiple individuals. Okay, at multiple individuals, at multiple marker loci, what is the genotype? That is your high throughput. Okay, Shinchan. इसके बाद भी कुछ मैं बता रहा है ना, मैं if I want to explain each and everything, your markers class will take minimum 3 hours. I have did it for our students. Okay, you can go and watch there. But here I will give some glimpse which are important for exams. If you for basic more understanding, you should attend our live classes. That's only the option. Based on PCR usage, PCR based markers, non-PCR based markers, your RAPD, AFLP, SSR, and non-PCR based marker, that is your RFLP. There is no, in that case, you will not use PCR. Molecular basis classification, your SNP arises from the variation in a single nucleotide, and non-SNPs, which are all other, apart from your SNPs, all other markers are non-SNP based markers. ठीक है? अरे मैं क्वेश्चन पूछ अभी, क्वेश्चन मतलब बस ऐसे बता रहा है, मतलब whoever in this particular attendees, okay, whoever from our courses, the master course students, ठीक है, आप कमेंट में बताओ, मैं मार्कर क्लास पूरा मतलब आपको समझने के लिए क्या क्या चाहिए पूरा बेसिक से कवर किया है नहीं किया है वो बताओ. Since there are so many are asking, there are so many doubts. If I anyway, they will definitely get doubts for the first time who are listening to this particular markers. They will get a doubts. But if anyone from our master course, okay, did you guys got the whole concept in a right manner or not? Batao. So that maybe they will also get something. So that yes, maybe they can go for our courses. Okay. I don't have any problem to cover here, but the thing is time. Okay, at this point of time, if I want to start each and everything from the basic, then it will be like 3 hours. Yes, sir. Yes, sir. Yes, sir. You did. Yes, sir. Thank you. Thank you, Rimi, Beat, Shruti. Okay, Nemani. What have you done, sir? What have you done means I have covered each and everything from the basic. That's what I am asking. It's 3-hour class. Yeah. You ate very lightly. What have you done, sir? कुछ ट्रांसफर होके आ रहा है क्या वो रोमन से आ रहा है मुझे मधु हेगड़े आप what you are telling covered everything Sir, yes. Madhu, you are telling something, but after conversion, it is coming different comment. Beat bones, आपका भी. You ate very lightly. क्या आ रहा है पता नहीं. Yeah, Lucky Patel. Okay. And what are the basis of polymorphism? Okay, the substitution, insertion and deletion. Okay, we can target those things. Your duplication, inversion, translocation. Okay, your recombination, application slippage, altered methylation pattern. Okay, you can target these DNA level aberrations. Okay, to see the markers. Okay, in each marker there are some basis of polymorphism. You can see the first one is RFLP, single restriction enzyme. You will use to generate fragments differing in their length. What you will do, you will take a, for example, individual 1 and individual 2. You will take an individual 1 and individual 2, and you will restriction digestion of that particular DNA. Here there is a restriction enzyme site in this particular individual. Here and here there are 3 restriction enzyme sites, that is recognition sequence. So there will be a 3 cuts. Okay, 3 cuts. But in case of your individual 2, here there is a 1 cut and here there is a 1 cut. Since there is a mutation or any insertion or deletion, that is your genetic basis. Due to that thing, mutation or insertion or substitution, anything, this got lost your restriction enzyme site 2, so that there is only 2 cuts. And you will next, what you will do, you can see here, you digest with the restriction enzyme endonucleases, then apply individual sample to individual wells on the agarose gel. Okay, then they will migrate after the restriction enzyme digestion. They will migrate. What you will do? Then you will denature it in the alkali condition. Then you will apply on a nitrocellular nylon membrane. Okay, the transfer into the membrane now. Then you will design probes. Okay, and add the radioactivity, whatever it may be. Then you will denature it, your probes also. Okay, then you will hybridize those probes into the same probes into the different individual DNA. Okay, this is the different individual DNA. The probes will be same. So that you are having the same probe, everything will be same. Your probe will go and attach here, and in case of an individual 2 also, it will go and attach here, the same probe. So you can see, since it cut has happened here and here and here, so you will get this fragment here, since there is a cut here. But since it is lost in this individual 2, there is no cut here, so it will get attached here, so that you will get this much product. Once you go for gel electrophoresis and southern hybridization. Southern hybridization is nothing but the hybridization with the probe. So you can see, since it is having small fragment, it will move too much further. And in this case, in individual 2, since it is a large fragment, it will be here. Are you getting it right? The difference here, this one, here, this one, based on the fragment size. That's what the principle will tell. Selection restriction enzyme generates fragment differing in their length from the same genomic region of different individuals of a given species. Okay, that's what you can see. You can see here. Okay, there are different fragment length, so that in gel there will be different mobility. This is your RFLP marker. Who developed it and when? Your RFLP. Sorry. बताओ. Yes, 1980 from Boston. Then that is the RFLP marker, and it is codominant. Likh do. It is codominant, since it will differentiate heterozygous and homozygosity. If it is homozygous dominant, it is one copy of your individual. The same copy you will get, the same thing will happen in the homologous chromosome, so that you will get a band. the single band here. But in case of heterozygous, if it is heterozygous, for example, okay, what will happen? There is one copy is like this, one in one homologous chromosome, in another homologous chromosome, it is like this. So you will get both the bands. In case of your, it is capital A. In capital A, small a, you will get this band also. Since it is capital A loci, it is, if you consider this as a small a, then you will get small a, this segment also. So you will get two product, one is here, one is here. This is what exactly the codominance in nature. You can be able to differentiate between two, homozygous dominant and homozygous, sorry, dominant homozygous and dominant heterozygous. Okay, this is about your codominant marker. Next, the PCR. After this, in 1984, there is a discovery of invention of PCR by Carey-Mullis. What you will do by using primers, from using forward and reverse primer, okay, we can artificially, okay, go for amplification of DNA of particular target region. This is our target region. So by using primers, okay, by using the primers, there are three steps: denaturation, annealing, and extension. By using modification of temperature, you can amplify this particular target region in multiple copies. This is what the principle of PCR. By using this principle, okay, the second marker, it got developed for RAPD in 1990, okay, by Williams et al. Okay. What exactly about the marker? It uses short arbitrary primers to amplify the random segment of the genome. For example, individual 1 and individual 2. We will use primers. Okay, for example, like we will use only single primer. It will act as both forward and reverse primer. So you can see it will attach here and here, here and here. And in this case, it will attach here, here and here. After the amplification, you can see it will amplify this also here, it will amplify this. Since there is no particular attachment here, here and here, so it will not get amplified, this particular region. If you take the profile, you can see profile will be like this. In A, there is two, they are got amplified. In this particular loci, it is only one amplified. Okay, when we will particularly see only the, when we will particularly see this region, okay, you just neglect this. Okay, in this particular individual, there is an amplification since it got attached in for both forward and reverse. But here only there is no attachment. Your primers, since your primer binding sites having some mutations or any deletions, again additions, okay, your primer will not going to bind. So there is no amplification of this particular thing. Amplification and no amplification. Okay, presence absence. You can see this one. Your A. Here in case of A, it is presence. In case of B, no presence. Okay, that's why it is dominance in nature. Okay, it will not differentiate. For example, capital A, capital A, capital A, small a. In this particular case, okay, there are two homologous chromosomes. Your primers will attach here. It is capital A. In homologous chromosome, again capital A. So again your primer will go and attach. So you will get a band. You will get a band. But in case of capital A, small a, one homologous chromosome, okay, your primer will go and attach. And in another, that is small a is like there is no, since mutation and something, your primers will not go and attach, you will not get a band. But the band is coming from this particular homologous chromosome. So you will get a band. You are not able to differentiate between these two. But in case of codominant, you can see, as I told, you can differentiate. Okay, differentiate between capital A and capital A, small a. You will get two bands and single band here. But in case of dominant marker like RIPD, okay, we're not able to differentiate between these two. That's what the dominance nature, dominant marker. Okay, लिख दो, सब लिख दो, ठीक है. Your RFLP is codominant, your RFPD will be dominant marker. Okay, this is about RFPD. Then SSR. SSR nothing but simple sequence repeat. In individual one, okay, there are region AT, 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 AT. In case of individual B, the repeats will be AT, AT and AT. When you design some primers, okay, like forward primer and reverse primer, okay, they will amplify this particular repetitive region. So you will get this particular region will get amplified. Okay, particular region will get amplified. Since it is containing 5 repeats, 10 base pairs, your size will be 10 base pairs. In this case, you will use forward reverse and primary reverse. Okay, your repeats will only having 6 base pairs. It will amplify 6 base pair product. Decay in individual 2. The simple sequence repeats are also known as microsatellites, or short repetitive DNA sequence, usually 1 to 6 base pairs repeats, okay, that are randomly repeated specific location throughout the genome. Okay, we will target those regions by using primers. ठीक है, अभी देखो, there is a size difference due to tandem repeats. This is your SSR marker. Again, it is codominant. Again, it is codominant. You can easily differentiate between two different homozygous dominant and homozygous heterozygous, sorry, dominant heterozygous. Okay, you can easily differentiate between these two. And also it is multi-allelic. Multi-allelic. Why it is multi-allelic? In some individuals it will give 10 base pair, in some individuals it will give 6 base pair, in some other individuals maybe repeats will be 12 base pair, 14 base pair. There are multiple alleles you can see. That is your SSR. ठीक है, समझ गया ना? Little and little in 1989. Good. Okay. Then ISSR. In this case, we have targeted repetitive region. What we are here will do? We will target the sequence between the repetitive region. This is your repetitive region 1. This is your repetitive region 2. We will target this intermediate region. We will try to amplify this region and we can see the differences. Okay. Here in this case, you will use this repetitive region as a primers. Okay. In some individuals, you will get your primer will go and attach and you will get a amplification product. In some individuals, due to the difference in this particular repeats, you will not get, in individual 2, you will not get an amplification. Again, it is presence or absence. So it is again dominant marker. Dominant marker. Okay. DNA fragments located between the adjacent or oppositely oriented microsatellite. You can see the amplification product. Okay. This is about your ISSR markers. अरे आपको ना वो सारे नहीं पूछता है, कौन डिस्कवर किया है ISSR, SSR, ठीक है, सारे सिंचन used in DNA fingerprinting. You can use all the markers in DNA fingerprinting. Okay, but SSR are mostly used DNA fingerprinting marker. Okay, but in the beginning, Alex Jeffrey, okay, he actually used VNTRs, okay, variable number of tandem repeats and RFLP markers in the beginning. Okay, this is your ISSR. If you want to take an differentiation, if you want to see the difference between these two, okay, region amplified microsatellite in case of SSR, in case of RAPD it is random, we don't know from where it is coming, the random amplification. ISSR region between the microsatellite. Primer, forward and reverse primer we will use. Here we will use only one single primer, it will act as both primer, forward and reverse. Here also we will use only one primer, which is repetitive sequence. The primer length 18 to 24, these are short, usually decamer, that is 10 base pair, and 18 to 24. The stringency, annealing temperature stringency will be high here. That means we only need that particular region amplification. Okay, we don't need any mismatch in case of primer. But in case of RAPD, we will give low stringency. so that it will go and attach wherever it want. Okay, so that we will get some amplification. It is completely random. Here also again high stringency. Okay, the low stringency will be in RAPD annealing condition. That is high stringency, low stringency. Number of bands, single band. For each, from each specific region, there will be a single band for each primer set. But you can see there will be many number of bands. In case of RAPD, they will go and attach in numerous space, numerous locations, so that you will get a so many number of bands. In case of RAPD, ISSR are again many bands. Reproducibility, highly reproducible. Okay, if you use the primers once again in the same individuals, again you will get the same result. It is highly reproducible result. But in case of RAPD, no. Once you use the primers, you will get one kind of result. Then again, if you use the same primer, then maybe due to the different technical barriers or technical glitch, again you will get a different pattern of banding pattern. So that it is not highly reproducible. It is poor reproducibility. Okay, again it is your ISS also poor. Sequence information for primer development, you need sequence information. In case of SSR, okay, we will target flanking regions of your repeats, so that you should know the sequence, so that you can design your primers. But in case of no need of primers, you will just use any 10 decamer primer to amplification. In case of ISSR also, there is no need for your sequence information. You will just use any repeats as a primers and you can go for amplification. Okay? कोई doubt, कुछ doubt? Then your AFLP, it is somewhat advanced. It is like a combination of your RFLP plus RAPD. It is combination of these two. Okay, what we will do first, we will do the restriction digestion. So we will get 3 different types of product here. Since we will use 2 different restriction enzymes, okay, we will get different fragments. What we will do now? We will do the adapter ligation. Adapter ligation means at the end of each product, we will attach some the nucleotides which we know. So we will use as a adapters. Then anyway, now we know this adapter sequence, so we can easily design the primers based on this particular information, sequence information. You will just use the primers and specifically you will amplify. Out of these different fragments, you will try to amplify only selected nucleotides, selected fragments. That's why we will call it as a selective amplification. Then you can see in the gel which are the products you have amplified. First restriction digestion, then adapter ligation, then selective amplification. Okay, restriction digestion, then adapter ligation, after that selective amplification. This is the procedure. It is a mixture of RFLP and RAPD procedure. Okay, it is dominant marker, presence, absence of the band. Okay, this is all about your AFLP marker. Next, SNP. As I told, variation in the single DNA bases. Okay, at specific location in the genome. Okay, for example, if I compare my DNA with any one of you, okay, if I compare with the Shinchan DNA, there you will get, okay, you will get millions of changes in single nucleotide base. Okay, we will target those things. That we will call it as a SNP. In case it is my DNA, it is like AT, A, G, C, A, T, G, in any location. It is Shinchan's DNA. Okay, E contains A, T, G, G, C, A, T, G. Okay, you can see at this particular location, my DNA is having A, Shinchan DNA is having G. Okay, we will call it as a SNP, single nucleotide polymorphism. We can also tell this is a mutation. Mutation भी हो सकते हैं ना? Okay, your SNP are nothing but mutation. But when you will call it as a SNP? Anyone? Okay, it is mutation only, but when we will call it as a SNP? बताओ 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 जल्दी खत्म करेंगे. When we will call our SNP as a mutations, we will call it as a SNP. When we will call SNP, okay, when it occurs at specific location array more than 10⁶ frequency. No. Mutation will be like 10⁻⁶ frequency. Okay. No, not Rimi. I have told this one in our live classes. बताओ कोई बताओ जल्दी. अभी बताओ सिंचन. बताओ आप बताओ. गिफ्ट मिलेगा तुझे. ओपन अनाउंसमेंट फॉर यू. किसी को मतलब ये क्वेश्चन को गिफ्ट मिलेगा. Okay. As I told, the single change in the any genome, for example, A, here in my DNA it is A, in Sinchan DNA it is G. Okay. When I am calling it as a SNP, it is mutation also. Here A it is G, I will call it as a mutation. But when I am calling it as a SNP, there is something, there is some rule. R also Sinchan 1%. क्या 1% क्या है? Biallelic 1%. बताओ जल्दी बताओ, सही से बताओ. What 1%? Found universal and abundant. Yes, it should be universal, and yet yes, it should be abundant. But there is some particular cutoff for that. सिंचन कुछ बता रहा है 1%, सही से नहीं बता रहा है. सिंचन और कोई? Yes, 1%. What is 1%? That's what I am asking. 1% variation in the population. Then what it is? Then what it is? It should be present in 1% of the population. Then yes, beat bounce occur in more than 1% of the population. Okay, then we will call it as a SNP. Okay, for example, it is my DNA. Abhi, this is Shinchan DNA. Okay, it is beat bounce DNA. It contains A, T, E contains A, T, and G, C, A, T, G, and someone's DNA. For example, it is Madhu DNA. E having A, T, A, G, C, A, T, G. Like that, there are number of DNA. You can see at this particular location there is a variation. 2 will contain A, 2 will contain G. Like that, if I take out of 100 individual at that particular location, okay, if some 30 person contains A and 70% people contain G. Okay, then we will call it as a SNP, single nucleotide polymorphism. But out of 100, okay, out of 100, 99 people contains A, only one person contains G. For example, out of 1000, only 10% contains A, and the another 990 people if they contains G, okay, then we will call it as a mutation. Mutation is a rare event. Okay, out of some 10,000 individuals, if any one or two contains, okay, if you calculate less than 1% the frequency, then it is like mutation, rare event. But if it is more than 1%, okay, then you can consider it as a SNP. Okay, वो तो basic phenomenon. Okay, this marker system is yield reliable, reproducible, okay, and amenable for automation. You can use automation, automatic techniques, and high throughput. High throughput is nothing but at a time you can go for multiple samples at multiple genomic locations. Okay, as I told, 35K chip at a time. For example, in case of wheat, we have 80K chip. That means at each genotyping, each individual genotype, in 80,000 location, you can see what's the DNA base pair at 80,000 location. That is your 80K chip, 80K SNP chip, we will call it as. Okay, at a time, you can see at 80,000 location what is our nucleotide base pair, what is our SNP genotype. Okay, this is all about your SNP. Okay, this is about markers. What are advantages of marker? They will not ask you guys. Okay, if they ask also, what are the advantages of markers? You can use this marker for indirect selection of a trait. That means when you know the genotype of a marker, you can easily tell the trait genotype. Okay, you are having the capital A marker is, you are getting the marker is capital A, then maybe your genotype, the trait you are looking for, if it is linked with that particular trait, then it is also AA. For any, if it is linked, okay, you can use these markers for indirect selection. You can go for marker-assisted selection, marker-assisted backcross breeding. You know the backcross breeding program. By using markers, okay, you can fasten that process by using indirect selection. Okay, foreground and background selection. These are all not for your exam. Okay, they will not ask that much deep. Okay, but if they ask, you should know the, okay, that basic thing. Okay, application, indirect selection of a trait. Okay, where you can, sometimes phenotyping selection will be very difficult. Okay, there you can use markers. Okay, you are having some resistance gene in your crop plants. Due to the disease absence, when there is no disease, or there is no favorable environment for disease, your disease will not get developed at that particular time. You can use the markers. Your markers are linked with that particular resistance gene. Your markers are positive. Maybe you can tell that your resistance genes also present in your crop plants. You can select in the season. There is no need to wait for the disease. Okay, and sometimes some traits will occur, the phenotypic traits will occur after flowering. In case of maize, okay, for example, the yield, you are having some, for anyway, yield is quantitative trait. Okay, any trait which are post flowering, you can see only after flowering the traits. Okay, when you are want to do the selection, okay, you will wait for flowering and you will see the trait. Now flowering is already happened. If you want to cross those crops which are having the positive traits, you can't do in that particular season. You have to go for next season to cross those selected individuals if it is post flowering trait. But if you use the marker, okay, before flowering itself, you will get to know the positive of the trait. So in the same season, you can go for crossing. There is no need to wait for the next season. Like, these are some advantages of the markers. Okay. Most of the markers present in the non-coding region. ऐसे हम नहीं classify कर सकता है सिंचन. ठीक है. There will be a, we will call some as genic SNPs. In case of SNP, there will be a SNPs which are in coding region itself. We'll call them as a genic SNPs. Okay, but we are not able to find those SNPs which are present in the genes. Okay, which are markers itself will act as a genes sometimes. Okay, we are not able to find those genes. That's why we'll use the non-genic markers mostly. Okay. Then we will discuss the last part of your particular session, which is CBD, okay, and your NBA and ITPG RFA. Since they are asking questions from this particular concept, okay. What is CBD? That is Convention on Biological Diversity, Earth Summit in Rio de Janeiro in 1992. It is officially they have accepted it and they started this approval. Then it came to force in December 1993. Currently 196 nations are parties of this convention and India also one of the signatory member. Okay, core objectives to conserve biological diversity and sustainable use of components of biological diversity, then fair and equitable sharing of benefits arising out of the utilization of genetic resources. Okay, the recent acting locally for global impact is the recent edition, 2026 edition. Okay, that theme of the International Day for Biological Diversity. Okay, what are the objectives? Conservation, sustainable use, and fair and equitable sharing. Okay, next. Among 42 articles, Article 8, 9, 10 are important. 8 is in situ conservation. It will tell us about in situ conservation. Your 9 will tell us about ex situ conservation. Then sustainable use of components of biological diversity. Okay, prior informed consent and mutually agreed terms are legal requirement for anyone wishing to access or use of genetic resources in some other countries. Okay, they should have prior informed consent. Okay, to that particular relation, you should be priorly informed and there will be mutually agreed terms. Okay, so that you can access the genetic resources. Next, there will be two protocols under your CBD. One is Cartagena Protocol, which is for biosafety. It was adopted in 2000 and entered into force in 2003. International agreement which aims to ensure safe handling, transport and use of living modified organisms resulting from modern biotechnology that may have adverse effect on biological diversity. Okay, to regulate those LMOs, okay, between the countries, okay, there is a protocol, Cartagena Protocol. There is an advance informed agreement is the most important legal procedure. There is a biosafety clearing house. It is nothing but it is an online platform or software where countries share the information about their biosafety laws, risk assessment and decisions. Okay, they have already uploaded on this particular website or software. That we will call it as a biosafety clearing house. Next, Nagoya Protocol. It is for access and benefit sharing. It is also comes under your CBD, adopted in 2010 and effectively came into force in 2014. International agreement which aims to access genetic resources for fair and equitable sharing. Access, prior information, prior informed consent again, and mutually agreed terms. These are the two important things. For access, you should have prior informed consent, and for benefit sharing, there should be a mutual agreement terms. Again, for this access and benefit sharing clearing house, again it is a platform, online platform or software for exchange information on how we can access the genetic resources and what are the benefit sharing between the two countries. Okay, this is about your Nagoya Protocol. Then the Biological Diversity Act, which is in case of India, okay, in 2002. It came into force in CBD entered into force in 1993 and sovereign rights of the states over their biological resources were reaffirmed. That means after CBD entered into the force in 1993, they told to the member countries to make its own their rules for sovereign rights of their states over their biological resources. Okay, since India is being a signatory to the CBD, introduced the Biological Diversity Bill in the Parliament. It passed in 2002, BDA 2002, and came into force in 2003. What are the acts which aims to again conserve India's rich biodiversity and promote sustainable use of biological resources within the country? and ensure fair and equitable sharing benefits arising from the use of biological resources and their knowledge. There will be a three-tier system in case of India. It should be starting with National Biodiversity Authority, which is located in Chennai. Okay, NBA, national level, state level, there will be a state biodiversity boards, SBB. Okay, they will take care about their state genetic resources. Then biodiversity management committee at the local levels. Okay, under each states regarding the different local heritage, there will be a biodiversity management committees. They will look after the local genetic resources. It will be three-tier structure in our Indian case. Okay, under the National Biodiversity Act. The role of your NBA will look into the complete rules and regulation. In case of India, they will formulate, they will try to try to see what exactly happening all over India with respect to the benefit sharing, with respect to access. And if any foreign authority or any foreign body, if they want to access Indian genetic resources, then they should come through National Biodiversity Authority, which is located in Chennai. As I told in the previous class also, if you want to export anything, okay, for import, it should be through NBPGR. If you want to export anything to any other countries, okay, and if they want to import other countries, then they should go through National Biodiversity Authority. Then what is the role of SBB? They can give suggestions for NBA. Okay, they can give some inputs from their state level, okay, to implement any rules and regulations. And also they are having the rights, okay, to identify some the biodiversity rich sites, and they can name it as a heritage sites. They are having that particular right. And what about BMC? They will act as local level, okay, and if anyone wants to access the local rural areas for their biological resources, then they should get an approval from those local BMCs. Okay, this is all about your NBA. Then ITPGRFA, this is the one of the important act again. Okay, it will be completely target food and agriculture. Adopted in 2001 and entered into force in 2004 under the FAO. Okay, under United Nations FAO. What are the objectives again? Again, the objectives are same: conservation and sustainable use for food and agriculture, fair and equitable sharing of the benefits arising out of their use, harmony with CBD for sustainable agriculture and food security. These are the objectives. Okay, as I told, these will be mainly target for the food and agriculture. What are the key feature? Multilateral system. Okay, in case of your CBD, that is bilateral system. Okay, that means there is one importing country and exporting country. They will talk about it, and they are having their rules and regulation. But in case of ITPGRFA, it is multilateral system. Okay, whoever which are member for ITPGRFA, okay, it will be like there is no need for rules and regulation for each bilateral country. Okay, you can access all other countries' resources with the proper permission. Okay, it is multilateral system. It creates global pool of genetic material for 64 most important crops. They have targeted these 64 crops. They have included in Annexure 1. And standard material transfer agreement. A uniform contract that governs how these seeds are exchanged, ensure that if a commercial product is developed from these resources, a portion of profits goes back to the global fund. Okay, that is standard material transfer agreement. Farmers' right article, that is 9. They will also cover farmers' right, protection of traditional knowledge, to right to participate in the decision making at the national level. They will give an option for the farmers. They will give some involvement. The right to equitable participate in sharing benefits arising from the utilization of plant genetic resources. Okay, in this case, mostly importantly, they have targeted farmers' right. Okay. Then benefit sharing fund, the money collected from the commercial use of seeds invested in projects worldwide that helps the small scale farmers to conserve the diversity of the field. by benefits which is arising from the particular exchange, okay, they will convert those into different projects, okay, where everyone can get benefit. Okay, next one, 64 crops, as I told, food crops, 35 food crops, 29 forage crops. It includes the different food and forage crops, but there are some notably missing: soybean, tomatoes, and sugarcane. Okay, they were originally kept out due to their political and commercial disagreements during negotiation. Okay, these are the 3 crops which are left out from this annexure 1. And total crops 64. Okay, it includes 35 food crops, 29 forage crops. अरे फिर से ले लो कोर्सेस, ठीक है, जरूरी तो नहीं है फिर से एडवर्टाइजमेंट करने के लिए, आपको सब पता है, ठीक है, आप देखो क्या कर सकता है, ठीक है, वो आर प्रिपेयरिंग फॉर नेक्स्ट ईयर, ओके, डोंट मिस इट आउट। ओके, मैराथन में आप सोच सकते हैं, मैराथन में तो ऑलरेडी मिल गए सारे, अरे भाई बस देखो इतना दिया ऑलरेडी। मैराथन में मैं ऑलमोस्ट लगभग every niche marathon I covered took some 100 slides and 300 slides, it's completed. In case of our courses, it will be more than 1500 slides, and from the different mentors again, and they're having some different knowledge and they're having some particular specialization, so you will get a lot of information in live classes. And again, it is one-to-one interaction. Okay, you can have a chance to live discussions also. This is all about our courses. This is about plant breeding marathon. Okay, part 1, part 2 and part 3. I hope I covered each and everything. Okay, whichever missed from my 3 part, that means that are not that much important. Okay, as I told, I sit and I saw the syllabus and PYQs. Based on that only, I targeted different concepts. Okay, whatever which are missing, like they are like minor one, and without knowing me, if they are missing anything, okay, you just go back and see those things in your revision timing. Okay, and other thing which are left like PPFRA and your patents and the seed chains, how it will release, it will be, everything will be covered in your seed tech marathon. Okay, anyway, it is related to seed tech, so that it will be covered in the seed tech marathon. ठीक है, अभी कुछ, इट्स अमेजिंग सेशन, थैंक यू सर, वेरी वेरी यूजफुल सेशन, थैंक यू सर। ठीक है, मैं अभी ऑलरेडी बता दिया, ठीक है, क्या करना है, आप कमेंट डालना है वो न्यू कोर्स लॉन्च वीडियो में, सो दैट एवरीवन, इट विल रीच ईच एंड एवरीवन ऑल ओवर द कंट्री, सो दैट दे विल गेट बेनिफिटेड। ठीक है, और मेरा वीडियो में तो कमेंट तो लगाते हैं आप लोग, मुझे तो उसको मतलब मुझे तो सेटिस्फाइड है मैं। ठीक है, उसके बाद कुछ बताना हो तो बताओ। अभी मैं तो खत्म कर दिया सारे प्लांट ब्रीडिंग का ईच एंड एवरीथिंग। आई थिंक इट विल बी वेरी हेल्पफुल फॉर योर एग्जाम्स। ओके, एग्जाम्स के बाद देखेंगे कितना क्वेश्चन आएगा, मतलब वो तो मैराथन से कितना आएगा, हमारा कोर्स से कितना आएगा, हमारा मॉक टेस्ट से कितना आएगा, सब कुछ देखेंगे। ठीक है, और कुछ बताना चाहिए, और कुछ बताना हो तो बताओ, नहीं हो तो हम कंक्लूड कर देंगे। इट इज लाइक 5 hours 36 अरे ठीक है, कैन वी कंक्लूड अबाउट मैराथन ऑफ पार्ट 1, पार्ट 2 एंड पार्ट 3? ठीक है, फिर मिलते हैं। ठीक है, बेस्ड ऑन व्यूज, बेस्ड ऑन कमेंट्स, ओके, if it will definitely boost our confidence, so that we will come up with different concepts and different updates, so that everyone will get benefited. Okay then, thank you so much. शेयर करो सारे ये वीडियो, लाइक करो। ठीक है, कुछ कमेंट से तो डाल दो। तो as I told, it will be like our fuel for our further investment of our time. ठीक है, thank you so much. Then good night and all the best for your ICAR PG 2026 exam. ठीक है, we will meet in the upcoming classes. Bye bye.